Please join me in welcoming Dr. Fauci.
Thank you very much. It's a pleasure to be here with you today. The idea that we are now a few days away from a new administration, there is no question that there will be a challenge to the coming administration in the arena of infectious diseases, both chronic infectious diseases in the sense of already ongoing disease, and we have certainly a large burden of that, but also there will be a surprise outbreak. 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 Outbreak.
The common term bird flu is for the H5N1 virus that was first picked up around 1997. And public health people have been watching this extremely carefully since that time, always concerned that that would be the next influenza epidemic. It kills about 60% of those who it infects. It kills about 60% of those who it infects. However, it has not been able to continue through human transmission. It has not been able to continue through human transmission. In other words, it has not developed the ability to go from person to person to person to person. It's a very serious disease, and we're hoping that it doesn't obtain the ability to be easily transmitted. We're hoping that it doesn't obtain the ability to be easily transmitted.
Gain-of-function research involves creation of pathogens that are more transmissible or deadly than naturally occurring strains. While this kind of research can have important public health benefits, it can also pose serious risks regarding biosafety and biosecurity. In 2014, the US government called for a deliberative process to determine what policy regarding the funding and conduct of gain-of-function research should be. In the meantime, they imposed a pause on gain-of-function research involving influenza, SARS and MERS viruses, SARS and MERS viruses, SARS and MERS viruses, and they imposed a pause on gain-of-function research involving influenza, SARS and MERS viruses, and commissioned a risk-benefit assessment of this kind of research. As part of the deliberative process, the US National Institutes of Health, NIH, also commissioned the Monash Center of Human Bioethics to produce an ethical analysis white paper. The centre conducted a review of the ethical literature and analysed the existing ethical and decision-making frameworks relevant to situations involving risk and uncertainty. The white paper develops a new framework to indicate where any given study would fall on an ethical spectrum, a useful guide for actual decision-making. The white paper was tabled at the White House and will help lift the US bloc on important gain-of-function research while keeping us safe.
Another really popular topic in the science of infectious disease right now, and in the popular literature, are what are so-called gain-of-function experiments. And so this has come up several times in several questions in the forum. And so this is research that adds functionality and in fact oftentimes increases virulence of existing pathogens, increases virulence of existing pathogens, increases virulence of existing pathogens to allow us to study them and study the characteristics they have when they're now worse off, right? And study the characteristics they have when they're now worse off. They're now worse off, right? But so many of us can immediately, from a sort of sci-fi standpoint, imagine what the costs and the fears associated with these experiments are. But so the important question is, what are the potential benefits are there for these? And if there aren't any then perhaps we shouldn't be doing them. But we need to understand what the potential benefits are if we're going to have a reasoned discussion about whether or not these experiments should continue.
So maybe not everybody have heard about it. I think there's been a lot of discussion about it, but I think one of the big issues that precipitated this discussion was it 2 years ago when there were two different labs that genetically engineered the H5N1 influenza virus, the highly pathogenic virus. When there were two different labs that genetically engineered the H5N1 influenza virus, the highly pathogenic virus that killed most of the humans that were infected, but never transmitted from human to humans. Killed most of the humans that were infected, but never transmitted from human to humans. And then they engineered these viruses. That they showed in ferrets could transmit from ferrets to ferrets, which is oftentimes a good model for transmission among humans.
However, it has not been able to continue through human transmission. In other words, it has not developed the ability to go from person to person to person to person. And we're hoping that it doesn't obtain the ability to be easily transmitted.
I think there were two scientific studies that were embargoed for 6 months or so from being published because nobody knew the risks associated with this, and so that's just a clarification of an example of what we mean by the gain of function.
I think that example is really important because we as a scientific community don't know what we're doing. It's really important because we as a scientific community don't know what we're doing. So it's really important to. It's true. I know. There was recently the formation of the Cambridge Working Group. Marcel Salaté is part of that, and they gathered together a whole range of scientists to figure out what might we do, because we really don't know whether labs should work on this and how, to what extent government authorities should step in and mandate that this work shouldn't be done, because it is absolutely clear that it's very dangerous work.
So there clearly uncertainties, but what are the potential? Why do this in the first place?
There's a long history, and the rationale is pretty simple. If you can identify a specific part, or say a gene in an organism that is responsible for disease, then you actually have a target that you can aim for therapeutics. Okay, and so the classical experiments, and this goes way back, is as long as we've had molecular biology or could do inbreeding of some sort, is to just take a genome and swap genes in one at a time. And see which ones give you the phenotype of disease. And that's how we identified a lot of these virulence factors. And that's what's led to a lot of the therapeutics. And so, that's sort of the background in which these can be beneficial. The transmission experiments get you into another realm because they're really changing the biology of the virus. They're really changing the biology of the virus of an organism. In one case, you're just asking, we know we have a bug that can cause a specific disease. What part of it causes that disease? In this case, we're trying to confer a new phenotype.
So my take on it was perhaps slightly different, and or maybe not, in that those experiments were really asking the question, is it possible for this virus, which occurs in nature, to actually become one that transmits between individuals? Nature does these experiments on a routine basis, on a daily basis, 1,000 times a day probably within us. And so in essence, what these scientists, those two groups, were asking was, can you do that? Or is it even practically possible for this virus to evolve to that state? That's really important information to have. And without that information, we won't be preparing appropriately for that. The embargo was actually motivated more from a sense of, can you provide this information now to certain bad actors who will then engineer this virus to do things that it shouldn't be doing?
Right.
Can you provide this information now to certain bad actors who will then engineer this virus to do things that it shouldn't be doing? Who will then engineer this virus to do things that it shouldn't be doing?
Right.
And so those experiments were already done. They were approved experiments. People went through, but done in very safe settings, with very highly reputed groups. So there was really nothing wrong with the experiments themselves. It was really how the knowledge would be used by bad actors. It was really how the knowledge would be used by bad actors.
During World War I, hundreds of thousands of people were killed or maimed by chemical weapons. After the war had finally ended, it seemed certain that this would be the future of warfare. But those who had seen the horrors wrought by chemical weapons worked hard to prevent their future use, destroying 97% of the world's stockpile. As a result, the world was made a safer place, with the freedom to follow other pursuits. Chemical warfare was a frightening new idea, even at its inception. But not all ideas appear sinister at first glance. Wartime technologies are often extensions of peaceful technology that already exists. This is an example of what is commonly called dual use. When new technologies are developed, we consider the many ways we intend to use them. But it is more important to consider the ways that others might use them, or even misuse them. Over the last 30 years, genetically modified viruses have successfully been used within contained laboratories. Soon, they could be used as a common therapy to treat cancer and a wide range of diseases. Genetically modified viruses are normal viruses that have had extra DNA added to them. This allows them to do things they could not have done before. Until today, scientists have not intentionally released genetically modified viruses into the environment, barring several tightly controlled exceptions. The problem with any deliberate release is that, in many cases, it is difficult to control where the viruses will end up. We know of thousands of natural plant viruses, but none are known that can edit a plant's chromosomes. However, man-made viruses can be created to edit chromosomes across species. A new program called Insect Allies aims to use this kind of gene editing technology to help farmers and communities protect their crops. Until now, this technology has only been used indoors, but the military agency funding this program is interested in releasing it outside. While their idea is not new, it has never been done before. Farmers spend a lot of time worrying about how to protect the health of their crops from hazards. With Insect Allies, armies of insects would be infected with man-made viruses that are designed to directly target their crops. These viruses would be used to strengthen the plants against a wide selection of hazards. As a result, the main focus of this project is on viral dispersal. The Insect Allies themselves would only serve as viral vectors. For this plan to succeed, scientists would need to be prepared for anything. When the alarm sounds, they would need to quickly infect insects with the correct virus, transport them to farmers, and release them. The insects would then be able to distribute their infections to crops. To be useful in any capacity, all of this would require global infrastructure. It would need warehouses designed to house insects year-round and staff to breed and maintain them. These insects would also require safeguards that would prevent them living and breeding beyond their one-time use. Without these kill switches, there would be a risk of the virus mutating and spreading, as pathogens tend to do. Unfortunately, it is still unclear how the spread of a genetically modified virus would be controlled. Should we be worried? The designers of the Insect Allies program claim that they can greatly assist farmers in protecting their crops, much to everyone's benefit. However, this kind of technology can easily be used to sabotage and control access to resources on a local or global scale. Right now, scientists are hard at work to safely develop insect allies. They are using multitudes of kill switches to prevent unintended outbreaks. But is this enough? With the appropriate resources, it would be easy for anyone to turn off these safeguards and cause a deliberate outbreak. This would create a new and highly targetable form of biological weapon, one that is undetectable and untraceable. It can be hard to predict the future. Today's technologies can become tomorrow's weapons. So, knowing all of this, do we really want to go viral? And what can we do to control its possible use?
Scientists are raising the alarm over a Defense Department research program that aims to use bugs to protect crops. The program in question is called Insect Allies, and it would look to enlist aphids, leafhoppers, and whiteflies to transmit genetically engineered viruses to crops. Those viruses could activate or deactivate parts of plants' genetic code, helping to fight diseases or resist droughts. But a group of scientists say the technology, and specifically the use of bugs to disperse it, could be easily weaponized. In a paper in the journal Science, the scientists argue if the program were benevolent, it could use spraying equipment to distribute the viruses instead of bugs. The program's manager told the Washington Post those fears were unfounded and that the program is focusing on delivering positive traits to plants. Either way, the Post points out, it's normal for DARPA projects to go nowhere, so the whole debate may be moot.
You stimulate all the muscles individually. So the beetle actually has no control over what it's doing.
Yeah.
Then. So you basically make it into a robot, actually.
Like a robot.
Could you tell us what you do here?
We use this system to detect the leg motion, so we can put some small marker on the leg.
And so this is the beetle that you're working with?
Yes. You can see the many wires, right? And my student at the Tao Finder, he is very good at the implementation and working on this leg motion control, actually. And he knows about the location of the leg muscles very well.
Can you tell me a bit what you're going to do?
Basically, I will press there are two buttons. One can change the working gait. The other one is I can change the step frequency. That means how fast the leg moves.
Yeah. And these LED lights, they correspond each to a muscle?
Yes, so in total 8 muscles.
Yeah. Okay. Let's see.
So the step frequency is 0.125 Hertz. So it's tripod, so it's very slow now.
And so now it's alternating its left and right.
Yes, alternating. So if I want to increase the step frequency, I need to press this button. So that if I press once, so it's faster. Now it's 0.25 Hertz.
So amazing that you just press a button and the beetle starts walking.
Mm-hmm.
It's I don't know. It seems almost unreal that you can kind of achieve this in a living creature.
If I press one more time, 0.5 Hertz, so it's faster. Then
this is really amazing.
1 Hertz. If I want to change to galloping, I press this button, so it will change to galloping. So, yes, it's galloping.
And this is when it runs faster, basically?
Yeah, the step frequency is 1 Hertz. Now it's 2 Hertz.
Wow.
Yeah. It struggles itself, but it still obeys the stimulation signals.
And do you think, is he struggling to kind of stop these stimulation signals?
Yes, I think so. But still, it obeys. You can see very clearly here.
And this just keeps on working like this?
Yes.
It doesn't get used to it or something? It doesn't...
No. I stimulate even like for more than half an hour, so it's still like this. We stimulate more than 7 days, more than 1 week.
And it keeps working?
Yes.
You're putting in electrical signals into the beetle now, but is it using up its own energy source, basically? Is it using the food that it eats to drive the muscles, or are the muscles driven by the electricity?
Yes, the food.
So you have to feed it to be able to keep on doing this?
Yes.
Looking at this thing, it's unbelievable, actually, that humans can, take over control over the willpower of this insect, because the muscles are being driven by the insect itself. I mean, it has to feed it, it has to eat to be able to move the muscles. And all that they are doing is putting a bit of electrical stimulation into the muscles, and they can control it to a degree that's amazing if you think about it. These may just be the first stages of this, but if you think about where this could go, this is really incredible. And how long would a beetle like this live?
The natural, regular beetles survive for 3 months to 6 months. Even after the implantation, beetle can survive for several months. We have never seen the beetle died right after the stimulation.
So it doesn't really hurt?
No.
All right, all I gotta do is blow on its back to make its wings go, and then I can control it with this controller by pressing a button and moving it to a side. Yeah. Okay. Hey, it worked. It went right. Can I try again? Als ik dan naar rechts druk, dan gaat hij naar rechts. En het werkt. I think the most fascinating thing that I've learned over these past few days is that technology and nature are creeping more and more together, and that we might be heading towards a future where technology and nature might become indistinguishable.
Dual-use research is research that's designed for the benefit of our society. However, there may be techniques in which there's an opportunity for misuse.
I believe that biotechnology is perhaps our greatest tool in trying to find ways to counter disease. It's a dual-edged sword, and it's perhaps our greatest challenge, because the very same techniques that would allow us to produce innovative countermeasures also provide opportunities for someone to use them in a bad way.
There have been a number of experiments that have produced unexpected results that, if technology fell into the hands of someone who wanted to do harm and had the opportunity to do so, it could be quite devastating.
Well, dual use in the scientific sense is taking legitimate research and the results and product of that research and using it, or finding that there are other ways to use it that are more harmful to humankind than what it was initially designed to do.
I think it is a real threat. It is potential, but it's also real. It is not as apparent in many cases, and it is immediately apparent in other cases.
I think there are certain things that can be done to minimize the risks, but there's always going to be the potential for negative purposes.
Oh, I'm sure the threat is real, because, I mean, if you can have biologicals that can cause harm to people, there's always going to be a threat that somebody will use it improperly.
It's a very broad category. One could easily envision how almost any research, no matter how seemingly unrelated to doing harm, a clever mind could potentially turn that around.
A piece of knowledge that might come out of a laboratory today is seemingly without harm whatsoever. But when put into a series of other pieces of information, could be devastating.
In the case of nuclear physics, there was an iconic experience, the mushroom cloud, that made it clear to nuclear physicists that their work had security implications. There has been no comparable iconic experience for life scientists.
The Biological Weapons Anti-Terrorism Act of 1989 was enacted into law May 22, 1990. It provided for the implementation of the Biological Weapons Convention as well as criminal penalties for violation of its provisions. This Biological Weapons Anti-Terrorism Act was drafted by University of Illinois international law professor Francis Boyle. The Act broadly defined several terms related to biological warfare. Those terms were vector, toxin, biological agent, and delivery system. The Act defined a biological agent as any microorganism, virus, infectious substance, or biological product that may be engineered as a result of biotechnology, or any naturally occurring or bioengineered component of any such microorganism, virus, infectious substance, or biological product, capable of causing death, disease, or other biological malfunction in a human, an animal, a plant, or another living organism. Deterioration of food, water, equipment, supplies, or materials of any kind, or deleterious alteration of the environment. These exact words can be found in the U.S. Code, and for more information on that, you can see my first documentary called Lethal Injection, The Story of Vaccination. Previous United States interpretation of the Biological Weapons Convention ban on biological agents was in line with the BWATA definition. However, the United States now maintains that Article 1 of the Biological Weapons Convention, which explicitly bans bioweapons, does not apply to non-lethal biological agents. According to the Federation of American Scientists, current U.S. work on non-lethal agents greatly exceeds limitations set forth in the Biological Weapons Convention. The Act made it illegal to buy, sell, or manufacture biological agents for use as a weapon. With passage of this Act, it became known as Public Law 101-298 upon its passage and signing, and provided for criminal penalties for those who violated its provisions. Just one problem. The Act specifically exempted what is called peaceful or defensive biological weapons research. In other words, the most deadly of viruses can be trafficked and studied, recreated, and genetically altered to be even worse if the research is labeled as peaceful or defensive, making the provisions of this act almost impossible to enforce, especially when it is the National Institutes of Health that are funding most of these defensive or peaceful biological weapons research, which they call as basic research. As we'll find out, there is no difference whatsoever between basic or so-called legitimate peaceful research and biological weapons research. They are one and the same. The act dictated sentencing for violators and stated that whoever knowingly develops, produces, stockpiles, transfers, acquires, retains, or possesses any biological agent, toxin, or delivery system for use as a weapon, or knowingly assists a foreign state or any other organization to do so, shall be fined under this title or in prison for life or any term of years or both. The Act also provides that if a quantity of biological agent or toxin appeared to have no peaceful purpose, it could be seized and subsequently destroyed. In addition, passage of the Act made violation of its provisions a federal crime. And so we can possibly understand the intent behind these so-called scientists who are conducting gain-of-function research on what otherwise would be called a biological weapon, why they are defending their actions. In other words, the terms peaceful and defensive are being used as an excuse to create the worst kind of biological weapons as a sort of workaround for this Biological Weapons Act, to avoid its provisions through dual-use research, and call them as basic or legitimate research. Again, there is no difference between basic, legitimate research and biological weapons creation. And this fact will be shown in great detail as we proceed. Francis Boyle, the author of this Biological Weapons Act, had this to say about COVID-19.
I think I found the smoking gun here. There was a recent scientific study published in Antiviral Research 10 February 2020 by 3 scientists from France and 1 from Montreal who did a genetic analysis of the Wuhan coronavirus, and they said, quote, "It may provide a gain of function of the 2019 coronavirus for efficient spreading in the human population compared to other viruses." Let me repeat that. May provide a gain of function to the 2019 coronavirus for efficient spreading in the human population compared to other lineage coronaviruses. That's the smoking gun for a offensive biological warfare agent. Gain of function properties is a tip-off. It's only useful for offensive biological warfare activity, and it is typically conducted in either it's so dangerous, in either a BSL-4 or a BSL-3 facility, and there in Wuhan you have the only BSL-4 facility in China. So I think it's clear it came out of this lab. Gain of function, means its DNA genetically engineered to be more lethal and more infectious. Gain of function means its DNA genetically engineered to be more lethal and more infectious. Clearly what we're seeing now with this coronavirus, it is basically SARS, which is already a weaponized version of a coronavirus. SARS, which is already a weaponized version of a coronavirus that has leaked out of that laboratory at least twice before. The weaponized version of a coronavirus that has leaked out of that laboratory at least twice before. And then it is given gain of function properties, which basically means it can travel by air for at least 6 feet and is more lethal. Second, we have an article here from the NatMed 2015, December 21, SARS-like cluster of circulating bacteronuses show potential for human emergence. This was at the University of North Carolina. In Chapel Hill. They have a biosafety lab level 3 there, and I have previously condemned them for using gain of function work on MERS, and I have previously condemned them for using gain of function work on MERS, which is the Middle East respiratory syndrome. It is like SARS, only more dangerous. It has a 33% lethality rate. And they were doing gain of function work there to make it even more lethal. Well, it turns out if you read the article, they admit that they were doing this with SARS, that they were giving it a gain of function activity. They admit that they were doing this with SARS, that they were giving it a gain of function activity. And it turns out part of their team was a researcher from China, Zheng Li Shi, Key Laboratory of Special Pathogens and Biosafety, Wuhan Institute of Virology, Chinese Academy of Sciences. And they gave a grant to the University of North Carolina to get their scientists in on this extremely dangerous Nazi-type biological warfare work. So it appears that what happened was, instead of stealing this technology, China bought it. And they bought it from the lab there at the University of North Carolina. They put their person in there. They took the technology and they brought it back to the Wuhan lab. It's right there. This fellow works for the Wuhan lab. And it also appears that the North Carolina lab got cells from Fort Detrick, which is the U.S. Major facility for the research, development, testing, stockpiling of biological weapons. Fort Detrick is the U.S. equivalent of Porton Down. So that's where they got some of their cells here. And they made it clear the work that they were doing was to increase the pathogenicity of original SARS by giving it this gain of function activity.
As I started looking at this information, and as I started seeing that this is actually a chimeric version, something that was developed here in the United States in 2015, it was published in Nature magazine, that it was developed in the University of North Carolina, Chapel Hill, where all the initial studies were done. The chimeric research, despite having a moratorium by the US government to prevent any chimeric research, by the way, just so your audience understands, chimeric research basically means that they're taking a naturally occurring substance or virus, in this case, and then mutating it, genetically modifying it, genetically modifying it, changing the configuration morphologically to gain function. It's called a gain of function study or gain of function research. And that basically means that they're making something that already may have some potential to cause harm, making it more harmful, making it more virulent, making it more resistant, making it more harmful, making it more virulent, making it more resistant. And basically that's what I found, that they took the SHC014 strain of the coronavirus, the surface antigen component, and brought in the backbone from the SARS coronavirus, put them together, and then inserted HIV and MERS, and then inserted HIV and MERS, and then inserted HIV and MERS orthologs on top of it to make a more virulent, more detrimental virus. Now, the thing is that
if I could jump in for just a moment here, because what I think you're talking about is the moratorium that was placed, and then $3.7 million from the National Institute of Health was transferred to China so they could continue this research, even though the moratorium. So they basically outsourced this research to China. This research to China. Is this the same topic we're talking about here? This news is just breaking?
Yes, this is exactly right. So it goes back to 2014. The U.S. government decided, based upon certain virologists at that time, saying that, look, there's no justification for this kind of research. There's a potential for it to cause harm, cause a pandemic. So there's no justification for us to do this type of research. So the government passed a moratorium in 2014. Fauci approved monies to be section for this type of research. So basically, and I'm getting really, every time I think about this, it gets me really flustered because I'm so angry that he basically broke the law. He more than broke the law. He created this entire cascade that we're seeing with the world shutdown was created by this initial aspect back in 2015. He broke the law. He went against government moratoriums. He took taxpayer money and he funded research that has now led to the COVID-19. In 2017, he was documented at Georgetown University saying that there will be a pandemic that this presidency will face, that this term will face. How did he know that in 2017 that something was going to happen in 2018, 19, or 2020? You can't predict the market from 3 days from today, what's going to happen. How did he know that there was going to be a pandemic? He stated the president of this president will face a pandemic. The exact quotes, I don't want to say the exact words. You guys have the video footage.
There is no question that there will be a challenge to the coming administration in the arena of infectious diseases, both chronic infectious diseases in the sense of already ongoing disease, and we have certainly a large burden of that. But also there will be a surprise outbreak. There will be a surprise outbreak. There will be a surprise outbreak.
My point is that when somebody starts making these type of statements, and then they were involved with the breaking of the law to fund the research that American taxpayers paid for, that then goes to Wuhan and now trying to create this diversion and say that it was China. God, I don't know whether it came from China or released in China, whether it was already released here. It's irrelevant. The complicity of what's going on right now, whether it was released in China or whether it was already here or however it happened, the U.S. system has condoned it, has jumped on it. They're opportunistic. They're shutting down the economy when they know, in fact, that there's no basis.
Is Fauci directly responsible for this pandemic because he maneuvered the money, he maneuvered around the moratorium, kept this chimeric research going in China? Is he directly responsible for not just the pandemic, but also the response that's killed the economy, put, 17 to 22 million people out of work? Is Fauci directly responsible?
I'm going to say this. I think that I've seen some petitions going around. I know Dr. Shiva said that Fauci should be fired. I think that's the nicest thing that could be done to Fauci. I think he should. He's a criminal. He's broken the law. He's gone against the government. I mean, to me, that seems like it's a traitorous thing to do when the government has passed a regulation and he's at one of the highest levels in the NIH. He's got a directorship at the NIH, and then he breaks the law, he breaks the moratorium, and then funds research against something that could potentially cause harm throughout the entire world. And he's in collusion with the foreign government to do. I mean, I don't know. That's a matter for attorneys to decide. But I'll tell you something. Fauci goes back to 1981. In 1981, he called HIV/AIDS the gay disease, and he was the one who funded the research behind a drug 3 years before it was even established that there was a real virus. So the virus was established in 1984. In 1981, he was pushing the agenda of a drug prophylactically to treat people for a condition that has now been seen to be related to lifestyle, nutritional status, all sorts of other things. He was pushing the agenda to push a drug that caused so much harm as a prophylactic, just to prevent HIV. Now, this goes back 1981. We're talking 39 years. This guy has a history of pushing an agenda. But now the president comes out and says there's a drug called hydroxychloroquine, which has been on the market for, God knows, 50, 60, 70 years, and another drug, Zithromax, which has been, it's a macrolide that's been around for at least, that class of drugs has been around for at least 70, 80 years. Maybe Zithromax has been around for, I don't know, 20 years, whatever the case is. These are established drugs. They're safe drugs. Using them in combination, there are doctors that have shown at least 99% efficacy. They've shown hundreds and hundreds of patients that have treated. Nobody's died. Symptoms resolve within an hour, hour and a half to a day and a half, 2 days. Okay, so you've got this class. The president brings it out. And Fauci says, get this, Fauci says, unless there's some studies to show that, it should not be used. Yet he's promoting a vaccine that we have no idea what the hell that's going to do. And he's saying that's okay to use? I mean, if that does not show a conflict of interest, I don't know what does. People need to wake up and realize there is a massive criminal component to this. This entire world economy, the whole world economy shutting down, it comes back to one person, in my opinion, and that's Fauci. Now, there's other people that are involved with the charade, obviously, but Fauci is the person that they put up in the front, and Fauci is the one who had the ability to stop this. Fauci has told a friend of mine, Judy Mikowitz, who I was doing an interview with just about an hour earlier, she went to jail and spent 5 years. She was threatened by Fauci 10 years ago when she came out to say that the viral studies and the research that she had done, and she was in charge of doing the research in Ebola to make it more virulent, She was in charge of doing the research in Ebola to make it more virulent. The gain of function studies on Ebola, she was responsible for that. When she realized the stuff that she had developed and the stuff that she was researching was being used for nefarious purposes, and she was going to be a whistleblower, her career was threatened, and more, her life was threatened. She refused to back down. She went forward, and guess what they did? They framed her and they put her in jail for 5 years. She's got a book that came out today, I believe it's called Plague something. It just came out today. I'm telling you right now, Fauci, it can all be traced back to Fauci, but Fauci is just one of the players. There's other people, and you can start looking at what the agenda is. It's wide open. Anybody can go in and do their own internet search. They just need to open their eyes, they need to open up their mind, and they need to be married to no decision or outcome. Just look at it for yourselves, people. You've got your brains. Use your own brain. Come up with your own sequence of events, how it happened. Do the research, and you will, if you're following a logical sequential thought pattern, you'll come to the same conclusion. There's no way that you can come to any other conclusion. All roads lead back to the same place.
So let's read from this smoking gun report out of Nature Medicine, published November 9th, 2015. A SARS-like cluster of circulating bat coronavirus shows potential for human emergence. Now understand that this type of dual use or gain-of-function research is all based on potential. The potential to make something much worse, much more virulent, much more contagious, and much more deadly. And so the word potential here is the main draw for such types of research. In the abstract, we read, the emergence of severe acute respiratory syndrome coronavirus, or SARS-CoV, and Middle East respiratory syndrome, or MERS-CoV, underscores the threat of cross-species transmission events leading to outbreaks in humans. Here we examine the disease potential of a SARS-like virus, SHC-014-CoV, which is currently circulating in Chinese horseshoe bat populations. Using the SARS-CoV reverse genetics system, we generated and characterized a chimeric virus, expressing the spike of bat coronavirus, SHC-014, and a mouse-adapted SARS-CoV backbone. The results indicate that group 2B viruses encoding the SHC-014 spike in a wild-type backbone can efficiently use multiple orthologs of the SARS receptor human angiotensin converting enzyme 2, or ACE2, replicate efficiently in primary human airway cells, and achieve in vitro titers equivalent to epidemic strains of SARS-CoV. Now, in case you're not familiar with the symptoms, that is basically a description of the current SARS-2 coronavirus, or COVID-19, affecting the lungs and many other organs. And that is based on the added function or gain of function research here described, where the spike or communication device, if you will, is able to connect to these cells through the ACE2 receptor. Additionally, in vivo experiments demonstrate replication of the chimeric virus in mouse lung with notable pathogenesis. Evaluation of available SARS-based immune therapeutic and prophylactic modalities revealed poor efficacy. Both monoclonal antibody and vaccine approaches failed to neutralize and protect from infection with COVs using the novel spike protein. I say again, Both monoclonal antibody and vaccines failed to neutralize and protect from infection using this particular novel or unique spike protein, one that does not happen in nature. On the basis of these findings, we synthetically re-derived an infectious full-length SHC-014 recombinant virus and demonstrate robust viral replication both in vitro and in vivo. Our work suggests a potential risk of SARS-CoV reemergence from viruses currently circulating in bat populations. Now, to put that in layman's terms, we took bat coronavirus, which likely cannot transmit to humans and certainly cannot be transmitted from human to human, and we added or put a new function, a gain of function, which is this receptor coming from a human or HIV virus. This, in other words, is what we're calling SARS-2. As we go further into the study, we find, quote, together, the results demonstrate that broadly neutralizing antibodies against SARS-CoV may only have marginal efficacy against emergent SARS-like CoV strains, such as SHC-014. Again, speaking to the ineffectiveness of vaccine treatments. To evaluate the efficacy of existing vaccines against infection with this strain, we vaccinated aged mice with double inactivated whole SARS-CoV, or DIV. Previous work showed that DIV could neutralize and protect young mice from challenge with a homologous virus. However, the vaccine failed to protect aged animals in which augmented immune pathology was also observed, indicating the possibility of the animals being harmed because of the vaccination. Here we found that DIV did not provide protection from challenge with this strain with regards to weight loss or viral titer. Consistent with a previous report with other heterologous group COVs, serum from DIV-vaccinated aged mice also failed to neutralize this strain. Notably, DIV vaccination resulted in robust immune pathology and eosinophilia. Together, these results confirm that the DIV vaccine would not be protective against infection with SHC-014 and could possibly augment disease in the aged vaccinated group. We can also look at just a few of the various reports that list the unbelievable amount of accidents that happen in these high-level labs. For instance, this report in Science, Mounting Lab Accidents Raise SARS Fears. This in April of 2004. And so this gain-of-function type of research has obviously been going on much longer than we realize. Reported in The Scientist, November 16, 2015, we read that the lab-made coronavirus triggers debate. The creation of a chimeric SARS-like virus has scientists discussing the risks of gain-of-function research. Now note that since COVID-19 has come out, this particular article has become very popular, with good reason. In the effort to calm the public, this update was added March 11, 2020. On social media and news outlets, a theory has circulated that the coronavirus at the root of the COVID-19 outbreak originated in a research lab. Scientists say there is no evidence that the SARS-CoV-2 virus escaped from a lab. And yet, by the end of this presentation, you will have no doubt that this and most other viruses have been lab-grown. Ralph Barrick, an infectious disease researcher at the University of North Carolina at Chapel Hill, last week, November 9th, published a study on his team's efforts to engineer a virus with the surface protein of the SHC-014 coronavirus found in horseshoe bats in China, and the backbone of one that causes human-like severe acute respiratory syndrome, or SARS, in mice. The hybrid virus could infect human airway cells and cause disease in mice, according to the team's results, which were published in Nature Medicine. The results demonstrate the ability of the SHC014 surface protein to bind and infect human cells, validating concerns that this virus or other coronaviruses found in bat species may be capable of making the leap to people without first evolving in an intermediate host, Nature reported. They also reignite a debate about whether that information justifies the risk of such work, known as gain-of-function research. If the new virus escaped, nobody could predict the trajectory. Simon Wayne Hobson, a virologist at the Pasteur Institute in Paris, told Nature. In October 2013, the U.S. government put a stop to all federal funding for gain-of-function studies, with particular concern rising about influenza, SARS, and Middle East Respiratory Syndrome, or MERS. NIH, the National Institutes of Health, has funded such studies because they help define the fundamental nature of human pathogen interactions, enable the assessment of the pandemic potential of emerging infectious agents, and inform public health and preparedness efforts. NIH Director Francis Collins said in a statement at the time, "These studies, however, also entail biosafety and biosecurity risks, which need to be understood better." Barrick's study on the SHC014 chimeric coronavirus began before the moratorium was announced, and the NIH allowed it to proceed during a review process. So to be clear, a moratorium on this type of research was declared, and the NIH allowed this SARS research to continue, despite that moratorium. During the review process, which eventually led to the conclusion that the work did not fall under the new restrictions, Barrick told Nature. But some researchers, like Wayne Hobson, disagree with that decision. The debate comes down to how informative the results are. The only impact of this work is the creation in a lab of a new non-natural risk. Richard Ebright, a molecular biologist and biodefense expert at Rutgers University, told Nature. Quote, "The results move this virus from a candidate emerging pathogen to a clear and present danger. In other words, when man interferes with nature, it becomes a present danger instead of something that can likely never happen." Peter Daszak, president of the EcoHealth Alliance, which samples viruses from animals and people in emerging diseases hotspots across the globe, told Nature.
The final piece of evidence here is Archive of Virology 2010. And this is research done with the Australian Animal Health Laboratory and again, the Wuhan Institute of Virology, where they DNA genetically engineered SARS and HIV. They DNA genetically engineered SARS and HIV, okay, to make a weapon, okay, to make a weapon. And they got a grant here from the Chinese Ministry of Science, Technology, etc., to do this. So once again, they bought the technology. They didn't steal it back to Wuhan. And so my reading of these articles, basically, these 3 articles, is that they took the technology from this death factory at North Carolina. They took the technology from this Australian research project. They brought it back to the Wuhan BSL-4 and tried to genetically engineer it all together as a sort of a turbocharged biological warfare weapon that would consist of SARS, which is already a weaponized coronavirus, SARS, which is already a weaponized coronavirus, gain-of-function properties, and HIV. Gain-of-function properties and HIV. And as you know, those Indian scientists already did an analysis of the coronavirus, and it was published online. I read it, and HIV was clearly in there. And HIV was clearly in there. HIV was clearly in there. I saw the pictures. So that, I think, is what we are dealing with here. We've never seen, at least released in the public, a biological warfare agent this dangerous, except the Amerithrax that was in October 2001 after the 9/11-2001 terrorist attacks. That was super weapons grade anthrax, 100 grams of spore. It too traveled in the air. It seemed to be based on nanotechnology, and we hadn't seen any. And at the time, I publicly stated it came out of the U.S. Biological Warfare Weapons Program. It came out of the U.S. Biological Warfare Weapons Program. At the time, I publicly stated it came out of the U.S. Biological Warfare Weapons Program, and probably Fort Detrick, which was later confirmed. I said that about the first week in November. I was even on the BBC saying that. And then an order was given to silence me. I was blackballed off all mainstream news media in the United States, Britain, Europe, you name it. So despite the fact my viewpoints are all over the world, no mainstream news media will touch me. So the Amerithrax, of course, was extremely dangerous, but not as infectious as what we're seeing here. So in a nutshell, that's where I see it as of today.
So those experiments were already done. They were approved experiments. People went through, but done in very safe settings with very highly reputed groups. So there was really nothing wrong with the experiments themselves. It was really how the knowledge would be used by bad actors.
Yeah, but it's gone beyond that now, because I think you're a signatory of the Cambridge.
So I signed the Cambridge Working Group document after some thought. I do feel we have to be very cautious when you start to say scientists shouldn't do things, because that way is a very slippery slope. The Cambridge Working Group is actually saying there should be a moratorium on it, and while we figure out what's going on, like there was for recombinant DNA in the 70s. And it's only on some pathogens, so SARS and MERS and a few of the flu strains. So SARS and MERS and a few of the flu strains. And it's only on some pathogens, so SARS and MERS and a few of the flu strains. To me, I think that the sort of question you're talking about is what evolution can do, what could come up. You have to ask yourself, do you need to do this on a virus which is horrendously risky for humans? So if you're asking general principles, and that's what's the great thing about so much of this DNA technology, we can ask questions of a whole range of things that either aren't for us or they're not that nasty. They're talking here about particularly nasty things. That if they got loose, catastrophe could be visited on us. That if they got loose, catastrophe could be visited on us. They're talking here about particularly nasty things. That if they got loose, catastrophe could be visited on us. So you have to ask, why was it necessary to do those experiments in those particular pathogens? What are we getting out of studying those? And the justification there has been, well, it's something to do with surveillance, not therapeutics development, surveillance, that 5 mutations to mammalian transmissibility was the sort of answer they were giving, and it's these 5 mutations. So you've got to keep an eye out for those, and if you see 3 of them in nature, we're really close to a disaster. That's the sort of logic. And to me, as an evolutionary biologist, I think, well, that's one pathway to mammalian transmissibility, but in ferrets, but there's going to be many other pathways.
That's right.
So the predictive power of identifying a few mutations strikes me as being very low. We don't know. I mean, that might be the only route. It might be predicted, but we don't know that. I'd like to see that in other systems, safer systems first. How good is it as a way of developing surveillance systems to generate horrendous viruses and then go looking for elements of those in nature? To generate horrendous viruses and then go looking for elements of those in nature. To me, that's a completely uncertain. And so given that we don't know if that works, and the risk if these things get out, and we know very many labs that have released pathogens we didn't want to lose, and we know very many labs that have released pathogens we didn't want to lose, and the risk if these things get out, and we know very many labs that have released pathogens we didn't want to lose. That's why I signed the thing in the end. It seemed to me very, very uncertain gains.
Sorry, yeah, so totally concur with that rationale. It makes perfect sense. The virtue of, but perhaps the value of getting that information is that we now know for a fact that there is a path in ferrets, that will lead to that. Not having done or not having access to the natural experiment, which actually would be devastating should that occur, we would not be able to ever know that information. So in this way, we're at least able to know for a fact that this is possible.
Yeah, I do take
and
that.
if it is possible, then we need to be watching out for it.
I do take that point, and I think actually Otto and I were over cocktails on the weekend discussing this very issue. And I think, well, Otto's not to put words in his mouth, was arguing that we did learn some interesting biology from that process. We did learn some interesting biology from that process.
It's really important because we as a scientific community don't know what we're doing, don't know what we're doing, don't know what we're doing.
We did learn some interesting biology from that process, and that might help us with risk evaluation.
Absolutely. Yeah, so yes, and so I must say, I am completely undecided on this topic. I think I might be slightly convinced by your argument, but I do think that it's very interesting in the case of H5N1 that there were essentially two sets of things that needed to happen. One was that you needed to change the epitope affinity of the virus, and then the other thing was that you needed to change the temperature range of the polymerase. And I think and so maybe the trivial result was that you changed those two things and you got gain of function. You changed those two things and you got gain of function, and you got gain of function. But I do think that one of the interesting corollaries is that with the change of temperature affinity, that's what allowed the virus to replicate high up in the respiratory tract of mammals instead of deep down in the lungs where the temperature is the same as the gut of birds. But interesting biology is that then, at least potentially, you could see that a lot of the virulence is really associated with the replication very deep in the lungs. And so it might be that there's kind of a trade-off or conflict, that you can get transmissible viruses, but they're likely to be much less virulent just because the function of transmitting is associated with being up here in a low temperature, less pathogenic environment. So I do think there was some interesting biology.
So the slippery slope then becomes, every experiment you do on influenza has the potential to actually go down that same path, create something that has increased transmissibility, if that's what you're testing for. Many of those experiments actually don't test for that. So any experiment you do with any pathogen has the potential to make it more or less virulent. And we know from, for example, studies that have routinely introduced antimicrobial resistance into pathogens, routinely introduce antimicrobial resistance into pathogens, routinely introduce antimicrobial resistance into pathogens. Some of them are drugs that for which if an organism such as TB becomes resistant now to 8 different drugs, there are no treatments for. There are no treatments for. There are no treatments for. How do you deal with that? And in fact, the importance of, I think, this sort of the sense of gain of function experiments onto specific parts means that you'd have to stop experimenting on pretty much anything.
Yes.
It's very difficult to predict what that gain of function might be.
I agree on that. There is obviously a very strong argument for gain of function experiments. I've got no issue with that. The question is whether we should do it where we're trying to enhance certain types of function, in this case mammalian transmissibility, where we're trying to enhance certain types of function, in this case mammalian transmissibility, in pathogens that we're genuinely very scared about. We have every reason to be very scared. And I think this discussion is an example of why we need a moratorium here. We need to pause to think, what sort of questions are we really going to get good things out of for taking these risks? And that has to be put up against the possibility these things get out. And you can measure the rate at which people lose pathogens. I mean, the pathogens get out of labs, and it's it's not zero. And you can measure the rate at which people lose pathogens. I mean, the pathogens get out of labs, and pathogens get out of labs, and pathogens get out of labs, and it's not zero.
Yeah. And this is clearly a discussion that's going to be continuing in the scientific community for some time to come. And I think I would really encourage more discussion about this. In fact, on the forums, to get some more perspective from the people that are taking this class in terms of what does the community that benefits from this science really get from the kinds of work that people like us are trying to do.
I can't think of a moment bigger than this one. I've likened this as the moment for biology akin to the moment when fissile nuclear material was figured out by physics and offered both a way to solve the problems of the universe and nuclear weapons.
These papers really represent a seminal moment in life sciences. We now really have been confronted with examples of where the science itself, which is very important in moving forward for the public's health, also pose a potential risk for nefarious actions or even those situations where this virus might escape from a laboratory. So we're really at a point for the first time of trying to balance the need to know versus the need to protect.
There is a need for dual-use research. That's the point for establishing of this committee, the NSABB. And the idea there is that we find some way to balance public health interests, freedom of speech, and freedom to pursue research of scientists with the public good and public concerns about the safety and security of research.
We believe that it would be not in the world's best interest to have all the detailed information come out. Now, having said that, we also have made a very strong call for an international discussion about where do we go forward. So the NSABB never saw itself as being the final arbiter on this. Really, we're, I think you might say, the first court of appeals, and we know we need more guidance from all the life sciences around the world to move to the next step.
I think we can learn a great deal about transmission of influenza virus through the air from this work. This is a question we know very little about. There are very few systems in which to study it, and these are the first advances that we have on studying the transmission in ferrets. So there's so many additional experiments that come from this. It would be a shame to suppress the data from these experiments.
One of the really disappointing parts of this discussion to date has been that everyone seems to have their own set of facts. And in particular, for myself, coming from a public health background where I've been in the trenches, fighting disease, doing surveillance, stopping outbreaks, I've watched a number of the life scientists who have made blanket statements about the public health utility of this kind of information or what we can do with it as an argument for why it should be widely disseminated. And in the first instance, none of this holds water. And it doesn't mean that the work isn't important. It doesn't mean life sciences shouldn't move forward. But there's been a lot of misunderstandings about what the real utility of this information is in protecting the public's health.
I think we need to have an Asilomar type moment where scientists meet and decide how to go forward. Unfortunately, since 9/11, we now have a lot of policy analysts who are participating in this debate, not scientists. And I think it's unfortunate in a way because the result of Asilomar, where there were largely scientists meeting, were the regulations that applied to recombinant DNA. So in an ideal world, that's what I would like to happen. But as you know, it's not an ideal world.
The mistake made at the Asilomar moment was that it was scientists talking to scientists. Very heated. It was not a, happy little let's all hold hands and agree. And then saying to the public, okay, we've reached an agreement, you all should trust us. And the backlash from community to community all over the world was enormous.
After you've pursued work, you've invested lots of resources in trying to get it done, and you've submitted for publication and be told that it can't be published. It would be far better to have discussions of this sort at the time that the work is first described.
Everybody can be a Monday morning quarterback and say we should have thought of this, we could have thought of this, and we actually did. We just didn't think that influenza was the one we had to worry about. But now we're where we're at. Those days are over with. We now realize we have to be very concerned about how we move forward.
Now, in my view, I can't think of an experiment done by a legitimate scientist seeking answers to a question that you wouldn't want to publish. I can think of many experiments that a nefarious scientist would do and you wouldn't want to publish, and they wouldn't either. But legitimate experiments, in my view, are all publishable.
The
issues about publication and about conducting this research really need to be addressed by the scientific community at large, with representatives from public health, with international representation. I think it's very important that this debate occurs now, and I think these papers have given us the opportunity to have this debate now.
Welcome to NIH Natcher Center. My name is Sally Howard. I'm the Chief of Staff to Secretary Kathleen Sebelius here at the United States Department of Health and Human Services. It's my distinct honor to welcome everyone to this international consultative workshop on highly pathogenic avian influenza H5N1 viruses. The biomedical research mission of the department is carried out by the Centers for Disease Control and Prevention, the Federal Food and Drug Administration, as well as the National Institutes of Health, the host of today's meeting. Our topic for the next 2 days, highly pathogenic avian influenza, is pertinent to the research and public health missions of the department, but as importantly, is an issue of major concern around the globe. Highly pathogenic avian influenza has long been recognized as a significant agricultural problem. It is widespread among poultry in parts of Asia and the Middle East, and to date has caused an estimated $20 billion US in economic damage across the globe. More worrisome is the possibility that HPAI, H5N1, will become a major public health threat. We already know from experience that this virus is capable in some instances of jumping hosts and moving from avian species into humans, causing severe respiratory illness. This is thankfully a relatively rare event, but when this leap occurs, it can be quite lethal. Some 600 human cases have been reported since 2003, with a mortality rate of approximately 60%. Although currently H5N1 is not well adapted for sustained human-to-human transmission, recent studies underscore the growing concern that the virus may acquire this ability, and thus the importance of understanding this virus and its potential to become a pandemic disease agent, to become a pandemic disease agent.
However, it has not been able to continue through human transmission. In other words, it has not developed the ability to go from person to person to person to person. And we're hoping that it doesn't obtain the ability to be easily transmitted.
There are different approaches for studying the ways in which the H5N1 virus can spread, adapt to new hosts, or become more pathogenic. One approach is to engineer into the virus genetically based traits that mimic those that might occur in nature. These so-called gain-of-function experiments are not without risks, however, and have generated much discussion over biosafety and biosecurity issues. I think we all remember about a year ago reading the New York Times article that talked about the doomsday virus. I think that launched us off onto a very vibrant discussion about this type of research.
The truth about the doomsday virus, as reported in the New York Times opinion section and editorial on March 3rd, 2012. 2 months ago, we warned that a new bird flu virus, modified in a laboratory to make it transmissible through the air among mammals, could kill millions of people if it escaped confinement or was stolen by terrorists. Now, Ron Faucier, the Dutch scientist who led the key research team, is saying that his findings, which remain confidential, were misconstrued by the press. He says that the virus did not spread easily and was not lethal when transmitted from one ferret to another by coughing or sneezing, and that it became highly lethal only when big doses were injected into the animal's windpipes. That is hard to square with his original assertions. Experts who read his original manuscript say it reported that the new virus spread through the air and remained as virulent as the natural virus, which has killed 60% of the humans it has infected. Dr. Faucier's new claims are only the latest bizarre twist in a global health debate that badly needs an objective, independent arbiter. The public needs to know whether this virus is a potentially big killer, and if so, how it should be contained. It needs to know what details can be published without giving terrorists a recipe for a biological weapon. And it needs to know that a mechanism will be put in place to assess all the risks and benefits of such research before it is approved, not after a new virus has already been created. The debate became public after a federal advisory board, the National Science Advisory Board for Biosecurity, recommended that papers prepared by Dr. Faucier's group and researchers doing similar work at the University of Wisconsin-Madison be published only after omitting details that might help terrorists. That drew charges of censorship from some scientists, and others warned that restricting the information would make it harder to track and combat an outbreak of a similar strain. The World Health Organization convened a closed meeting of 22 experts last month, which concluded that the research should eventually be published in full. The group was dominated by participants with a clear stake in publication, including the researchers who made the viruses, the journals that want to publish their papers in full, and developing countries that want access to full details in exchange for having contributed the viruses that were studied. Now, this country's National Institutes of Health, which financed the research and has its own reputation on the line, is asking the Biosecurity Advisory Board to reconsider its call to redact details before publication. We welcome a new appraisal from a board that has already shown considerable independence. We hope it will look beyond the security and terrorism issues and voice its opinion on what safety precautions should be required to prevent the virus from escaping and whether the work should proceed at multiple labs or possibly be halted. These issues need to be resolved by experts who do not have institutional biases or turf to protect. The World Health Organization should be in the best position to oversee a response to what is a global problem. Its first effort was one-sided and disappointing, but it has pledged to convene further meetings with a much broader range of experts and interested parties. It must ensure that these forums are not rubber stamps for what the narrower special interest group just concluded. These are complicated issues and the stakes are enormous. Governments and scientists have a clear responsibility to get this judgment and future efforts right. Now, unfortunately, this forum we are watching currently, though the warnings of danger and the death of the entire population was made very clear by the participants there, this so-called public forum, which the public never really saw, was indeed merely a rubber stamp event. In other words, it meant nothing. There was no actual public opinion brought forth. What follows is a group of government bureaucrats, drug company representatives and salesmen, and other so-called scientists, including the aforementioned Ron Faucier, who originally did these experiments that are being debated.
These safety and security concerns are global in scope, and thus any path forward for this type of research must be developed through an international dialogue, and hence the purpose and value of today's meeting. Here at the Department of Health and Human Services, we've had extensive discussion about the analytical framework that should govern our decision-making when considering HPAI, H5N1 gain-of-function research. The discussions over the next 2 days will provide important insights to consider as we finalize our approach to decisions about the type of research we should fund, and can also inform your own considerations of these issues in your home countries. In light of that, it is especially gratifying to see the robust participation at this meeting from countries around the globe, including those for whom H5N1 is an ongoing agricultural and public health concern. I know those from, there are a number of people from Southeast Asia in attendance. We're glad to have you share your views about the value of the research, its potential risks, the risk to public health preparedness of not doing the research.
Ah, the sociopath's favorite logical fallacy. It's somehow unethical not to mutate and recreate deadly viruses that could escape the lab accidentally or on purpose, thus killing the entire population of Earth, though it would likely never happen in nature.
We're glad to have you share your views about the value of the research, its potential risks, the risk to public health preparedness of not doing the research, which again is the counter to the biosecurity risks of doing this research, and the principles that should guide any research in the future. Dr. Dijkgraaf is co-chair of the Inter-Academy Council, past president of the Royal Netherlands Academy of Arts and Sciences, and director of Institute for Advanced Study at Princeton.
And the subject matter, gain-of-function research on highly pathogenic avian influenza H5N1, is a topic of special concern, of course, to scientists across the world, but also one which the scientific community in the Netherlands has given a lot of thought, since we had very active researchers working on this and understanding the agricultural and public health threats. It's also of interest because some of this work fits perfectly the characteristic of dual-use research of concern. A reference to important scientific work that can nonetheless yield information and technologies that in the wrong hand could be misused. Yield information and technologies that in the wrong hand could be misused to cause great harm to the public health or global security. This dual-use question has been a topic of concern to the Royal Netherlands Academy of Arts and Sciences, which actually spearheaded the development of both nationally and internationally, the development of a biosecurity code of conduct that emphasizes awareness raising, oversight and accountability among scientists. And these are themes that have great pertinence to our aims today as well. In charting a path forward, it's essential that scientists enter into a dialogue with those who have other perspectives, including security experts, safety experts, and the public at large. I expect that we will hear very diverse points of view, and that is a good thing. The organizers have made every effort to represent a balanced representation of the issues as possible. And it's important to emphasize there are no foregone conclusions in this workshop. All points of view are fair and will be taken into account by the US Department of Health and Human Services and by all of us at this meeting in charting the best course forward. And certainly this issue is a prime example that I would say motivates a coordinated global approach.
In one sense, we're dealing with a very circumscribed topic, highly pathogenic avian influenza H5N1 virus. But in a larger sense, the subject of this meeting literally affects every individual in the world. Every citizen in every country has a stake in the research that will or will not go forward with respect to these highly pathogenic agents. This meeting is about advancing a global dialogue on the issues that directly and indirectly emanate from research with this highly pathogenic agent that has the character of dual use that Professor Dijkgraaf referred to. We want this meeting to be a place where we can invite every point of view to be expressed openly, candidly, in a forthright manner, and respectfully. It's very important for us to keep in mind that our aim is not to reach a consensus amongst those here or amongst the panelists or in any group that is represented here. Our goal is to share and to hear individual points of view so that they can inform both the framework and the larger dialogue that surrounds these issues.
In other words, your public opinions mean exactly jack shit at the end of this forum. After all, the agency that's funding these research projects is the same agency that will decide which projects to fund. And that's why many of you watching this have never heard anything about this topic or this forum, even as COVID-19, a man-made lab-escaped gain-of-function virus, a bad to worse case scenario in this forum, grips us all.
Our topic, of course, is centrally focused on the safety and responsible conduct of research. And the research program in the United States government is focused particularly on the work of the National Institute of Allergy and Infectious Diseases here at the NIH. And we are very privileged in opening our conversation over the course of these 2 days to be able to hear the perspective of the director of the NIAID, who in this audience needs no introduction. But it is for me a great pleasure and a source of privilege to be able to introduce to you the director of the NIAID, Dr. Tony Fauci.
Thank you very much, Harvey. It's a great pleasure to be here with you this morning. I want to take this opportunity, as you see on this first slide, to first welcome you all. I know I see so many friends and colleagues in the room who have traveled a great distance to be here with us today to discuss this extraordinarily important topic, but also to give a brief overview, as Harvey said, a considerable amount of the activity that goes on in this area from a research standpoint comes from the NIH and NIAID. As you know, we're part of the Department of Health and Human Services, as is the Centers for Disease Control and Prevention, who also have a considerable amount of contributions, both from a public health surveillance as well as from a research standpoint, in the topic for today, which is gain-of-function research on highly pathogenic avian influenza H5N1. Gain-of-function research, gain-of-function research, gain-of-function research on highly pathogenic avian influenza H5N1. So from the standpoint of research in general, we at the NIH and the CDC have been involved in the study of influenza literally for decades and decades. I don't need to tell this audience, but I know there are those who are listening in who may not have the historical perspective, is that seasonal influenza itself is an extraordinary threat to global health, resulting in approximately 500,000 deaths per year worldwide. Every once in a while, unpredictably, we have a pandemic. Unpredictably, we have a pandemic. History has shown us that pandemics occur and almost certainly will continue to occur unpredictably. On this slide, you see the history from the last century and this century of the now iconic pandemic influenza of 1918, which killed between 50 and 100 million people. A serious pandemic in 1957, again in 68, and then most recently in 2009, the first of the pandemics of the 21st century. Now, highly pathogenic avian influenza, as you've heard from Sally, is widespread among poultry in parts of Asia and the Middle East. Infections of humans, i.e. jumping species, is rare, but when it occurs and causes severe respiratory illness. Currently, this virus is not well adapted for sustained human-to-human transmission, but there is concern that has been going on now for approximately a decade. In fact, the first cases were in 1997, but since 2003, there has been considerable concern that the virus may ultimately acquire the ability to adapt itself better for human-to-human transmission.
Again, Dr. Fauci supports the sociopathic logical fallacy that, because we don't know whether highly pathogenic H5N1 could mutate naturally to infect humans more easily, but we're pretty sure it won't, we must genetically engineer and mutate H5N1 virus today, now, as soon as possible, into an even more deadly, more contagious, unnatural virus. And while we're at it, let's make it resistant to all known drugs. And hey, why not make it airborne too?
This is the now familiar map of the worldwide cases as of just a few weeks ago. As Sally mentioned, there are about 600 cases, but importantly, and I'm going to get back to that in a moment, because this is one of the issues which actually brings us here today and caused the discussion that has been intense over the last 13 to 14 months, and that is the 59%, the reported 59% mortality, which makes this a most unusual, smoldering pre-pandemic threat. As part of our research endeavors, clearly interventions in the form of diagnostics, therapeutics, and vaccines were important. But on the lower part of the slide, you see basic research. And that has always been and will continue to be an important part of our mission and our activities. If you look at basic research as we've approached it through the years, long antedating the appearance of H5N1 highly pathogenic virus, through the years, long antedating the appearance of H5N1 highly pathogenic virus, as part of that influenza basic research was intensive study of host adaptation, transmissibility of influenza viruses, pathogenesis, and virulence. And integral to that study has always been the issue of gain of function research, not only for influenza, but essentially for all infectious diseases research. For all infectious diseases research. Gain of function research, not only for influenza, but essentially for all infectious diseases research. For all infectious diseases research. Now, there are a few ways to look at gain of function research. There's the naturally occurring mutations which naturally give gain of function, and investigators study these effects on the phenotypes of interest. Does this mutation make something more transmissible, more pathogenic, or adapt to host better? Or, what historically investigators have done is to actually create gain of function, by making mutations, passage adaptation, or other newer genetic techniques such as reverse genetics and genetic reassortment. When we do that, often some phenotypes appear and others disappear. For example, it is commonly seen that when you increase a transmissibility, you may see a decrease in pathogenesis, or vice versa. You may deliberately increase pathogenesis, and see a decrease in transmissibility. But the bottom line is that gain and loss of function research is critical to understanding disease pathogenesis, antimicrobial resistance, and host responses, as well as to developing better techniques of surveillance, vaccines, and therapeutics. Specifically, to gain of function research on HPAI H5N1. What we're talking about now is the gain of function research in studies that increase predominantly the transmissibility, as is the case that I'm going to get into in a moment, as well as pathogenesis and alteration of host range of the virus. That increase predominantly the transmissibility, as well as pathogenesis and alteration of host range of the virus.
However, it has not been able to continue through human transmission. In other words, it has not developed the ability to go from person to person to person to person. It's a very serious disease, and we're hoping that it doesn't obtain the ability to be easily transmitted.
Now, the reason we are here today in this room with H5N1, highly pathogenic influenza, and we're not in this room discussing so many of the other gain of function research that we do, is because naturally occurring HPAI H5N1 viruses cause a reported almost 60% mortality in humans, which triggered a concern, understandably, clearly, that if you give a gain of function of a pathogenic virus to make it more transmissible, that's a whole different story than some of the other things we faced. So let's get down to what happened. Very briefly, you know historically, and I'm going to go very quickly through the historical perspective that brought us here today. There were studies done by two NIAID-funded investigators, Ron Fouché and Yoshi Kawaoka, in which H5N1, a strain from Indonesia and a strain from Vietnam, were altered in a gain of function, in which H5N1 were altered in a gain of function, either by direct mutation or by reassortment, were altered in a gain of function, either by direct mutation or by reassortment. And by passage in ferrets, the mammal model for this virus, for humans, there was an increase in fact, a development of aerosol transmissibility in a mammal which this particular virus did not have. There was a development of aerosol transmissibility, in a mammal which this particular virus did not have. Because of that, this triggered concern. This was after the experiments had been done and after they had been submitted to the journals. And from this press statement, we put it before our National Science Advisory Board for Biosecurity here at NIH, which is representative and advisory to the entire department of HHS as well as to other agencies. And the conclusion was made, and this is critically important for us to understand, and I take this right out of the statement that came from the NSABB, that due to the importance of the findings to the public health and research communities, the NSABB recommended that the general conclusions highlighting the novel outcome be published, but the manuscript did not include methodologic and other details that could enable replication of the experiments by those who would seek to do harm, What also was discussed that might accidentally be released, So it is not just people who would deliberately do harm. As Sally mentioned, following that, there was an explosion of reaction, sometimes bordering on the very extreme, as shown by this editorial from the New York Times, an engineered doomsday. This is just one, but as some of you may recall, there was a lot of activity talking about worst-case scenarios where the world might be destroyed. In that setting, the influenza community voluntarily imposed upon themselves a pause of 60 days from January 2012 on any research involving highly pathogenic avian influenza virus, leading to the generation of viruses that are more transmissible in mammals. That was supposed to be a 60-day, and as you know, the moratorium is still in place, the voluntary moratorium on the part of the investigators. But soon thereafter, there began to be an open discussion clarifying that research in different fora outside of the NSABB. The NIH and the CDC, because we do similar work, also agreed that we, in our own intramural investigators, we would refrain from these types of experiments and essentially abide by the moratorium that was signed on by a significant number of our extramural investigators. And I'm talking about the investigators at NIAID and other institutes as well as the CDC. From there came open discussion. Many of you remember the meeting in Geneva in February of 2012, where importantly new data were presented and even more importantly original data was substantially clarified, which led that group, which was predominantly influenza people, with a consensus to delay publication but ultimately to publish them after there had been some more emphasis and discussion of safety, public health, and communication issues, but also they recommended to extend the moratorium in order to have further discussion, as well as reaffirming the PIP agreement. Then back in the United States, what happened was a series, as you can see from the dates on these, of open discussions in different forums. There was a meeting sponsored by the ASM on biodefense, shown here, the people who were a part of the panel, myself included, Bruce Alberts of Science, Mike Osterholm of the NSABB, and Ron Fouché, one of the investigators. And we discussed openly the situation of the publishing of these data and the experiments that would be performed. The NSABB re-met the next month, and they examined the revised manuscripts with the clarification of data, and they voted unanimously to have Kawaioka manuscript be communicated in full, and in the 12-6 decision also that the Fouché manuscript be communicated after appropriate scientific review and revision, which, as you'll see in a second, actually occurred. Then we had a National Academy of Science workshop where we looked at the lessons learned. How did we get to where we were, and what do we need to do in the future to proceed in a responsible manner? The next day, the publication of the manuscripts occurred, as shown here in the Nature and Science titles. But the discussion continued, and one of the questions that was asked by the investigators, since this was a voluntary moratorium on their part, is how long is this moratorium going to go on, and what are really going to be some of the guidelines that we could actually follow on the basis of this? We at NIAID, being the major funders of most but not all of these people, we obviously were connected to that because they wanted to know, since you're our major funders, what kind of research will you fund? There was a major meeting of the Centers of Excellence for Influenza Research and Surveillance this past summer, at the end of July and the beginning of August. And as many of you know, I went up there to address the group. And what I recommended is that they continue the moratorium, but that we have the opportunity to discuss in an open fashion that is not only influenza researchers, but people like yourselves. And in fact, the transcript edited of my discussion at the Sears was recently published in MBio, in which I referred to this workshop. And I said the meeting participants would consider the general principles concerning the rationale for risks and benefits of such experiments and what lines might be drawn in their conduct and the reporting back and forth with the funding agencies. And so here we are. And this is the workshop that I was referring to that will take place today and tomorrow. And the purpose from the standpoint of, and I'm speaking from a pure research standpoint, is to review the key issues related to the gain of function of these viruses, scientific, public health, biosafety, biosecurity, and importantly for the decisions we have to make now is the considerations of the possible criteria for funding by HHS, i.e. NIH, CDC, of gain of function research on highly pathogenic avian influenza. As you know, there's a draft framework that Harvey referred to that we will be discussing in detail, and you'll have the opportunity to comment on, to guide funding decisions. And that's the critical point I want to make from the standpoint of the NIH about this gain of function research. Now, just to put in perspective, are we talking about a major chunk of what we do? No. As a matter of fact, this is relatively small, because the research, gain of function of transmissibility, et cetera, on H5N1 highly pathogenic is a very minor part, but an important part of our portfolio. It's part of 4 flu research projects that contain this. It's around 10% of the entire H5N1 portfolio and less than 1% of the total NIAID flu research funding. But it has triggered a very important question. Now, I want to close on these last 2 slides because I believe it's important that you keep this in consideration. First of all, questions have come up about the concern of the danger of people that you fund. NIAID, NIH, certainly CDC, only funds and conducts gain-of-function research on H5N1 highly pathogenic avian influenza viruses for researchers who are highly trained, skilled, experienced, and adequately regulated. This issue has not been a major concern about the investigators, certainly the ones that I just mentioned, who clearly fall into that category. That was not the concern. The concern was that the products or information that were generated by these experiments might be used by others in a way that could harm society, either carelessly, in an unregulated fashion, by inexperienced people, or even by deliberate misuse. Now, there are some disagreements, and we've heard them in the broad discussion in those months that went from the time of the moratorium until now. There's disagreements as to the scientific and/or public health value of these experiments. But I believe the people who feel that they shouldn't be conducted are in the minority. But I believe the people who feel that they shouldn't be conducted are in the minority. But I believe the people who feel that they shouldn't be conducted are in the minority.
Did anyone ask you or me? Are we, the over 6 billion people known as the general public, what you call the minority? Did anyone ask the entire human population of Earth if these potentially deadly, completely anti-nature, anti-scientific, gain-of-function, dual-use studies should continue to be done? Is this the first time you've even heard of this?
Because even the most concerned members of NSABB felt that the experiments should be done, but the distribution of the knowledge should be restricted. Now here comes the rub. As of today, there is no mechanism to provide restricted access to information regarding research funded by NIH. So if NIH funds a grant, it is assumed that the results will be published. The only mechanism for restricted access is classification right now. NIAID does not, nor will we fund or do classified research. So really the fundamental question with regard to our involvement is, and the discussion in general, is the issue is the risk to global health of the work that we fund, the risk of not funding that research versus the risk to the global health of the information harming society. That really is the critical question. So therefore, even though, as Harvey said, there are going to be no conclusions or consensus, from our standpoint, the question is, should or should we not fund this research? And that's the thing that we're going to be concentrating on in the discussions that occur. Thank you very much.
Oh, riddle me this. When has Dr. Fauci spoken about this gain-of-function dual-use bioweapon research on SARS, MERS, flu, and apparently every other viral disease known to man in any of his 2020 public speeches and reports, standing right next to President Trump regarding this man-made SARS-2 COVID-19?
Good morning, everyone. By way of introduction, I'm Amy Patterson. I've had the pleasure of emailing many of you in the room. In a phrase, we are here because of scientific responsibility. And while collectively we bring a broad, diverse range of expertise, from infectious disease expertise, epidemiology, public health, countermeasure development, safety, security, ethics, agriculture, law, we bring a wealth of diverse perspectives and opinions on the topic at hand.
We as a scientific community don't know what we're doing, so it's really important to, it's true, I know. Yeah.
But what we share is a common interest in advancing scientific understanding, but first and foremost, a desire to protect public health. And underpinning that common interest is a shared value, a value that when we conduct science, it's important for us to not only think about its aims, its purposes, but also about its implications, its potential repercussions for society. And that's essentially why we're here today. As Tony mentioned, the focus of this meeting is what we're referring to as gain-of-function research. And in the context of this meeting, we are referring to research that will confer new biologic properties, research that will confer new biologic properties by enhancing the transmissibility, enhancing the pathogenicity, or potentially expanding or altering the host range, that will confer new biologic properties by enhancing the transmissibility, enhancing the pathogenicity, or potentially expanding or altering the host range of highly pathogenic H5N1. And as Tony mentioned, the reason that is the focus of this meeting is because case reports to date, particularly since 2003, have underscored the high human mortality associated with human infection with this virus. The high human mortality associated with human infection with this virus. So the notion of potentially conferring additional biologic attributes to this agent is really what underscores the concern today. Conferring additional biologic attributes to this agent is really what underscores the concern today. As Tony mentioned, researchers in this community of work demonstrated their commitment to responsible science by declaring a pause on the research, a pause on research that would increase transmissibility of high path H5. That pause has been in place since January of this year, and we're just about to close out 2012. The community is seeking guideposts for its own path forward. And while this meeting is not going to make a decision per se about that pause, we believe that the discussions here today will illuminate the guideposts that are sought for by that community. And importantly, this meeting is going to provide a wide range of perspectives, hopefully ideas, concepts that we haven't yet thought of, that will inform the Department of Health and Human Services as we finalize our framework for how we will approach making decisions about which of this research we will support, if any, and if we do support it, under what conditions will it be funded and supported. The perspectives that you offer over the next 2 days are going to be taken to heart as we work to finalize this framework over the next several weeks. We believe, in addition, that this forum will make a significant contribution to the international dialogue on this issue. We hope that the meeting will highlight ideas, perspectives, and approaches that will inform your national efforts, for those of you who come from other countries, and help inform the way that you think about policy, the way you might approach your own funding decisions. And at this juncture, I'd like to quickly review just the topics on the agenda, and again, some of the meeting mechanics that I think might come in handy for you. Our first panel will entail a discussion of HPAI H5N1 gain-of-function research, its design, its trajectory, its hoped-for aims in terms of addressing public health issues. And after a very quick break, we'll segue into the next panel, which will focus on the risks and concerns associated with this research. Now, to aid you in communicating the proceedings of this meeting to friends and colleagues back home, a summary will be prepared in the coming weeks, and it'll be made available to you and to the public. Please know that this is a public meeting. We do have members of the press in attendance. And while we're not webcasting this meeting, we are making a video recording of the event, which we will plan to post for the benefit of those who could not be here today.
This is a classic example of how governments keep public information in the private. Or in other words, knowing that the public is not going to watch this type of forum. From local to city to county to state to federal governments around the country and around the world, this is how public forums remain hidden. As you can see here, the YouTube channel for NIHOD shows 2.7 thousand views of this first initial gain-of-function research. The welcome and introductory remarks we just heard. Then for panel 1, which we're about to hear, that dropped significantly to 1.1 thousand views. And of course, panel 2, the risks and concerns associated with this research, drops down yet again to 656 views. This has been up for 7 years, folks, and this is how many views it has had by the public. In other words, it hasn't been viewed by the public at all. And this is the reason I've included it here today. Otherwise, you'd never know it existed.
And with that roadmap in front of us, let's begin our first panel.
So a pleasure to thank you, Dr. Patterson. It's a pleasure to introduce the first set of co-moderators for the first panel, which will focus on the HPAI H5N1 gain-of-function research and its implications for global public health, and then they can actually introduce the panel members. So the co-moderators are Dr. Kanta Subarao, Chief of Emerging Respiratory Viruses Sections here at the National Institute of Allergy and Infectious Diseases of the U.S. National Institute of Health, and Dr. Robert Webster, member of the Department of Infectious Diseases in St. Jude Children's Hospital at Tennessee. Thank you.
Thank you, Dr. de Graaf. It's my honor to be invited to be a coordinator of this very important topic together with Kanta Subarao. I believe that Dr. Fauci, Drs. de Graaf and Finberg have given us our mission for today and very clearly laid the background. I want to step back in history for a moment or two. It was over 15 years ago that I went to Hong Kong and the initial outbreak was going on. 18 children were infected, 6 had died. The decision was to eradicate this virus from Hong Kong, and that was successfully done. And I recall at the party on New Year's Eve, the group, including myself, wished Margaret Chen great success at eradicating this virus from society. And that was our mistake. So look where we stand today. There are still something like 60% of those infected die. We don't understand the pathogenesis and mechanism of control. We don't understand the pathogenesis and mechanism of control. This virus has spread from that region of the world across much of Eurasia and is now endemic in some of the most highly populated areas of the world, including Egypt, Bangladesh, and Southeast Asia. So we move forward. We've heard the experiments that explain that this virus can be made transmissible. This virus can be made transmissible.
It has not been able to continue through human transmission. In other words, it has not developed the ability to go from person to person to person to person. We're hoping that it doesn't obtain the ability to be easily transmitted.
Now we have to face the downstream effects of those discoveries. This is panel 1 of the highly pathogenic H5N1 gain-of-function research, its implications for global public health. That's very important because the scientists have been talking to each other now for nearly 15 years, but the general public haven't bought in to all of the aspects and consequences of the recent research.
And our first speaker is Dr. Ron Fouchier, who's a professor in the Department of Virology at the Erasmus Medical Center in the Netherlands. Ron.
In 2007, the so-called Fink report listed 7 categories of experiments that could lead to dual-use research of concern, such as increasing virulence, or transmissibility of pathogens, or inducing resistance to drugs, such as increasing virulence, or transmissibility of pathogens, or inducing resistance to drugs. And those are the issues that are discussed today. This list contains 5 or 6 categories of research that have been used in H5N1 virus research to date. that have been used in H5N1 virus research to date. The reason why so many categories have been used is that this list of 7 of the Fink report exactly represents the research agenda of the infectious disease field. When outbreaks with novel pathogens occur, whether it's enterohemorrhagic E. coli or SARS or SARS or SARS or SARS or pandemic H1 or highly pathogenic H5N1, infectious disease specialists get asked the same question time and time again. Why does this pathogen cause such a severe disease, and why does it kill humans? Can this pathogen be transmitted between humans, and via which routes? Can this pathogen adapt further and infect additional hosts or cause different types of disease? Which drugs that we have work, and can the pathogens become resistant to these drugs? To answer these questions, infectious disease specialists have to use gain-of-function research. The HHS proposed framework for H5N1 research indicates that alternative methods exist to answer these questions. And it would be great if that were true. HHS has listed comparative genomics, predictive modeling, and loss-of-function experiments as examples of alternative methods. And although each of these methods can have significant value, none provide the definitive answers to the questions that were asked. These alternative types of experiments mostly yield answers that are partial, suggestive, and correlative. These experiments are therefore generally only performed as a first series of experiments, but the final proof of the pudding has to come from gain-of-function experiments.
Yes, Ron, we get it. But this is the equivalent to the logic that in order to know for sure the destructive power and patterns of radioactive dispersal of a nuclear bomb, we must drop one on a large, unsuspecting city. That way we'll know for sure.
Gain-of-function approaches have answered numerous critical questions from the public health perspective related to H5N1. And it's important to realize that some of these answers do not result in direct applications, but are critical for the advancement of science and public health on the longer term.
Dual use is clandestinely defined here by one of its main purveyors. Some of these answers, he says, do not result in direct applications, but are critical for the advancement of whatever science and public health in the long term means. In other words, the results have nothing to do with the purpose of the actual or current experiment being funded, but are done intentionally for dual use, or in this case, future purposes, because they would not be funded or allowed otherwise. The real problem, as we'll see, is that no one really knows what science means. Science is not the scientific method, but some corporate structure of patented ideas and biological life forms, artificial intelligence, and, of course, artificial life to go along with it. This will all become clear as we progress.
Examples of these are studies addressing the role of the HA basic cleavage site in virulence, or the role of HA receptor binding specificity in relation to tropism, host range, and transmission, or the role of NS1 in evading innate immune responses of the host. Through gain-of-function research, we now also know that H5N1 can become resistant to drugs. Through gain-of-function research, we now also know that H5N1 can become resistant to drugs, which has resulted in increased interest in drug design and drug discovery programs. We got to learn that H5N1 can become airborne transmissible in mammals. Resulting in stronger advice from the infectious disease community to countries where H5N1 outbreaks occur to stop these outbreaks, as H5N1 can no longer be seen as just a poultry problem. The transmission research further provide guidance to the WHO and FAO surveillance programs to get to the next level and allow the identification of parts of the world that should be prioritized for H5N1 eradication. So there's plenty of positive news for the advancement of science and public health that has come from gain-of-function research. When flu researchers like myself like myself from around the globe announced a voluntary 60-day ban on this type of research. This was done to provide some time for organizations and governments around the world to find solutions for the challenges and for the opportunities that stem from this work. And it is sobering to see how much time has been spent on the challenges and how little on the opportunities. As a consequence, the benefits from the gain-of-function research have not yet been claimed to the full extent. And this is why I have been a strong advocate to convince the NIH and other organizations to stop the research pause and give the green light to continue the research. To stop the research pause and give the green light to continue the research. We have identified key questions for surveillance, key questions for further fundamental research, and we have generated the tools and reagents to better evaluate vaccines and drug candidates. In my opinion, it is undesirable and perhaps even unresponsible to maintain a ban on this follow-up research. All of this research is aimed to prevent flu pandemics or to mitigate their impact if they cannot be prevented altogether.
Here again, we see this sociopathic logical fallacy card being played. We are supposed to believe that force evolving a more virulent, more contagious, more drug-resistant, more deadly, and of course, more patentable, and thus more profitable viruses will help prevent the spread and pandemic potential of the natural viruses we copy and intentionally mutate through gain-of-function research by causing those mutations that most often cannot even happen in nature in the first place. To compare what happens in the lab to what happens in nature is patently ridiculous.
So in summary, I argue that gain-of-function approaches are essential to advance science and public health in general, but certainly for H5N1. Gain-of-function approaches have contributed enormously to advance science and public health. Laws and regulations are in place, and they have been shown to work. Some incremental refinement may be required, but whole new layers of rules and regulations are not needed.
Thank you. Our next speaker is Dr. Aditama, who's the Director General of Disease Control and Environmental Health in the Ministry of Health in Indonesia.
Ladies and gentlemen, for Indonesia, the research is very important as a guide for risk assessment. As the virus evolves naturally, we need to monitor whether it is acquiring the mutation as found in the research. The research is also potentially to improve vaccine development program. I believe that the benefit of conducting gain-of-function research generally outweigh the risk as long as the research is done in accordance with the ethical clearance and in biosecure laboratory setting. At this point, I would like to raise the importance of active involvement and participation of the country scientists from where the virus is found to be able to solve the real public health problem in the field, in the community, in the people. Thank you, ladies and gentlemen.
Thank you very much. Our next speaker is Dr. Robin Robinson, who's the Director of the Biomedical Advanced Research and Development Authority and the Deputy Assistant Secretary for Preparedness and Response at the Department of Health and Human Services in the U.S.
Thank you, Kanta. Thank you, Rob. And thank you for the opportunity to be here this morning.
And so we have on this panel Ron Fouchier, the actual listed scientist that did this original gain-of-function work that caused this debate in the first place. And now we come to Robin Robinson. Now, does Mr. Robinson have any conflicts of interest here? Any reason he should not be on this panel, perhaps? Well, Dr. Robin A. Robinson is the Chief Scientific Officer for RenovaCare, a cell renewal company. Cell renewal being a definite dual-use qualification. From their website, we read that Dr. Robin A. Robinson is a respected authority on the development of breakthrough biomedical technologies and a seasoned expert in consummating collaborations with leading U.S. government agencies, Fortune 500 companies, academic research groups, and foreign governments, for which he was cited in 2018 as one of the top 100 innovators in medicine. Notably, Dr. Robinson was appointed as the first director of the Biomedical Advanced Research and Development Authority, or BARDA, at the U.S. Department of Health and Human Services. with an annual budget of $1.35 billion and a staff of 250 scientists and medical experts. He brought BARDA into prominence as one of the top 10 fully integrated research and development organizations worldwide, supporting advanced development and acquisition of more than 240 drugs, vaccines, diagnostics, and medical devices for man-made biodefense threats, pandemic influenza, and emerging infectious diseases, including Ebola and Zika viruses. Yes, Ebola and Zika viruses are currently receiving gain-of-function research studies. Man-made mutations. 32 of these medical countermeasure products that BARDA supported were approved and licensed by the FDA during his 12-year tenure. He concurrently served as Deputy Assistant Secretary, Office of Assistant Secretary for Preparedness and Response in the U.S. Department of Health and Human Services, under appointment by the Health and Human Services Secretary Michael Levitt. Prior to his public service at BARDA, of course, he was a pharmaceutical director of vaccines at Novavax, Incorporated, resulting in discovery and development of more than 12 vaccine candidates. Before entering the pharmaceutical industry, he was an assistant professor in the Department of Microbiology and Immunology at the University of Texas Southwestern Medical School, conducting research on the molecular pathogenesis of herpes virus and, surprise, HIV-1. During his tenure at BARDA, Dr. Robinson initiated over $535 million in contracts. It then goes on to say that Dr. Robinson is a consummate deal maker, having successfully established over 60 partnerships with federal agencies, including the United States Food and Drug Administration, or FDA, one of the most corrupt organizations in the world, Centers for Disease Control and Prevention, same, National Institutes of Health, the U.S. Army Medical Research Institute of Infectious Diseases, in other words, dual-use biological weapons, the Defense Advanced Research Projects Agency, or DARPA, again, heavy biological weapons research, the Joint Program Executive Office for Chemical and Biological Defense. Of course, the Department of Defense used to be called the Department of War, and the Defense Threat Reduction Agency. His more than 80 non-governmental and commercial partnerships included the Gates Foundation, the Wellcome Trust, the pharmaceutical companies like Sanofi, GlaxoSmithKline plc, Novartis, Merck, and Roche Genentech, Amgen, Johnson & Johnson, Crucell, the SIGA, Regeneron, and Emergent, and many others. Dr. Robinson has developed collaborations with universities and foreign governments, including the Johns Hopkins University, North Carolina State, and the United Kingdom, Canada, Australia, France, Germany, and others. At Revenacare, Dr. Robinson is responsible for setting the company's scientific strategy and leading our science and engineering teams in research and development activities. Dr. Robinson is a key participant in new product innovation and development and is charged with advancing the company's technology along with its FDA submission pathway. Just one more case of a pharmaceutical worker going into government and then back into private practice using his government connections for profit. Dr. Robinson oversees and directs Revenacare's strategy, execution, and engagement with government agencies, contract research organizations, academic institutions, and select opinion leaders. Dr. Robinson chairs the Revenacare Scientific Advisory Board. Now, does this sound like someone who should be deciding on the fate of the entire human population when it comes to dual-use or that is, gain-of-function research? No sane person could answer in any other way than absolutely not. And yet, here we are in this supposedly public forum.
The Biomedical Advanced Research and Development Authority at HHS in the Office of the Assistant Secretary for Preparedness and Response is a new agency established in 2006, but has been at the forefront in making available funds for supporting the development and the availability of countermeasures such as vaccines, antiviral drugs, diagnostics, and medical devices that would be needed in a number of different events, including biothreat events such as anthrax, smallpox, pandemic influenza, and emerging infectious diseases. Relative to this type of research, we have actually done an assessment, and we're not able to identify any of our research that was being funded being in dual use. But it's this last area of advanced development of medical countermeasures with H5N1 where we've moved forward with development of cell-based recombinant molecular vaccines and new antiviral drugs. And in that regard, the gain-of-function activities and research would have a very big effect also in our areas for diagnostics that we work very closely with Centers of Disease Control. And we would benefit greatly by being able to predict what new viruses are circulating and to be able to use the stockpiles that we have made available. The stockpiles have actually become very operative in the course of immunizing the vaccines, laboratory workers and manufacturing workers for these vaccines that make the H5N1 stockpiles. In addition, those stockpiles of antiviral drugs can be made available to those workers that are at high risk. It's the issue of what are the potential benefits and risks going forward if we weren't to do this research that I call your attention to, especially on the antiviral. Because of the prevalence of antiviral drug resistance occurring so quickly with some of the existing antiviral drugs that we have in our stockpiles, we and the NIH have had to look at other targets. And relative to viral targets, lack of gain-of-function studies would really affect some of the viral RNA polymerase inhibitors, antiviral drug candidates that we have and others in the U.S. government are sponsoring the development. Antiviral drug candidates that we have and others in the U.S. government are sponsoring the development. Because if those functions the mutations were to affect the function of those genes, that those targets would no longer be effective, then those antiviral drug candidates would become obsolete. The mutations were to affect the function of those genes, that those targets would no longer be effective, then those antiviral drug candidates would become obsolete. Those antiviral drug candidates would become obsolete. Secondly, host cell surface receptors. We've had to move to look at host targets for the effectiveness of antiviral drug candidates. And so as we move forward with, say, sialase surface receptors for the hemagglutinin binding, what effect would they have if we do not move forward those research? And lastly is an area in which the cancer field has moved very briskly forward, and is to understanding what are the gene expression profiles of a given disease, the treatment of that disease, and also the prophylaxis or adaptive immunity associated with the disease, in this case being influenza, to understand those genes that are expressed or whether they be turned off or turned down during an infection or vaccination or after antiviral drugs. And these gain-of-function studies would actually then limit us being able to understand that pathogenesis profile. Thank you.
Thank you, Robin. Our next speaker is Dr. Thomas Inglesby, who's the director for the Center for Biosecurity at the University of Pittsburgh Medical Center in the U.S.
Thank you, Kanta and Rob. Thank you, Dr. Fauci, Dr. Patterson. Three major benefits have been argued for creating lethal and transmissible H5N1: basic science understanding, surveillance, and vaccine and drug development. So will this research benefit basic science? I think yes, clearly, I think that's inarguable, and we'd all like to know more about the biological basis for transmissibility and pathogenesis and the genetics of H5N1. But in my view, this isn't enough to justify the risks. This isn't enough to justify the risks. So second, does the work seem to benefit surveillance for H5N1? And in my view, I don't see it as being practically useful for on-the-ground surveillance systems, but I'm sure we will have a discussion about that today. Only a tiny fraction of avian influenza infections in the world are sequenced, and when they are sequenced, it can take years to analyze them, and surveillance systems aren't set up to provide immediate sequence information that links to immediate action on the ground. It's still not clear to me what actions would be taken if surveillance found H5N1 strains that matched lab-engineered strains. Right now, the recommended action is to cull the flocks of birds known to be infected with H5N1. So if we found H5N1 strains in nature that matched the lab-engineered strains, what would we do beyond what is now recommended? Third, would engineering H5N1 strains to become transmissible benefit vaccine development? Over the past year, I don't think there has been a concrete argument sufficiently describing the case for this, but there are leading experts in the room today who I think will speak to this. And Robin, we just heard from Robin on this. So overall, are the benefits worth the risks? In my view, no. If high-path H5N1 is engineered to spread like seasonal flu, and if it escapes by accident or deliberately, millions of people's lives could be put at risk, even if the fatality rate is 100 times lower than the current WHO fatality rate of 59%. Once novel flu gets going in the population, it's unlikely we could stop it. Incubation time is short. Shedding occurs before fever's onset, so it spreads like wildfire. In 2009, H1N1 proved the point. By the time it was recognized, it was spreading around the world with no way to stop it. Flu strains circle the globe annually, infecting as many as 10 to 20% of the world, despite large supplies of vaccine and medicine. In the case of H5N1, we have only enough vaccine for a small number of people. So we shouldn't support the work with the belief that we could stop engineered H5N1 from spreading if it escapes from the lab accidentally or deliberately. So we shouldn't support the work with the belief that we could stop engineered H5N1 from spreading if it escapes from the lab accidentally or deliberately. Accidents are rare, but they happen. Mark Lipsitch wrote a valuable article in MBio describing the recent history of accidents, including lab accidents with SARS in 2003, the 1977 flu escape, and the lethal meningitis in a lab worker in California last year. Most accidents only affect lab workers, so they have little consequence in society beyond the lab. But accidents with engineered transmissible H5N1 could be a threat to the global community. On the proposed HHS framework for H5N1 funding decisions, I think it's a very good step forward. It's better to have it than not. And the criteria are all generally valuable. But I have concerns, which hopefully we can discuss later today. In specific criterion 4, I don't think biosafety risks can be sufficiently mitigated if we don't even agree on what the risks are. Some of us in the room think the high side risk is an H5N1 global pandemic, and some of us, I think, believe that there is not a serious risk. Criteria 5 and 6. It's one thing to say that physical security of strains can be guaranteed, but how can any scientist or any government agency know that publishing the work won't lead to replication with the intent to do harm? Even if we had perfect knowledge of the world of malevolent actors in 2012, we don't know what the world will look like in years ahead. Published information will be available forever. And finally, there's wide room to interpret the criteria, so reviewers will hold great latitude and power. Beyond the framework, here's my bottom line. I think we should continue the moratorium. I don't think the benefits of engineering H5N1 to become transmissible are enough to warrant the extraordinary risks. We should pursue aggressively alternative approaches to study transmissibility, safer strains, safer vectors, comparative genomics. I think this is a major moment for science broadly, and I do think we should explicitly seek the views of scientists not in the flu community or even here today. We should explicitly seek the views of scientists not in the flu community or even here today. And if we do decide to proceed with funding and with ending the moratorium, I think that we should all who are involved in this work acknowledge the extraordinary risk. The public has a right to know that everyone is on the same page regarding the risks. The public has a right to know, the public has a right to know, the public has a right to know that everyone is on the same page regarding the risks. I think we should also proceed in unprecedented biosafety conditions and with international agreements on how to proceed. Thanks.
Thank you. Our next speaker is Dr. Daniel Gerstein, who is the Deputy Undersecretary, Science and Technology, Department of Homeland Security in the United States.
Good morning. I'd like to present just two themes from a Department of Homeland Security perspective. The first is that H5N1 threat characterization studies, including these gain-of-function research, are essential for protecting against a broad range of biological threats, but we must also mitigate against the threats and risks of such research. And the second theme I'd like to develop is that I'd like to share with you some thoughts about how the Department of Homeland Security has developed capabilities for managing this dual-use research of concern that we hope might prove interesting to others. First, on the H5N1 gain-of-function research, here's how we see the dilemma. Obviously, there's benefits for public health and security communities. It's useful and valuable to understand the pathogenicity of transmissible H5N1. On the other hand, there is this risk of misuse of technology as well as the misuse of the information resulting from the experiments. And so what we have to think about is this cost-benefit equation. But let me state unequivocally that the Department of Homeland Security does strongly support gain-of-function research. Let me state unequivocally that the Department of Homeland Security does strongly support gain-of-function research. Does strongly support gain-of-function research. The Department of Homeland Security does strongly support gain-of-function research. It does further health preparedness and response. It promotes innovation, and we certainly don't want to constrain scientific discovery. We do threat characterization work on a wide variety of pathogens, and in fact, we would call this threat characterization work gain-of-function. And in fact, we would call this threat characterization work gain-of-function, and we feel it's absolutely essential for understanding disease, but most of what we do happens to be with respect to other what we would call bioterror agents. Most of what we do happens to be with respect to other what we would call bioterror agents, although we have done some work with highly pathogenic avian influenza. My second topic is really to discuss our approach to managing what we consider to be important but also potentially dangerous work. So when we think about it, let me just start out by saying there is some research that we believe should not be conducted. It is simply too dangerous. There is some research that we believe should not be conducted. It is simply too dangerous. And when we evaluate whether or not it's worthy of doing this sort of work, we rely on the NSABB, National Science Advisory Board for Biosecurity, 7 experiments of concern as an excellent foundation for thinking about the problems. We rely on the NSABB, National Science Advisory Board for Biosecurity, 7 experiments of concern as an excellent foundation for thinking about the problems.
The so-called Fink report listed 7 categories of experiments that could lead to dual-use research of concern. And those are the issues that are discussed today. This list contains 5 or 6 categories of research that have been used in H5N1 virus research to date. The reason why so many categories have been used is that this list of 7 of the Fink report exactly represents the research agenda of the infectious disease field.
We rely on the NSABB, National Science Advisory Board for Biosecurity, 7 experiments of concern as an excellent foundation for thinking about the problems. We also recognize that at lower levels, we have to train project managers and conduct appropriate biorisk management. We also know that we have to write into our contracts things like biosafety, biosecurity, internal review processes before publication, and looking at information that might result from our studies to see if any classified information might be released. We have also developed a structure which we call our Compliance Assurance Program Office, which helps us in looking at the various experiments to determine if they are violating any of the NSABB experiments of concern, or whether they raise perceptions of noncompliance with our BWC, Biological Weapons Convention, or CWC, Chemical Weapons Convention treaties. Biological Weapons Convention or Chemical Weapons Convention treaties. The Compliance Review Group meets on a quarterly basis, and they have the mission to look at this cost-benefit ratio as well. And some very practical questions in addition to the NSABB 7 experiments of concern that we ask ourselves are, does this experiment need to be done? Is the information necessary for science and or security? Are there other, more appropriate ways to gain the benefit of the information? What is the least dangerous way to accomplish the experiment? Is the experimental design sound? Is the experiment being conducted in the appropriate containment level? And are there limits on what results should be released or published? And so we do come back to this very delicate balance, this need for legitimate science with the need for security. And with that, I look forward to your questions. Thank you.
The takeaway here is that the Department of War, or Defense, that is, Homeland Security, has in its possession a homemade, weaponized, highly pathogenic, extremely virulent, and contagious H5N1 virus. Several variations, in fact. But if they label it as legitimate basic research, it's okay. And this is the foundational problem. Basic research that any scientist might do nowadays with current technology is the same exact research that would be done in biological weapons. This is why it is called dual-use research of concern. And remember, these homemade biological weapons are being stored in a military biosecurity lab near you, whose safety record is no doubt questionable. Not that it would matter, because this is a global disease. Release it in one country, it is sure to escape into another.
Thank you. Our next speaker is Dr. Adel Mahmoud, who's a professor of molecular biology and public policy at Princeton University.
Thank you very much. It's a pleasure to be with you here. The problem with the discussion we're having today is that it is happening after the fact. It is happening after the fact. I wish we would have had this discussion a little bit earlier, because when the Fink report came several years ago, it mentioned some of these experiments that we are now suffering from or discussing and concerned about. It mentioned some of these experiments that we are now suffering from, It mentioned some of these experiments that we are now suffering from or discussing and concerned about. So one issue is we are discussing it after the fact. The second issue is we're discussing it after the facts, which unfortunately got mixed up with public announcements about the results of research before the research was submitted and evaluated and published in a refereed journal. So we faced a public outcry and the public comments in the newspapers before even the data itself was examined and became a subject of scientific examination. Now, we need also to go back to understanding that we basically do not understand how pandemics of flu occur. We basically do not understand how pandemics of flu occur.
It's also really important because we as a scientific community don't know what we're doing,
We have been concerned about H5N1 for approximately 15, 20 years now, and it was H1N1 that came out in 2009. So in some ways, the necessity of research is absolutely upon us. I mean, nobody is going to say we do not and we cannot stop research that will examine why one strain of the flu virus will be transmitted into human populations and result in an outbreak or a pandemic, and another has been circulating around with very, very few cases, and in some ways, even the 59% of the announced mortality or case fatality rate of H5N1, I'm not sure that that is really accurate data, because I don't think we understand the epidemiology in humans that well to say, here is the denominator and here is how can we calculate the case fatality rate.
It's really important because we as a scientific community don't know what we're doing.
So we are now faced with two issues. One is a study that was done that was identified from the beginning, or a series of studies were done that were identified from the beginning under the rubric of experiments of concern. And they were approved and funded because they were legitimate studies that needed to be done to understand, even if we don't think H5N1 will go tomorrow to be transmitted in human populations, understanding its range of transmissibility and pathogenesis is a valuable scientific contribution. No arguments about that. The question is, A, approving it, B, understanding how it will be communicated, and C, funding it. And who should fund that kind of research, which is the question that Tony finally put on the table. Should it be funded by NIH or should it be funded by private source? So in effect, the unfortunate thing is that we're coming from behind. And the opportunity now is, as a community, we just have to start thinking a little bit going forward rather than just analyzing what happened in the past. And that's really a challenge, and I hope the discussion today will contribute to that. Thank you.
Thank you. Our next speaker is Dr. Ilaria Capua. She's the Director of Research and Development Department at the Institute of Zooprofilattico Sperimentale della Venezia, Italy. Ilaria.
Good morning, everyone. For those of you who don't know me, I run an international laboratory which is active in surveillance, mainly in animals, for influenza, mainly in birds, and particularly in developing countries. Malik Peiris, who's sitting next to me, wrote in a paper for Science, I think, that nature is the worst bioterrorist of all. So we know that there are countries out there which have endemic infection caused by H5N1, and they really don't know where to bang their head to get rid of this infection. These gain-of-function experiments, if they have a reason to exist, is because we really fear what is out there in the field. Because we think that there is a bioterrorist out there, which is Mother Nature, which can generate a virus that can become a very severe pandemic virus. So the work that has been carried out by the two teams is extremely important in identifying signatures or characteristics of these viruses so that we understand which viruses in the field have the potential of becoming potentially pandemic.
In other words, these so-called scientists have a belief system that they, mankind, can improve upon nature's or God's design, and more importantly, believe they, collective incorporated science, can protect man from nature and its supreme laws. Now, we are starting to uncover the foundational dual usage intention behind much of this gain-of-function mutation of nature. But this won't be made clear until the next panel is called. However, in an effort to understand this so-called scientific mindset of this scientific community, which is exclusive and even xenophobic of the general public, we're not dealing here with true or natural science. We're dealing with a religious belief system called scientism.
The science delusion is the belief that science already understands the nature of reality in principle, leaving only the details to be filled in. This is a very widespread belief in our society. It's the kind of belief system of people who say, I don't believe in God, I believe in science. It's a belief system which has now been spread to the entire world. But there's a conflict in the heart of science between science as a method of inquiry based on reason, evidence, hypothesis, and collective investigation, and science as a belief system or a worldview. And unfortunately, the worldview aspect of science has come to inhibit and constrict the free inquiry, which is the very lifeblood of the scientific endeavor. Since the late 19th century, science has been conducted under the aspect of a belief system or worldview, which is essentially that of materialism, philosophical materialism. And the sciences are now wholly owned subsidiaries of the materialist worldview. I think that as we break out of it, the sciences will be regenerated. What I do in my book, The Science Delusion, which is called Science Set Free in the United States, is take the 10 dogmas or assumptions of science and turn them into questions, seeing how well they stand up if you look at them scientifically. None of them stand up very well. What I'm going to do is first run through what these 10 dogmas are, and then I'll only have time to discuss one or two of them in a bit more detail. But essentially, the 10 dogmas, which are the default worldview of most educated people all over the world, are first that nature is mechanical or machine-like. The universe is like a machine, animals and plants are like machines, we're like machines. In fact, we are machines. We're lumbering robots, in Richard Dawkins' vivid phrase, with brains that are genetically programmed computers. Second, matter is unconscious. The whole universe is made up of unconscious matter. There's no consciousness in stars, in galaxies, in planets, in animals, in plants, and there ought not to be any in us either, if this theory is true. So a lot of the philosophy of mind over the last 100 years is trying to prove that we're not really conscious at all. So the matter's unconscious, then the laws of nature are fixed. This is dogma 3. The laws of nature are the same now as they were at the time of the Big Bang, and they'll be the same forever. Not just the laws, but the constants of nature are fixed, which is why they're called constants. Dogma 4, the total amount of matter and energy is always the same. It never changes in total quantity, except at the moment of the Big Bang, when it all sprang into existence from nowhere in a single instant. The 5th dogma is that nature is purposeless. There are no purposes in all nature, and the evolutionary process has no purpose or direction. Dogma 6, biological heredity is material. Everything you inherit is in your genes, or in epigenetic modifications of the genes, or in cytoplasmic inheritance. It's material. Dogma 7, memories are stored inside your brain as material traces. Somehow everything you remember is in your brain in modified nerve endings, phosphorylated proteins. No one knows how it works, but nevertheless, almost everyone in the scientific world believes it must be in the brain. Dogma 8, your mind is inside your head. All your consciousness is the activity of your brain and nothing more. Dogma 9, which follows from dogma 8, psychic phenomena like telepathy are impossible. Your thoughts and intentions cannot have any effect at a distance because your mind's inside your head. Therefore, all the apparent evidence for telepathy and other psychic phenomena is illusory. People believe these things happen, but it's just because they don't know enough about statistics, or they're deceived by coincidences, or it's wishful thinking. And dogma 10, mechanistic medicine is the only kind that really works. That's why governments only fund research into mechanistic medicine and ignore complementary and alternative therapies. Those can't possibly really work because they're not mechanistic. They may appear to work because people would have got better anyway, or because of the placebo effect. But the only kind that really works is mechanistic medicine. Well, this is the default worldview, which is held by almost all educated people all over the world. It's the basis of the educational system, the National Health Service, the Medical Research Council, governments, and it's just the default worldview of educated people. But I think every one of these dogmas is very, very questionable. And when you look at it, they fall apart. I'm going to take first the idea that the laws of nature are fixed. This is a hangover from an older worldview before the 1960s, when the Big Bang theory came in. People thought that the whole universe was eternal, governed by eternal mathematical laws. When the Big Bang came in, then that assumption continued, even though the Big Bang revealed a universe that's radically evolutionary, about 14 billion years old, growing and developing and evolving for 14 billion years, growing and cooling, and more structures and patterns appear within it. But the idea is all the laws of nature were completely fixed at the moment of the Big Bang, like a cosmic Napoleonic code. As my friend Terence McKenna used to say, modern science is based on the principle, give us one free miracle and we'll explain the rest. And the one free miracle is the appearance of all the matter and energy in the universe and all the laws that govern it from nothing in a single instant. Well, in an evolutionary universe, why shouldn't the laws themselves evolve? After all, human laws do, and the idea of laws of nature is based on a metaphor with human laws. It's a very anthropocentric metaphor. Only humans have laws. In fact, only civilized societies have laws. As C.S. Lewis once said, to say that a stone falls to earth because it's obeying a law makes it a man and even a citizen. It's a metaphor that we've got so used to, we forget it's a metaphor. But I want to spend a few moments on the constants of nature too, because these are again assumed to be constant. Things like the gravitational constant, the speed of light, are called the fundamental constants. Are they really constant? Well, when I got interested in this question, I tried to find out. They're given in physics handbooks. Handbooks of physics list the existing fundamental constants, tell you their value. But I wanted to see if they'd changed, so I got the old volumes of physical handbooks. I went to the Patent Office Library here in London, and they're the only place I could find that kept the old volumes. Normally people throw them away when the new values come out. They throw away the old ones. When I did this, I found that the speed of light dropped between 1928 and 1945 by about 20 kilometers per second. It's a huge drop because they're given with errors of only fractions of decimal points of error. And yet, all over the world, it dropped, and they were all getting values very similar to each other with tiny errors. And then in 1945, it went up, 48, it went up again. And then people started getting very similar values again. I was very intrigued by this, and I couldn't make sense of it. So I went to see the head of metrology at the National Physical Laboratory in Teddington. Metrology is the science in which people measure constants. And I asked him about this. I said, what do you make of this drop in the speed of light between 1928 and 1945? And he said, oh dear, he said, you've uncovered the most embarrassing episode in the history of our science. So I said, well, could the speed of light have actually dropped? And that would have amazing implications if so. He said, no, of course it couldn't have actually dropped. It's a constant. So, well then how do you explain the fact everyone was finding it going much slower during that period? Is it because they were fudging their results to get what they thought other people should be getting, and the whole thing was just produced in the minds of physicists? We don't like to use the word fudge. I said, well, what do you prefer? He said, well, we prefer to call it intellectual phase locking. So I said, well, if it was going on then, how can we be so sure it's not going on today? And that the present values are produced by intellectual phase locking. And he said, oh, we know that's not the case. I said, how do we know? He said, we've solved the problem. And I said, well, how? He said, well, we fixed the speed of light by definition in 1972. So I said, but it might still change. You said, yes, but we'd never know it because we've defined the meter in terms of the speed of light. So the units had changed with it. So he looked very pleased about that. They'd fixed that problem. But I said, well, then what about big G, the gravitational constant known in the trade as big G? It's written with a capital G, Newton's universal gravitational constant. That's varied by more than 1.3% in recent years. And it seems to vary from place to place and from time to time. And he said, oh, well, those are just errors. And unfortunately, there are quite big errors with big G. So I said, well, what if it's really changing? I mean, perhaps it is really changing. And then I looked at how they do it. What happens is they measure it in different labs. They get different values on different days, and then they average them. And then other labs around the world do the same, and they come out usually with a rather different average. And then the International Committee on Metrology meets every 10 years or so and average the ones from labs around the world to come up with the value of big G. But what if G were actually fluctuating? What if it changed? There's already evidence, actually, that it changes throughout the day and throughout the year. What if the Earth, as it moves through the galactic environment, went through patches of dark matter or other environmental factors that could alter it? Maybe they all change together. What if these errors are going up together and down together? For more than 10 years, I've been trying to persuade metrologists to look at the raw data. In fact, I'm now trying to persuade them to put it online on the internet with the dates and the actual measurements and see if they're correlated, to see if they're all up at one time, all down at another. If so, they might be fluctuating together, and that would tell us something very, very interesting. But no one has done this. They haven't done it because G is a constant. There's no point looking for changes. You see, here's a very simple example of where a dogmatic assumption actually inhibits inquiry. I myself think that the constants may vary quite considerably, well, within narrow limits, but they may all be varying. And I think the day will come when scientific journals like Nature have a weekly report on the constants, like stock market reports in newspapers. You know, this week, big G was slightly up, the charge on the electron was down, the speed of light held steady, and so on. That's one area, just one area where I think thinking less dogmatically could open things up. I don't have time to deal with any more of these dogmas, but every single one of them is questionable. If one questions it, new forms of research, new possibilities open up. And I think as we question these dogmas that have held back science so long, science will undergo a reflowering, a renaissance. I'm a total believer in the importance of science. I've spent my whole life as a research scientist, my whole career. But I think by moving beyond these dogmas, it can be regenerated. Once again, it can become interesting and, I hope, life-affirming. Thank you.
I believe the right way and the meaningful way to understand what science is and to define science, if you like, is through its methods rather than in terms of its topics. And the important point is that if science has well-defined methods, it may not be the case, and in fact it isn't the case, I'm going to argue, that those methods apply to all forms of knowledge. So what are science's methods? I think science has some very distinctive characteristics, most of which we're all kind of familiar with, although we perhaps haven't made a list of them, but they're things that are listed up here, things like observation, experiment, measurement, systematization, mathematization, and so forth. And these characteristics of science, I believe, can be brought together into two primary abstract categories. And so we can really, in a certain sense, boil down what we mean by natural science into the insistence upon reproducibility. So science depends upon repeatable experiments or observations. And clarity, and this means the unambiguous descriptions, things like measurement, sometimes things like mathematics, that science insists upon. And these characteristics, I would say, imply that science's scope of application is limited. What is scientism? Scientism is the belief that science is all the real knowledge there is. And I think scientism is at the heart of the question of the relationship between science and Christianity, or science and religious faith. Scientism is not a finding of science. It's a worldview. It's a worldview that has an integrating cosmology. That is, it has an integrated understanding of how the world is and what it fundamentally goes to make it up. It has an interpretive lens or filter. In other words, it's something that you look through that colors your interpretation of the world if you adopt a scientistic position. And it has a number of other things. For example, it has a justifying narrative history. It has, you might be surprised to know, it has a community of believers, and those believers, that community, look to scientism as a source of ethics. Now, why have I pointed out these things out? I've pointed them out because I submit that these are the kinds of characteristics of a religion.
Scientism exposed.
It might come to a surprise to most people that one of the most dangerous worldviews one can hold is scientism. The failing of science is not really about the scientific method. It is about a largely unrecognized epistemology that developed as a result of the successes of science. This faulty epistemology can be called scientism, and we should make a sharp distinction between science and scientism. Science is a method of inquiry and the knowledge acquired by that method. The scientific method involves hypothesizing, experimenting, observing, and drawing conclusions. Central to the method of science is that no theory is ever considered final. All theories are subject to scrutiny and re-examination, and it is assumed that all will eventually be proven false by a more comprehensive theory. Scientism, on the other hand, is the belief that the methods of science are superior to any and all other methods of determining truth. Science believes things because they have been observed to be true. Scientism believes things because science says they are true. Most of us are guilty of scientism to some extent. We hear about things having been scientifically proven and we assume that means they must be true. We didn't do the experiments. We didn't look up the results. We didn't analyze the data. We simply assumed that if the science proved it, it must be true. As humanity emerged from the Middle Ages, a battle began to rage between the fathers of the Church and the new intellectuals of the Enlightenment. That struggle continues today, even though it had seemed for a time that the debate was over and that the science was settled.
We will restore science to its rightful place and wield technology's wonders.
The issue is that science is no longer science. True science is simply the scientific method or empirical data. What is commonly called science these days is actually just the religion of the world, full of fabrication and an anti-God agenda.
The debate is over. The science is settled.
Exposing scientism for what it is, you have science and then you have non-science. And nowadays you have a lot of stuff that's masquerading as science, and there's a force behind that. There is an adversary that we have in this creation that sets about to lie about everything.
I see scientism as a spiritual agenda. I see it as a spiritual deception. It's something that has been put in place and masqueraded as fact and as reality, when in fact most of the things that are paraded today are theories of men.
The deception will pull you any direction, and this is where scientism is. I think this is the whole purpose of scientism. I think there are dark forces behind it trying to pull people into what they think is logic, and it's really speculation, it's really theory. It's not as concrete as everybody thinks it is.
Science is great. Scientism, however, is a religion, is a belief system.
Scientism assumes that science is the controlling reality about life, so anything that can be validated scientifically ought to be done. Other things are subjective fantasy, like love, beauty, good, evil, conscious, and ethics. Science, which originally simply meant the study of the natural world, has in this view become conflicted with scientific naturalism, materialism, and a philosophy that the natural world is all that exists. Humans are reduced to objects that can be inspected, experimented on, and ultimately controlled. In 1922, G.K. Chesterton warned that scientism had become a creed taking over our institutions, a system of thought which began with evolution and has ended in eugenics.
And again, remember, there's science and there's scientism. What have you fallen for? What have you believed in? Has it been real science or has it been scientism?
We no longer feel ourselves to be guests in someone else's home and therefore obliged to make our behavior conform with a set of pre-existing cosmic rules. It's our creation now. We make the rules. We establish the parameters of reality. We create the world. And because we do, we no longer have to justify our behavior, for we are now the architects of the universe. We are responsible to nothing outside of ourselves, for we are the kingdom, the power, and the glory forever and ever.
Darwin considered this by far the strongest single class of evidence. This textbook says, the human embryo growing in the mother has gills like a fish. Those little folds of skin are not gills. Those little wrinkles under your chin when you're growing up later develop into bones in the ear and glands in the throat. They never have anything to do with breathing. I've seen folks that have 5 or 6 chins and they can't breathe through any of them but the top one. Those are not gill slits. Ernst Haeckel, though, said the turning point in his thinking was when he read Darwin's book. He made huge charts of his posters of his drawings of these embryos and traveled all over Germany and just about by himself converted the Germans to believing in evolution. Haeckel took a drawing of a dog and a human embryo and he changed them to make them look exactly alike. On top are Haeckel's fake drawings, underneath are actual photographs of what he claimed he was drawing a picture of. Now either he's a lousy artist or he's a liar. Well, it turns out he's a liar. He was convicted of fraud by his own university, proven to be a fraud. But guess what? Haeckel's fake drawings are still used in textbooks in your state right now. It's only been proven wrong 125 years ago. I know it takes a while to get textbooks up to date, but that ought to be plenty of time. Adolf Hitler said, you let me control the textbooks, I'll control the state.
You have science, you have true science, which is actual knowledge, things that are repeatable, testable, observable, different facts and things that you can really dig into as reality. But then you've got this thing in modern day society called, well, what's referred to now as scientism. It's a lot of theology dressed up as science. And it needs to be exposed. Because as Christians, we want to focus on the truth. We want to focus on God, face Him, and just ignore all the distractions. But while we're facing God and we're observing that, we also need to be aware of what the distractions are so that we can help others understand. And so we want to expose the lies for what they are.
As the saying goes, the greatest trick the devil ever played was convincing the world that he didn't exist. And to do that, he has to first convince the world that God doesn't exist. And through science, I think he's achieved it. Through scientism, science itself is a beautiful thing, but scientism, when it turns into a theology, when it turns into a religion, that is when it's a very bad thing.
Our culture's materialistic worldview is rooted in scientism. According to scientism, if something is not rational or not verifiable through the physical senses, then it is not real. We see then that scientism blinds us to everything in reality that is beyond the scope of the scientific method, no matter what the method may reveal to us. So how much of reality is left out? Almost all of it.
Make no mistake, the very foundation of all mankind's knowledge depends on what is believed to be the truth about the origins of all that exists. No one can go further back than the beginning in a search for the true origin of all that exists. Therefore, whatever one believes is the truth about the origin of everything automatically becomes the foundation upon which all other knowledge is processed.
In the beginning, there was darkness, and then bang, giving birth to an endless expanding existence of time, space, and matter.
And they know the biggest problem with that is how can you have something come from nothing? So now you have these scientists that start talking really nutty, like the biggest problem with cosmology today is nothing. You've heard that probably.
Essentially, you can get a universe from nothing without any supernatural shenanigans. That basically, by quantum mechanics and the laws of physics we understand in principle, an entire universe with 100 billion galaxies, each containing 100 billion stars, can come from nothing because its total energy could be zero. And therefore, you don't need to literally violate any laws of physics to create a universe. And no, we don't know that for certain, but even that possibility is amazing. And I find it worth celebrating the fact that we can probe such things, and we shouldn't be afraid of a universe that maybe has no purpose, because we create our own purpose, and our own purpose adds more meaning to our lives. It means our brief moment in the sun matters because that's the only moment we have.
Which it means that everything came from nothing and then exploded. So when you really drill this stuff down to the basics, it doesn't make much sense. But since you're hearing this 24/7, and all the school systems teach it, nobody even thinks to question it.
And so you have this same thing in the mainstream science world, scientism. You have people that are studying cosmology and astronomy and physics and chemistry and biology and all this stuff, but a lot of times they're not even studying the real thing. Instead, they're just studying what somebody else has said about it. And then those people, they know stuff based on what somebody else had said. And so it keeps building on, the deception keeps going until finally every once in a while somebody in the middle of that wakes up and starts to look at the evidence and says, hey, wait a minute, this is not lining up with what we're teaching and what we're believing. Something's wrong. And of course then that guy is immediately ousted, not just for challenging the authority, because that can be a good thing. Sometimes they like that. But if you're challenging the authority and bringing in real truth, that they don't like.
It was a paradigm shift. There is now another way to explain the naturally occurring phenomena around us, and this is science.
Since the Middle Ages, scientists have developed sophisticated new theories about the enormity of the universe and our place in it.
God is an ever-receding pocket of scientific ignorance.
It really is the most poetic thing I know about physics. You are all stardust. You couldn't be here if stars hadn't exploded, because the elements, the carbon, nitrogen, oxygen, iron, all the things that matter for evolution weren't created at the beginning of time. They're created in the nuclear furnaces of stars, and the only way they can get into your body is if the stars were kind enough to explode. So forget Jesus. The stars died so that you could be here today, okay? And anyway.
Pythagoras identified numbers as the most fundamental elements of creation.
As we look at science in this day and age, we're finding that the whole world is filled with these mathematical constants. You've got pi, you've got the speed of light, you've got the force of gravitation. So the idea that numbers describe the world seems to be the case.
He wanted to produce philosophers like himself, so you had to be very circumspect, you had to be very self-controlled to be able to even be in his school.
Pythagoras turned the numbering system of the Babylonians into a sacred science.
They believed that number was like God, and to connect with this was to be talking to God. And so from that arises a whole lot of symbolism that later becomes numerology, Kabbalah, things like that.
So the long and the short of it is that the Pythagorean mysticism, fetching disguised as something scientific, reduced practically everything to mathematical numbers. Does that ring any kind of bell in our information age, frequently called the digital age? Because factual math is so dependable, so exact, so capable of producing technological marvels, people have had very good reason to trust anything that is said to be mathematical supportable. The general belief is that both math and science are strictly objective enterprises and strictly secular tools in the hand of a rather elite band of altruistic laborers for mankind. The theoretical science establishment has cultivated the image that it is uniquely removed and aloof from any contamination by philosophies and religions. The establishment's mantra is that mathematics and science represents a dedicated search for truth.
Archaeology is the search for fact, not truth. If it's truth you're interested in, Dr. Tyree's philosophy class is right down the hall. So forget any ideas you got about lost cities, exotic travel, and digging up the world. We do not follow maps to buried treasure, and X never, ever marks the spot. 70% of all archaeology is done in the library. Research, reading. We cannot afford to take mythology at face value.
And dark matter is a great example. The whole idea of dark matter came from the idea that galaxies spin too fast. They spin so quickly that the observable matter, which is based on light, it's the light that we can see from the galaxies, that there isn't enough mass there, that they're spinning so fast that they should fly apart based on the conservation of angular momentum. So we've made this assumption that there's more mass there that we can't see, and that's what dark matter is. That's where it comes from. It's based on that assumption that we see these things out there in space that are, very, very far away, and that they're spinning so fast and there's all this mass there that it defies the basic laws of physics here on Earth. And so besides the idea that might not even be true, that those things might not be that far away, we're forcing things in the night sky to have to comply with our physics here on Earth. Now that in itself is an enormous assumption, but besides that, dark matter comes from that assumption. So we just build assumptions upon assumptions. And then dark energy, the other one that is supposed to make up an enormous amount of our universe, this dark energy comes from assuming that the law of gravity is true, because gravity says that all masses attract all other masses, but we have this infinitely expanding space. Everything's moving away from each other, but supposedly some calculations were done, I think in the 70s, that showed that long ago, since the Big Bang, they assumed Big Bang happened, that all of the masses should have started pulling each other back together, that all of these galaxies and everything should be coming back together into one big mass. But calculations have shown that that's not true, or the theory is that there's this dark energy that's pushing all of the galaxies apart. So it's counteracting this assumed force of gravity. So now we assume that there's another force that's pushing them all apart. And not only that, is galaxies appear to be disks, whereas gravity would want to make things spherical, or make things orbit in a spherical type state. So the dark energy is not only pushing all of these galaxies away from each other and arranging them in weird ways that doesn't really seem to make sense, it's also flattening galaxies into disks to give them the shape. So we have this mysterious force called dark energy. That's where these theories come from. They're based on assumptions, and it's really, like I said, it's kind of a house of cards. And so I started, I didn't realize it, but at the time I started imagining men in lab coats just figuring these things out. And it's like, who are these people? And now I look at this, and when I see this stuff now, that scientists have a new theory on semi-radial tachyons or something I saw recently about how those are some new particles they're finding. And I see these men in these white lab coats, and I kind of laugh to myself, like, who are these people? They're never really described to us, and it's just all these theories, all these crazy things that just come up in the news about science this and science that. And don't get me wrong, I love science, but scientists, it's like they're priests almost, and it's getting out of hand. We really got to just take a step back. Wait a second here. We're in dreamland here. There's a book I'm reading by a man named Lee Smolin who's a theoretical physicist, just saying, whoa, we got to back up and get back to testable, scientific method physics. And it doesn't seem like mainstream science wants to go that route. It really seems like They want to keep all of this theoretical stuff that really, to me, is just kicking the can down the road, avoiding the question, who made this? Just add more and more time, more and more billions of years, more and more expanding infiniteness that has no explanation, no beginning, no real beginning at least.
Science is supposed to be point A, B, C, and D. You come up with a hypothesis. You then come up with a way to test that hypothesis, and hopefully it's a repeatable method that you can get the same results. And you can then verify if your hypothesis is true or false.
Well, then who did create the heavens and the earth?
Why do you use the word who? You see, you immediately beg the question by using the word who.
Well, then how did it get created?
Well, by a very slow process.
Well, how did it start?
Nobody knows how it started. We know the kind of event that it must have been. We know the sort of event that must have happened for the origin of life.
And what was that?
It was the origin of the first self-replicating molecule.
Right, and how did that happen?
I told you we don't know.
So you have no idea how it started?
No, nor has anybody.
Nor has anyone else. What do you think is the possibility that intelligent design might turn out to be the answer to some issues in genetics or in evolution?
It could come about in the following way. It could be that at some earlier time, somewhere in the universe, a civilization evolved by probably some kind of Darwinian means to a very, very high level of technology and designed a form of life that they seeded onto perhaps this planet. Now, that is a possibility and an intriguing possibility. And I suppose it's possible that you might find evidence for that if you look at the details of biochemistry, molecular biology. You might find a signature of some sort of designer.
Wait a second. Richard Dawkins thought intelligent design might be a legitimate pursuit?
And that designer could well be a higher intelligence from elsewhere in the universe. Well, but that higher intelligence would itself have had to have come about by some explicable or ultimately explicable process. It couldn't have just jumped into existence spontaneously. That's the point.
So Professor Dawkins was not against intelligent design, just certain types of designers, such as God.
We've backed ourselves so far into this corner of bowing to science and twisting the Bible to agree with science that we've handicapped ourselves in major ways, especially with what's coming down the pike in terms of scientific discovery.
C.S. Lewis thought that science was a good thing, but he also thought that it held some really strong dangers. The biggest danger really was the penchant to control. In a scientific view, if you think that is the only way that we have knowledge of the world, and so if you think that if I have the scientific truth about something, that's, end of story, I know everything, that really tends to feed a power trip. Whether you're a scientist or you're a politician who's trying to latch on to the prestige of science, you really have people who are going to abuse their power because they think, look, we're the only ones who know what should happen because we know how the universe really works. Therefore, we should be able to dictate what our cultural beliefs are. We should dictate what our government should do, how we should design governmental programs. We should dictate all manner of public policy, and that anyone who doesn't have a scientific training or isn't part of the consensus view of science is basically stupid.
I think scientism is taking us to some very dark places in the near future. Basically becoming gods themselves. I think this is the whole idea that Satan has spawned with men, is to become like God and to tinker with life when it comes to the DNA, when it comes to cloning, when it comes to creating the perfect human, the superhuman, merging animal species with humans, being able to give human gills, to live underwater, just these superhuman abilities to become like gods, to become supermen. I think this has been the
quest of scientism for a very long time while deceiving the world.
We're going to be diving into the brave new world of CRISPR. And as you see, as you saw in that film, CRISPR gives scientists the awesome power to tinker with the software of life.
For thousands of years, humans have bred horses in much the same gradual way nature does. But soon, they could actually be designed in the lab, along with other livestock, plants, and even humans. Because a new technology has made editing the genome almost as easy as using a word processor. For better or for worse, every life on Earth will soon be affected by our ability to reprogram the software of life.
I didn't want to let the evening go without hitting the headlines that you made yesterday when you, with your article in Science, talking about the Human Genome Project, Right. You called for a huge publicly and privately funded project to construct large plant and animal genomes, including humans, from scratch.
And this was before the era of easy gene editing that we have now, but about 10 to 12% of these reproductive age people said that if there were a test that would be predictive of superior intelligence or superior athletic ability in their children, well, they would want to have it. And as we've seen, for instance, with consumer genetics, that people won't be denied in terms of the types of information that they'd like to be able to achieve. But
once it's proven to be safe for particular ways, we're going to have a conversation about what the limits are for how we would use that technology. There are still people who would, who, like I said, see that as a line in the sand that we should never cross. But my belief is that we will and should cross it.
They ask questions that sound a little bit like science fiction, but many of them are becoming science fact very quickly. Do you think, I mean, you're actually pushing, the science forward. Do you think the consequences are well understood, the sort of leveling of the plate?
Yeah, I don't think we understand. I mean, luckily for us, we sort of sidestep in regenerative medicine. We get a chance to sidestep this issue for a little while longer because it's still a bit of a hypothetical question. Can we grow body parts that are, right now, we're just trying to catch up to what nature can do. And so for a while, we get the chance to just, I think similar to people in AI, to get a chance to say, oh, we're just trying to catch up to what nature can do. But eventually, it's going to catch up to us, and we'll have to start answering these questions. Do we want to enhance and do more than what nature can do?
It turns out that the really intelligent people are the only ones who actually get the boost, and suddenly there is kind of a superpower that you could have just for a small few. That would be really remarkable. It would be contra everything we've learned so far, but it would be remarkable, and it might change the conversation somewhat. But I think, from my perspective, what I'd rather do, rather than putting the brakes on it, is increase access and availability so
Even
that
if
people...
that entails a little bit of creative destruction.
Well, why would it cause creative destruction?
Consider the statement, quote, "We should only believe what can be scientifically proven," end quote. Can that statement be scientifically proven? Well, obviously not. And thus the scientific naturalist's position refutes itself, and so it cannot be true.
This indoctrination and replacement of true natural science with the religious dogma of scientism manifests itself in many ways and causes ripple effects throughout actual scientific studies. In fact, we find that because of this scientistic view of the world, this strange lens that so-called scientists look through to see the world, the unfortunate results can be accounted for by simply looking at the so-called scientific studies that are produced through scientism instead of science. The Washington Post, for instance, reports that many scientific studies cannot be replicated, and that that's a problem. BBC News in February of 2017 reported that most scientists can't replicate studies by their peers. Likewise, The Guardian reported that a recent study delivers a bleak verdict on the validity of psychology experiment results. Of 100 studies published in top 10 ranking journals in 2008, 75% of social psychology experiments and half of cognitive studies failed the replication test. A very good sign that the scientific method is indeed not being followed. The University of Idaho reported that its study finds scientific reproducibility does not equate to scientific truth. From May of 2019, it states that reproducible scientific results are not always true, and true scientific results are not always reproducible, according to a mathematical model produced by University of Idaho researchers. Their study, which simulates the search for that scientific truth, was published today, May 15th, in the Journal of PLOS 1. Independent confirmation of scientific results, known as reproducibility, lends credibility to a researcher's conclusion, but researchers have found the results of many well-known science experiments cannot be reproduced, an issue referred to as a replication crisis. Over the last decade, people have focused on trying to find remedies for the replication crisis, said Berna Deviser, but proposals for remedies are being accepted and implemented too fast without solid justifications to support them. We need a better theoretical understanding of how science operates before we can provide reliable remedies for the right problems. Our model is a framework for studying science. When science itself as an institution needs to be studied for the reasons why it has a replication crisis, it becomes fairly obvious that the cult of scientism has taken over. Clear back in 1999, we had the same problem as science reported that data in key papers cannot be reproduced. Another journal reported that way less than 30% of science articles are reproducible. This in 2012. Studies show very low reproducibility for articles published in scientific journals, often as low as 10 to 30%. Here is a partial list. And it gives examples such as the biotech company Amgen and a team of about 100 scientists trying to reproduce the findings of 53 landmark articles in cancer research published by reputable labs in top journals. Only 6 of the 53 studies were able to be reproduced, equating to about 10%. Scientists at the pharmaceutical company Bayer examined 67 target validation projects on oncology, women's health, and cardiovascular medicine. Published results were reproduced in only 14 of 67 projects, about 21%. The Project Psych file drawer, dedicated to replication of published articles in experimental psychology, shows a replication rate of only 3 out of 9, or 33% so far. The commenter states that my hair is standing on end as I read these numbers. Unbelievable! The reproducibility of published experiments is the foundation of science. No reproducibility, no science. If these numbers are true, or even half true, it means there is something fundamentally wrong in today's system of scientific research and education. On a practical level, the U.S. government gives nearly $31 billion every year in science funding through the National Institutes of Health only, which is mainly distributed in research grants to academic scientists. The 10% reproducibility rate means that 90% of this money, or $28 billion in taxpayer money, is wasted. That's a lot. How are the taxpayers supposed to respond to the scientist plight for more research funding given these numbers? Would you give more of your own money to someone who delivered you such a result? Beyond the practicalities, there is also an interesting philosophical question. Since the middle of the 20th century, life science research concepts and technologies have rapidly grown from the discovery of DNA to sequencing of genomes. Amazing technologies like microarrays, mass spectrometry, and high-throughput assays, imaging, and robotic surgeries were introduced, making biology a data-rich science. One would expect that all these new tools would make science more rigorous and precise, but something opposite is happening. Any ideas? What could be the reason? Who is the main culprit? The International Weekly Journal of Science, ironically called Nature, states in August 2015 that over half of psychology studies fail reproducibility tests. The largest replication study to date casts doubt on many published positive results. Don't trust everything you read in the psychology literature, Nature states. In fact, 2/3 of it should probably be distrusted. The journal The Conversation in 2017 reported about the science reproducibility crisis and what can be done about it. Back to the International Weekly Journal of Science, or ironically Nature, we find that 1,500 scientists lift the lid on reproducibility. Survey sheds light on the crisis rocking research. This in May 2016. It reports that more than 70% of researchers have tried and failed to reproduce another scientist's experiment, and that more than half have failed to reproduce their own experiments. Those are some of the telling figures that emerged from Nature's survey of 1,576 researchers who took a brief online questionnaire on reproducibility in research. The data reveal sometimes contradictory attitudes towards reproducibility. Although 52% of those surveyed agree that there is a significant crisis of reproducibility, less than 31% think that failure to reproduce published results means that the result is probably wrong. And most say that they still trust the published literature. In layman's terms, we'd call that indoctrination, and this perhaps gives us an idea of the percentage of how many so-called scientists out there really belong to the religion of scientism. In the PLOS journal, we read, "Why most published research findings are false." Dating from August 30th, 2005, the abstract summary states that there is an increasing concern that most current published research findings are false. The probability that a research claim is true may depend on study power and bias, the number of other studies on the same question, and, importantly, the ratio of true to no relationships among the relationships probed in each scientific field. Simulations show that for most study designs and settings, it is more likely for a research claim to be false than true. Moreover, for many current scientific fields, claimed research findings may often be simply accurate measures of the prevailing bias. Published research findings are sometimes refuted by subsequent evidence, with ensuing confusion and disappointment. Refutation and controversy is seen across the range of research designs, from clinical trials and traditional epidemiological studies to the most modern molecular research. There is increasing concern that in modern research, false findings may be the majority or even the vast majority of published research claims. However, this should not be surprising. It can be proven that most claimed research findings are false. Now, one thing you must start being conscious of, considering the vast array of so-called side effects from drugs, vaccines, and other pharmaceuticals, if the falsities of these so-called scientific studies is indeed this bad and probably worse, and when we consider that pharmaceutical companies are actually doing their own studies, they are not independent studies for their own drugs, then we must also consider that many of these falsified studies, these irreproducible studies, are the studies being used to prove both the safety and efficacy of these chemical drugs, vaccines, and other pharmaceuticals. And if this is the case, we no longer have to wonder why the whole population seems to be sick and dying. Even the pharmaceutical companies' own rags ask the question, should drug companies conduct their own clinical trials? And in this article from Pharma Times, we read that drug firms carrying out clinical trials on their own medicines creates an unacceptable conflict of interest. Increasing evidence points to a conflict of interest for the drug industry, which results in bad evidence, which in turn distorts medical decision-making and harms patients. And finally, one of the problems is that the industry can choose which data to publish and which to leave unavailable, pointing to the difficulties in getting clear information about the number of suicide attempts in industry trials, for instance, referring to Merck's anti-inflammatory drug Vioxx, which was removed from the market only after the FDA claimed it was safe and effective due to the pharmaceutical company's so-called scientific research. Over and over, we see systemic problems with clinical trials, research papers, and so-called science. PubMed's editorial on drug research, marketing before evidence, sales before safety, has in its title an obvious conclusion. The Washington Post reports that as drug industry's influence over research grows, so does the potential for bias, and that companies should have no role in testing their own drugs. Another article from the New York Times from 2013, which states the pharmaceutical industry has responsibility for evaluating its own products. Even on the face of it, I'm sure you'll agree, this makes no sense. Again, it can be proven that most claimed research findings are false. As has been shown previously, the probability that a research finding is indeed true depends on the prior probability of it being true, that is, before doing the study at all, the statistical power of the study, and the level of statistical significance. Essentially, this boils down to whether or not you're seeking the truth. Remember, the truth needs no science to prove it is the truth. Truth is self-existent, self-evident. But in modern so-called science, they're not seeking the truth. They're seeking to alter, to manipulate, to mutate the truth into their own creation. That is, into an invention that is patentable, sellable, for profit. Thus, these so-called scientific tests cannot be passed, for no truth has ever been reached. The only facts that are being produced are not of nature. Therefore, their reproducibility, when searching for truth, is null and void. The Guardian again reports that it's not any more breaking news. Many scientific studies are ultimately proved wrong. Most theories are eventually consigned to the rubbish heap, but this is scientific business as usual. True science has been so far abandoned that now these so-called scientists are stating the reasons why it's time to publish research failures. Publishing bias favors positive results. Now there's a movement to change that. This is very much a double-edged sword. On one hand, negative results will certainly help these so-called scientists in their dual-use research. On the other hand, we find that in these unpublished testing results, if published, many drugs and vaccines would never be allowed to carry the label of safe and effective. After hours of testing, retesting, checking, and confirming, Dr. Jones closes her lab book. Her experiments have not disproved the null hypothesis, and they don't support her new theory. She leaves her negative results behind, moving on to work on something new in the hope that she'll have a positive outcome next time. Meanwhile, what happened in her lab will stay in her lab. No point in publishing negative results, after all. Or is there? Publication bias affects the body of scientific knowledge in different ways, including skewing it towards statistically significant or positive results. This means that the results of thousands of experiments that fail to confirm the efficacy of a treatment or vaccine, including the outcomes of clinical trials, fail to see the light of day. Now, this should disturb you that thousands of experiments that fail to confirm the efficacy and safety of vaccines, that they are failures and fail to be published. In other words, the information you need to know as an informed vaccine recipient, including deciding whether your children should be vaccinated, is never published. The amount of people that are harmed in these trials, in other words, is never published. The Conversation Journal again asks, back in 2013, scientific evidence, what is it and how can we trust it? He states that the phrase scientific evidence has become part of the vernacular, thrown about like a hot potato during discussions of major environmental, health, or social issues. Climate change is one example. The EU's ban on neonicotinoid pesticides is another. We've heard numerous mentions of the associated evidence indicating the importance of the issue and the need for action. This evidence is presented by proponents in much the same way that evidence is given in a court case, usually to back up policies or decisions that will impact people's lifestyles. But unlike in a court case, we are rarely told exactly where the evidence comes from and why it's evidence. So-called scientific evidence is information. Remember, information is not knowledge. Scientific evidence is information gathered from scientific research, which we've just shown is more often wrong than right, untrue rather than true, which takes a lot of time and patience to conduct. But there are a few things that all this research needs to have in common to make it possible for decision makers and ultimately all of us to accept it as evidence. Unfortunately, it appears that time has passed, that the institutions of science, and that includes all arts and sciences, unfortunately, from psychology to medicine to pharmaceuticals to what we're speaking of today, the corruption is so deep and ingrained in most scientific research and publications, including the very journals that publish them, that the very foundations of so-called science and its institutions can never be trusted again. Yet another article from Science Alert in 2015 states that scientists tried to replicate 100 psychology experiments and 64% failed. A landmark study involving 100 scientists from around the world has tried to replicate the findings of 270 recent findings from highly ranked psychology journals, and by one measure, only 36% turned up the same results. That means that for over half the studies, when scientists used the same methodology, they could not come up with the same results. CBS News in January 2016 reported Bad Science, 5 most notable studies retracted in 2015. Now, listen carefully. The scientific method is a painstaking process of observing nature, in other words, truth, reality, asking questions, formulating testable hypotheses, conducting experiments, and collecting data, and then sometimes just making stuff up when reality doesn't match your expectations. Or maybe it just seems that way when you're reading through the retraction notices that scientific journals are posting with greater and greater frequency. There has been a tenfold increase in the percentage of scientific papers retracted because of fraud since 1975, according to a study published in 2012 in the Journal Proceedings of the National Academy of Sciences. Among the more than 2,000 retracted life science papers that researchers reviewed in this study, only about 20% were retracted because of honest errors. A whopping 70% were pulled as a result of scientific misconduct, that is, lying, cheating, and/or stealing. And most recently, in April of 2020, the Science Daily reported many published psychology experiments lack evidence of validity. From the Virginia Commonwealth University, an examination of nearly 350 published psychological experiments found that nearly half failed to show that they were based on a valid foundation of empirical evidence, suggesting that a wide swath of psychological science is based on an untested foundation. Now, this brings us to a very important point. And that is to say that much of the so-called scientific research that is happening behind the scenes in many or most of these research projects have nothing to do with the actual reasoning, intention, or conclusion of the project. In other words, the funding that is going towards these projects is being used for nefarious purposes, that is, dual use purposes. And so I can clearly and truly state that after the hundreds of research papers and publications that I have been through, more often than not, the conclusion of the study has nothing to do with the research that was done. That is to say that much of the research being conducted was what you might call side work, on the side, or completely irrelevant to the study at hand. And so, as the H5N1 researcher Ron Fauchier stated earlier, much of this research, which is often not reproducible or even necessary, is done for future use. In other words, a dual use, or use for later, let's just say, nefarious research purposes.
But the contradiction is that surveillance is going down in the field, and we try very hard to receive isolates from these countries, and by the time they reach our labs, they're already 6 months old, and by the time they're sequenced, it takes another couple of months, and by the time they're available to the scientific community, it takes another month. And so the question I have, and I would like to pose, is this, is how can we say that these gain-of-function experiments are so important if they are not counterbalanced by efficient surveillance in the field? And the same refers, I think, to the countermeasures developed. If I were to say to you that if a virus in one of these countries were picked up, it wouldn't be picked up in time before it would cause a lot of problems. It wouldn't be picked up in time before it would cause a lot of problems. And I would lie to you if I said to you that these countries are able to react fast, because they're not. Because they have other priorities and they have other problems, and we hear every day on TV what is happening, for example, in Egypt. So to wrap up, what I wanted to say is that if we really believe that these experiments are essential for all the reasons that have been listed before by the speakers before me, then we must fund adequate surveillance programs which cannot be based on a personal relationship basis, like we have a relationship with this university or that university. We need to make sure that international organizations like FAO and OIE have funds to continue capitalizing on the infrastructure that was built over the past 10 years. Because all of those investments that were made in surveillance, in capacity building, and in opening up the repositories and generating knowledge from those countries, unfortunately, is going down because of lack of international funds. Thank you.
Thank you. Our last panelist to speak today is Dr. Malik Piris, who's a professor at the Center of Influenza Research and the School of Public Health at the University of Hong Kong. Malik.
Thank you, Kanta and Rob. I'd like to address this primarily from the point of view of public health and primarily on the issue of gain-of-function research and understanding transmissibility. There were two lessons I think I would draw from the recent pandemic of 2009. The first is the fact we heard about the emergence of a novel pandemic virus in April 2009. By August 2009, 45% of children in Hong Kong had already been infected. Now, if we wanted to try to blunt that first pandemic wave, none of the current strategies of vaccine development would be able to achieve that. None of the current strategies of vaccine development would be able to achieve that. And Ilaria pointed out the question, my comment about nature being the greatest bioterrorist, but nature is also the most successful gain-of-function exponent. The blind force of evolution and trial and error is a formidable force that we have to deal with.
Bear in mind that this notion that nature or God is the worst bioterrorist of all is not meant as religious, for this is organized crime against nature or God and its universal self-evident law. The blind force of evolution here is equated to the design or law of nature, or in other words, chaos theory, where evolution is evidence of God's blindness or non-existence, and therefore man's destiny is to both conquer and bring forth a new world order to the universe, or that is God and nature, through the various arts of so-called science. These, again, are the tenets of scientism, not natural science. But the true source behind this fallacious justification for unnatural dual-use research is still yet to be revealed.
But nature is also the most successful gain-of-function exponent. The blind force of evolution and trial and error is a formidable force that we have to deal with. And as I said, transmission remains one of the black holes of biology and infectious disease. Transmission remains one of the black holes of biology and infectious disease.
Because we as a scientific community don't know what we're doing.
And I'd just like to end by saying that we of course have to address the ethics of action, but we also need to address the ethics of inaction. So as was pointed out, this balance is something we do have to address, but do not forget the ethics of inaction. Thank you.
Thank you, Malik.
Well, I'd like to thank all of the speakers for keeping on time and presenting their work. I'd like to go back to Dr. de Graaf and ask, are there any comments or Dr. Finberg that you would like to raise before we have a cross-panel discussion?
No one on the panel expressly, although perhaps indirectly, Dr. Gerstein referred to the question about the benefits from the research being public as opposed to the research being conducted. And I wonder if any on the panel would like to comment on that aspect of the questions on the implications and benefits for this research.
Anyone on the panel volunteering, or shall I nominate someone to speak? Because that's my role. Ron, would you like to respond?
Of course, always, Rob. I do not think that science would advance if the data would be disclosed, shared only with a very small group of people with a legitimate need to know. We've seen it in physics, I think. I think in physics there's good examples of non-public information and how it actually stops the field moving forward. And I do not think that we have that luxury position in this type of research. So these data have to be shared. So I really don't think that classifying this research and not sharing the data is that that's a valuable option at all.
Dr. Mahmood, did he really answer your question?
No.
That was my impression too. And so maybe we should redirect the question again to him. Was the information, scientific information, fully made available? Did the press get this information too soon?
It is my impression that that's not the case. Of course, we did not. So we get the impression that all of this work and all of the results were kept under the radar, which is absolutely not the case. We were asked to address this question through consultations within an international community within the WHO and also a blue ribbon panel of the NIH. So the fact that this research was being done has been open from the beginning to the end. There have been several publications with negative data throughout the year since 2006. All of those have been published, and then it would be inappropriate to not publish positive results. In the Netherlands, we have a code of conduct on doing dual-use research, and this code of conduct says that the scientists should not act as Dr. Jekyll in their labs. We have consulted with the NIH, with our own university, with other experts in the flu field, whether we should openly discuss our results at the meeting prior to the publication. And the answer was unanimously yes. The infectious disease specialists were of the opinion that this work should be publicized openly, and this decision was not made individually by myself. And therefore, I think that we have been very responsible in communicating this result. It was very unfortunate that there was one person in the press who then felt that she should be reporting this in the written literature without proper knowledge of fact. And that has been very unfortunate. So from the beginning, wrong information has come out, not from me, but from the lay press. But there's unfortunately very little I can do about that.
Other comments on this topic around the table?
Without really spending the time on this subject, that is the danger of communicating outside the normal scientific methods, which is we have data, we submit it to a refereed journal, and there are some journals also that will prevent you from presenting the data publicly before it is submitted for publication. So I think in general, the scientific community has some sort of guidelines. Data need to be communicated through a refereed, peer-refereed journal and a scientific publication before we start talking about it to the public. Now, we cannot blame a journalist because that's their job. They want to run up with any information that creates havoc or creates news. For them, it sells newspapers.
Maybe we should move on and look at other topics. I'm not convinced that our panel really addressed each of these questions very thoroughly. Kanta, what is your feeling about? Did our panel do a good job of answering our 4 questions?
So I think maybe the 4th question, are there risks associated with not conducting the research? I think some of the panelists have answered that, but maybe we can open it up and see if anyone who didn't, who addressed specifically what their expertise was in, would like to add anything. So this gets back to what you call the ethics of inaction. Malik. Does anyone have? No?
No, we were silence on that one. So
okay.
Let's turn to question number 3 then, and to Robbie Robinson, or really probably 2 as well. What changes or benefits have come to decisions within stockpiling, vaccines, antivirals, after the publication of these studies? What changes have occurred? Have they brought benefits? Have they brought you problems?
Thanks for the question, Rob. As I indicated in my earlier remarks, one of the direct effects was that we needed to provide or ensure that H5N1 vaccines and antivirals were available not only to the workers in vaccine manufacturing facilities, but also to laboratory workers. So we have an active program now to provide those vaccines to those that may be at risk. An H5N1, working with one of the vaccine manufacturers across, at least in the U.S., that we've done that. So that's a direct effect.
And the question is directed to our resident drug salesman. If the purpose of mutating a virus through gain-of-function research is to study new or novel traits of infections, in other words, those which have not happened before in nature, because you cannot patent anything that is real, anything that is natural, it must be new. And so these folks always have to create something new and fresh and patentable and profitable. And so if the purpose of mutating a virus through gain-of-function research is to study new or novel traits of infection, including these exact same 7 points of contention listed as dual-use research of concern, then what possible benefit would a vaccine for an unmutated virus confer to a lab worker whose job it is to create new mutations, that is, new strains of the virus, some of which are specifically designed to utilize new receptors and to be therein resistant to vaccines and antiviral drugs, as any good bioweapon should be?
Whether or not we have moved forward with different antiviral drug development or vaccine development, I think what it did, it actually confirmed what we were doing already. We knew that we were going to have to move to different classes of antiviral drugs. This really points that way, and as I mentioned earlier, we are looking at other targets, other viral targets such as RNA polymerases, host surface targets, and there will be more. This is an area in which we feel that, and NIH feels together, that we need to move more briskly, and without the results of some of these studies, we're not going to really understand whether these antiviral drugs are going to be effective. I did not talk about vaccines, but as we go forward with the stockpiling, it really points to a concept that has been talked about, and that is that can we stockpile the signatures of these viruses in a way, molecularly into these sequences, such that we can have a pandemic stockpile that would be more closely aligned to a possible H5N1 pandemic, with what if it becomes more, the virus will become more transmissible. Without understanding that whole concept really becomes obsolete, and we can't go forward with it at all.
Ah, the age-old question, which comes first, the chicken or the egg? Or in this case, the disease or the cure? This mad, scientistic, fallacious logic approves the invention of a disease agent so as to procure a preventative vaccine for such a man-made lab-grown virus that would never exist without first being invented or purposefully mutated and patented through gain-of-function research. And finally, our resident drug dealer approves the sharing of that research so that all bad actors around the world have access to and can invent their own remutated strain, a strain the U.S. government would not, therefore, be prepared for. But why? Because he's an international multi-level drug pusher and marketer, and there's a very limited market for drugs and vaccines without first artificially creating viral chimeric diseases. It's just business.
And I would say that we have actually moved somewhat forward with this over the past 2 years that was independent of these results, of actually putting together a stochastic mechanism of stockpiling vaccine antigens in a different way. Stockpiling of the virus reagents, stockpiling of the virus strains, stockpiling of actually antigens, without actually making huge stockpiles too. But certainly we have 4 different strains of H5N1 that we have over the past 7 years maintained. Certainly we have 4 different strains of H5N1 that we have over the past 7 years maintained.
Let me translate again for you. We produce and stockpile vaccines for one-time mutated strains, which we know are never going to be used simply because we know the virus will naturally or unnaturally by us mutate again, making the current vaccine useless against the new mutated strain of the future. In this way, we make profits from both this stockpiled waste and from future, mostly unused vaccines that we know must be wasted again when the virus mutates or is purposely mutated by us again in the future. In this way, all of my business clients are satisfied.
Certainly, we have 4 different strains of H5N1 that we have over the past 7 years maintained. Thank you.
Thank you.
Can I just ask or make one comment? I think as part of this process, it would be useful, in addition to hearing the good comments from Robin, to have a panel of representatives from industry whose job it is or will be to make H5N1 vaccine in the event of a pandemic, and to ask them explicitly, what are your research needs for vaccine development going forward? How much do they overlap with the gain-of-function experiments that are being envisioned? Perhaps some of them are useful to vaccine developers, perhaps some of them aren't. I have an open question still about whether continued research in increasing transmissibility of HPAI, H5N1 is necessary for future vaccine development. I think everyone likes to have it, but is it necessary in terms of vaccine development going forward for H5N1? My understanding, tell me if this isn't true, Robin, is that in the event of a future H5N1 pandemic, we're going to need that strain that's causing the pandemic to generate the vaccine that we make for the world. We're going to need that strain that's causing the pandemic. Tell me if this isn't true, Robin. We're going to need that strain that's causing the pandemic to generate the vaccine that we make for the world. But in any event, I certainly don't make vaccines for a living, but I think the people who do make vaccines for a living should be heard in this debate if one of the primary justifications for going forward is vaccine development.
Dr. Pieris, would you like to have a response to that, please?
Yes, I mean, I think the benefits from this type of research is not to specifically identify the strain that's going to be matching the pandemic exactly, not to specifically identify the strain that's going to be matching the pandemic exactly, but number one, to prioritize the fact that a lethal virus such as H5N1 can become pandemic, not to specifically identify the strain that's going to be matching the pandemic exactly, but number one, to prioritize the fact that a lethal virus such as H5N1 can become pandemic. Because if you just think back to 4 or 5 years ago, there was increasing belief that this virus had been kicking around for 14, 15 years. It has not made it so far, and it will never do so.
But is it necessary, Malik, to use the highly transmissible pathogenic virus to do those studies?
If you wanted to answer the question of whether H5N1 can acquire mammalian transmissibility, there is no other way to do it other than use the highly pathogenic H5N1 virus. Now, if you're asking whether you could, for example, delete the cleavage site of that particular virus and do this with a less pathogenic strain, possibly, but I think by deleting the cleavage site, you change quite significantly the viral load, the replication characteristics. By deleting the cleavage site, you change quite significantly the viral load, the replication characteristics. So it probably will not give you the crucial answer.
I just have one comment. Dr. Paris's comments, we're talking about the need to do this work in order to show or to prove H5N1 could be transmissible. I think there's a legitimate debate in the community about whether or not that was an important and justifiable aim for the research, about whether that work should have been done to prove the point that H5N1 could be human transmissible, to prove the point that H5N1 could be human transmissible. Whether or not you agree with that point now, it has been proven. It has been proven. It has been proven. So the question there would be, do we need to continue to do more work, additional work, create additional strains, H5N1 transmissible strains, to prove that it can become human transmissible when that point is made? I don't think that continues to be a good justification for going forward.
Ron, could I ask you to mention the benefits of going forward from where we stand?
Of course. So Malik nicely mentioned the word black hole for our knowledge of transmission research. The word black hole, black hole, black hole.
Because we as a scientific community don't know what we're doing, don't know what we're doing, don't know what we're doing.
Black hole for our knowledge of transmission research, and that's really where we were a year ago. And these two studies have not shed very bright light on that black hole, have not shed very bright light on that black hole. We just now know that it is possible in principle that H5N1 can become airborne. We just now know that it is possible in principle that H5N1 can become airborne, that H5N1 can become airborne. The mutations that we have identified have not become available through any other type of research than gain-of-function research, through any other type of research than gain-of-function research, through any other type of research than gain-of-function research, and we need to follow up on the function of those mutations that we have identified. They were an unknown, they were an unknown, they were an unknown, and they form a very fundamental question in virus transmissibility. We have shown that an Indonesian virus or a Vietnamese virus can acquire this ability to go airborne. We have shown that an Indonesian virus or a Vietnamese virus can acquire this ability to go airborne, We do not know about Egyptian viruses in China. And so there are particular genetic lineages of H5N1. We need to investigate whether these mutations or other mutations could also drive other viruses to go airborne. We need to investigate whether these mutations or other mutations could also drive other viruses to go airborne,
In other words, the accidental release of any man-made virus could cleave onto and inadvertently mutate, that is, add a gain or loss of function to, some other viruses already existing in, or that is, created by nature or man. As a dual-use biological weapon would weaponize or activate a previously released but incomplete, that is, inactive or non-lethal virus, the otherwise harmless half of what is now a bioweapon. But since they don't know yet, these mad scientists want permission and funding to forcibly cause it to happen unnaturally in a lab, risking this very scenario to accidentally happen in nature if it escaped or is weaponized.
So those are just very short-term action points, to understand the basis of this increased transmissibility, the context dependence, and also to do vaccination studies and antiviral drugs to test whether they would actually work against these viruses that have very distinct biological properties, So far, we have been testing our vaccines and drugs with viruses with the wrong biological properties. We now have strains at hand that have the right biological properties. Might not be the exact strains that caused the pandemic. These might not be the exact strains that caused the pandemic. But will inform us much, much better about whether our vaccines and antivirals will actually work.
The insanity goes on. Man-made, that is, lab gain-of-function viruses, are obviously not the same strain that may cause a natural pandemic. But we're supposed to believe that these deadly man-made strains will somehow show if vaccines work against future naturally mutated strains? If this is true, then why do we supposedly need a new flu vaccine every year? Answer? Because all viruses mutate in nature, despite man's psychotically driven interventions.
Now I'd like to turn it over to Kanta to lead the public discussion.
Right.
The one question that I have is Gary Monk from Hackensack, New Jersey, is we're tossing around biorisk, But in the laboratories that have been doing this work, from bioassessment, have you developed a baseline on your laboratorians? Do you look for seroconversions to see if transmissibility within a contained environment is actually affording laboratory safety?
I think Ron, did you?
I cannot answer that question because our laboratory workers have been vaccinated against H5N1, and so it would be impossible to show seroconversion of those people because they have been vaccinated.
In other words, there is no way to test lab technicians working on gain-of-function viruses like H1N1 or SARS for a new infection with newly contagious lab-mutated viruses because vaccinated lab workers already show antibodies to the root virus structure, which is similar enough to the newly mutated strain that no new antibodies, that is, seroconversion, would appear in the blood. So what about testing for COVID-19 antibodies? Ironically, Ron here just exposed the testing fraud for what it is. That is, if anyone has ever been exposed to any natural or man-made coronavirus in the past, and there are plenty of them, including the cause of the common cold, the test cannot distinguish between the new lab-mutated strain we call SARS-2 and, well, the common cold. You will show positive for quote-unquote coronavirus. From a 2020 article in Time magazine, we read, "What to know about COVID-19 tests, from PCR to antigen to antibody tests." People often talk about COVID-19 testing like it means only one thing, but in reality, the U.S. Food and Drug Administration, the FDA, has so far granted emergency use authorization to more than 200 different tests meant to detect a current or past infection from SARS-CoV-2, the virus that causes COVID-19. These COVID-19 tests fall into three main categories: the PCR, antigen, and antibody tests. And this article explains the differences between those three tests. The most popular test is the PCR test, a test that its own creator said was not to be used as a diagnostic test. It states that the majority of COVID-19 testing happening in the U.S. right now uses polymerase chain reaction, or PCR, technology. These tests detect disease by looking for traces of the virus's genetic material on a sample most often collected via a nose or throat swab. The U.S. Centers for Disease Control and Prevention, the CDC, considers the PCR test the gold standard of COVID-19 testing. But, like all tests, they are not perfect. Studies have suggested as many as 30% of COVID-19 PCR tests results are inaccurate. For comparison, the CDC in 2018 estimated that rapid flu tests have about the same rate of incorrect results. With COVID-19 tests, false negatives seem to be much more common than false positives. So if you get a positive result, you very likely do have the virus. If you get a negative result, but have the coronavirus symptoms or recently encountered someone sick with the virus, you should still self-isolate until symptoms subside. And here we have another example of very bad science. Again, the creator of this PCR test was very clear that this test should not be used as a diagnostic test. However, the CDC considers it the gold standard, meaning the best possible test you can take for COVID-19, and the gold standard has a 30% inaccuracy rate? Well, apparently we do have an accuracy or clarity problem, a reproduction problem in science now, don't we? And that's because this test simply looks for a corona-type virus, not a specific strain. To be clear, this is why vaccines don't work. All viruses mutate spontaneously in nature. There is no vaccine against mutation. And we've heard these so-called scientists state that very fact, that mutation is the very problem with creating a vaccine, and it is why some years the flu vaccine has been a completely different strain than the one that actually is making up the season. But don't take my word for it. Let's listen to what the world's so-called experts have to say about the seasonal flu's ability to self-mutate.
Influenza is unpredictable. Influenza is unpredictable. Influenza is unpredictable, and we need to monitor both the impact of the 2009 H1N1 and seasonal flu throughout the next several months.
The one thing you could predict about influenza is that it's unpredictable. It's unpredictable. It's unpredictable. It's unpredictable.
The virus still is very stable. But as we all know, influenza virus is highly unpredictable, highly unpredictable, highly unpredictable.
Но, как нам всем известно, вирус гриппа очень непредсказуем.
And has great potential for mutation.
Our goal since the beginning of the outbreak is to be one step ahead of the unpredictable virus, the unpredictable virus, the unpredictable virus, one step ahead of the unpredictable virus, and do the planning and preparation necessary to keep Americans safe and secure, the President's #1 priority. We want to help stop the spread of the H1N1 virus.
The outbreak in the past year has been characterized by a great deal of unpredictability, and in fact, influenza may be the most unpredictable of all infectious diseases. The most unpredictable of all infectious diseases. The most unpredictable of all infectious diseases.
As people keep telling me, the predictable fact about the flu is it's unpredictable. It's unpredictable. It's unpredictable.
Interestingly, in the time it would take to conduct a randomized double-blind placebo-controlled study on each year's regular flu vaccine, the flu season would already be over before the shots were ready for distribution and safe public use.
Vaccines, given the current technology, and even in more modern technology, you don't make a vaccine overnight. You have to first find out what the virus is you're dealing with, and then you go through a multi-step process in order to get enough vaccine to protect the population. That multi-step process generally takes several months, usually along the line of 6 or more months.
Take the first question here now.
Harvey Feinberg brilliantly pointed out at the time, in the 1970s...
Could you identify yourself,
Laurie
please?
Garrett from the Council on Foreign Relations in New York. Thank you. In the 1970s, we had a dominant theory that all biologists in the flu field believed in, of sort of historic cyclings of strains, and that there was a sort of timing predictability of when pandemics might occur. And that contributed to the over-response in 1976 to swine flu. Now I'm hearing that, well, we know the 2 mutations, the 3 mutations, the 4 mutations necessary, so Ilaria is just going to have to set up her entire biosurveillance system just for those mutations. And if we can keep following those mutations, we somehow will know when a pandemic's coming. But Ron just told us we have whole lineages we haven't looked at. There might be a whole other range of mutations. So are we actually setting ourselves up for building a whole biosurveillance apparatus, vaccine response, diagnostics, everything against a false canard? And nature, the ultimate bioterrorist, will give us a completely different set of mutations that ultimately will be the pandemic strain.
I just wanted to add that maybe I was completely misunderstood. What they have shown is that this combination works, which doesn't mean that other combinations of mutations won't work. So the point I was coming from is that surveillance, if we really need to improve it, needs to be improved so that we can look for these mutations, but we don't have to leave other potential mutations out and other combinations out, which we don't know about yet.
Right. Thank you.
Hi, Dawn Woolley, Wright State University. With regard to these natural household transmissions that were talked about, in particular the ones mentioned from Indonesia, have we fully exploited those natural transmissions? Have we learned everything that we can learn from that before going on to create new variants in the lab that may or may not ever occur? Before going on to create new variants in the lab, before going on to create new variants in the lab, to create new variants in the lab that may or may not ever occur?
Would you like to answer that?
It's important to note that a lot of this comparative genomic type of analysis is being done on transmission cases and has identified critical changes in H5N1, but not the ones that we identified through gain-of-function series of experiments. Series of experiments. And so the mutations have been there in previous pandemic viruses, H1s, H2, H3, but simply by doing comparative genomics on existing clusters of human-to-human transmission has not led to the identification of the critical mutation. That information has been buried under other information, and the only way to dig up that information and to show it there are actually other phenotypes required to confer airborne transmissibility is by doing gain-of-function approaches.
Translation: the only way to study a mutation that hasn't actually happened in reality, in nature, and which probably won't, and very likely cannot happen in nature without man's intervention through gain-of-function mad science, is to cause such a mutation artificially in a lab setting by forcing an unnatural mutation or evolution that otherwise is never to be found in nature.
The question from this microphone.
Stefan Elba, I'm a professor of international relations at the University of Sussex in the United Kingdom, and I have a question to the panel about the international aspects of this, because of course one of the things we're very aware of is that there's great variability around the world in terms of countries to respond to public health emergencies. So when we're doing these kind of gain-of-function research experiments, we are generating new risks. And however small we perceive them to be, they disproportionately affect people around the world, in the sense that some countries will have very limited abilities to respond to any kind of new dangers that might emerge as a result of these kinds of experiments. Some countries will have very limited abilities to respond to any kind of new dangers that might emerge as a result of these kinds of experiments.
The obvious, even self-evident answer: when creating a deadly viral bioweapon intended to infect as many people as possible, the intention is to spread the virus in a country that is, indeed, totally unprepared to fight against the virus. Wouldn't be much of a weapon if the enemy had a cure.
So my question to the panel is, would it actually be easier for us to deal with some of these issues if we had more meaningful international collaboration to share medical countermeasures and to share the kind of benefits of this research as we move forward?
The actual applicable question is, would you feel better knowing you've just created an unnatural, lethal strain of highly infectious virus by design, which could perhaps be used as a dual-use bioweapon if you shared the whole genetic sequence of that now weaponized virus with the rest of the world in easy-to-obtain, open-access format, so that any lab, government, or terrorist cell, or a guy in his underwear in his garage can then recreate it despite any type of safety level or oversight?
The second question, just very briefly, is has anybody thought about the legal ramifications of this? If anything did ever happen, what the kind of legal exposure would be for scientists or funders if a new virus were created and did get out, if anything did ever happen, what the kind of legal exposure would be for scientists or funders if a new virus were created and did get out, and did cause substantial economic and human harm. Thank you.
Yes, I'd like to follow up on two pieces of that. First, on the international, I think it's absolutely essential that we have international collaboration.
The conflicts of interest here are phenomenal. Here we find Daniel M. Gerstein is, in fact, a senior policy researcher at the Washington office of the RAND Corporation. If you're not familiar with the RAND Corporation, you should be. Mr. Gerstein has a PhD in biodefense, attending George Mason University, has an MS in national security, a National Defense University, an MMAS in National Security Strategy, U.S. Army Command and General Staff College, an MS in Operations Research from the Georgia Institute of Technology, and a BS in Engineering from the United States Military Academy. Now, we read from the USA Today that Apple and Google release coronavirus contact tracing technology for public health mobile apps. This on May 20th, 2020. Apple and Google to harness phones for virus tracking. New software the companies plan to add to phones would make it easier to use Bluetooth wireless technology to track down people for who may have been infected by coronavirus carriers. That April 10th from the Associated Press. Apple and Google are releasing contact tracing technology to help public health officials develop mobile apps that can identify and test people who've spent time with or near someone infected with the coronavirus, the companies announced Wednesday. A number of U.S. states, including North Dakota and Alabama, and 22 countries have received access to the technology that was designed to help governments better track and contain the spread of the virus, the companies said. In a statement, North Dakota Governor Doug Bergman described the smartphone technology as being critical to getting communities and economies back up and running. This technology is very much supported by the RAND Corporation. To give you just one aspect of this company's influence in our culture, we see that Captain Kirk's predecessor in Star Trek was RAND Corporation Predictive Programming. In 2013, it was reported that in a rare and recently unearthed interview from 1965, the actor who preceded William Shatner as first captain of the Enterprise stated that the series was based on the RAND Corporation's projection of things to come. Actor Jeffrey Hunter, who played Captain Christopher Pike in the Star Trek pilot The Cage, told a Hollywood columnist in January of '65 that he hoped the pilot episode would be picked up as a series because he was intrigued by the fact that the series was based on the RAND Corporation's projection of things to come. Here we see the opinion of RAND, which is that automatic contact tracing should be integrated into the pandemic response system with widespread access to testing and a large number of manual contact tracers. And recently they released this video, COVID-19, the realities of automated contact tracing. The Atlantic reports, we were warned. When the inevitable inquiry into the government's response to COVID-19 happens, it will conclude that signs of a coming crisis were everywhere. How were we warned? We were warned in 2012 when the RAND Corporation surveyed the international threats arrayed against the United States and concluded that only pandemics pose an existential danger in that they were capable of destroying America's way of life. And that's why it's called predictive programming. We were warned in 2015 when Ezra Klein of Vox, after speaking with Bill Gates about his algorithmic model for how a new strain of flu could spread rapidly in today's globalized world, wrote that, quote, a pandemic disease is the most predictable catastrophe in the history of the human race, if only because it has happened to the human race so many, many times before, unquote. If there was anything humanity could be certain that it needed to prepare for to prevent the deaths of a lot of people in a little time, it was this. From the RAND blog, we read, it's going to be the vaccination, stupid. From August 21st, 2020, it states that the increasingly positive news on COVID-19 vaccine development is also bringing growing alarm over whether Americans will trust these vaccines when they become available. Indeed, as the possibility for a vaccine increases, so too may rise tensions and protests. As a former head of Rand Health and as the current chair of the Rand Health Board of Advisors, we are part of a small group of Rand, UCLA, and Stanford researchers who are attempting to tackle the logistical challenges of opening schools and the economy without negative health consequences. Here is what we know and what we would propose to increase public acceptance of widespread vaccination. While we were clearly not prepared for this virus, we now need to understand how we are going to roll out any proven vaccine. Unfortunately, there's no such thing. Here are some ideas about approaching vaccine distribution strategically in this environment. It states that, firstly, a crash effort in vaccine literacy should include the federal government, including the President, Cabinet, Congress, and Supreme Court, and all federal employees, whether they are Republicans or Democrats, should publicly get the vaccine together. Social media platforms should not be allowed to function as unfiltered vectors of anti-vax falsehoods. Now keep in mind that if you're anti-vaccination, meaning against vaccination, it really just means you're pro-nature. You just want to be left alone. If you are pro-vaccine, you have a belief system, a scientific lens that you are looking through, believing that vaccines actually work, when in truth there is no scientific evidence whatsoever that shows vaccines to be safe or effective. This will be shown in depth as we proceed through this series of documentaries. Second, we need an equitable distribution plan that reflects social realities. The vaccine should go first to health workers, essential workers, teachers, and the elderly, and people whose living circumstances, from prisons to apartments, make it impossible to self-isolate if they get the disease. Now, anytime a corporation, or for that matter government, labels you as non-essential, you know there is a huge social and civil problem. We need to ensure there is no discrimination, racial or otherwise, in the distribution process. And vaccinating teachers is particularly important because we cannot expect them to risk their lives to teach their students. There's broad agreement that keeping the schools closed will increase the gap between advantaged and disadvantaged children. A generation is at stake. Most importantly, we should consider whether and how to provide at the time of vaccination an electronic readable card confirming that the person had been vaccinated. We should also consider whether it is good policy or even legal to require employees, guests, students, or others to present a readable card proving they have been vaccinated if they want to work, fly, eat at a restaurant, study in a classroom, go to the opera, take a cruise ship, etc. And now the true intention of the Rand Corporation can be seen for what it is.
Ran into this article, MasterCard joins Blockchain Digital Identity Alliance. I didn't know what the Trust Over IP Foundation was, but it includes Accenture, Evernym, IBM, R3, that's private blockchain out of Singapore. But we're all familiar with ID 2020 to provide a digital identity for everyone. Now this is somehow lumped into the COVID credentials initiative to develop immunity passport. You're going to have to pay for this monthly or quarterly. At least some prices are coming out between $20 and $90. This was first pushed by the airlines, electronic travel authorization, to make sure you wouldn't infect anybody on the plane. COVID Credentials Initiative, deploy privacy-preserving verifiable credentials. This is where it gets deep and strange. Now 60 Fortune 500 companies formed their own self-sovereign identity groups to make sure that we all get these immunity passports. At least that's a surface layer, because when we get into the universal registrar, otherwise known as a decentralized identifier, It goes deep, and it keeps rewinding to the same companies again and again, from the finance to the operating systems. And the thing standing in the way right now are these interoperable overlays, because it's all about getting you registered on this thing and somebody else to oversee you as a guardian. This is where we tumble down the rabbit hole. Individual organizations willing to take legal responsibility for managing the agent wallet on behalf of the person under guardianship, meaning you're owned by somebody in the corporate world. Self-sovereign identities, decentralized identity, overseen by sovereign, the identity for all counsel, I4A. And the guardianship means you're an owned piece of property. They're going to digitize individuals, organizations, natural things and man-made things. You want to touch any of that? You're going to have to be using V code. And to give you an idea of the money and backing behind this, these are just the players in Singapore that are bringing this to life. Australian politicians say you won't be able to partake in any type of everyday life if you don't get this COVID passport. RFID chips are not mandatory for now, but you shall not buy, sell, or trade without this mark. And then beyond the catchy headlines, which most people don't go after, if you read further into it, you're going to see this ID 2020 Alliance is basically started by Microsoft. And then it mixes in all these biometric technologies and digital identities. But then there has to be a storehouse or the technology behind it to run it all. It's not just blockchain, that's its own thing, but then there has to be an interface between that and companies holding on to this information. And that's where we get into Accenture, IBM, Oracle, and the whole gamut of tech companies. And it does seem at the end of this is going to be a global ID system, not country-centric. Now, the way to soft trickle this thing in here is your immunity passport. This is pushed by the airlines. So if you fly for business, you're definitely going to have to get it. If you're involved in the travel industry, you must get it or else you will lose your job. So it's a way for the soft drip feed disclosure into this. Immunity passport, airlines are calling for it, digital ID tracking system. So if you want to ever travel again and get on an airplane, they're going to make you go to your doctor and get a test. And before you can get on the airplane, you'll have to enter their credentials into some system weeks before or a week before you travel, and this will be on a central database, and then the airlines will have access to it. Now, I don't know how much access they have to your medical records, but they'll have an access to see a positive, negative reading, and that's where it begins. And here's where we tumble down the rabbit hole. Because 60 Fortune 500 companies right now are creating something called the Self-Sovereign Identity Group. And what they're going to do is focus all their efforts into creating these immunity passports, which are going to be required for you to get on a subway or get into a taxi or take an Uber, or if you want to book an Airbnb in the future, you're going to be required to do this. And as I've heard, somebody said that even to enter a grocery store in the future, you'll have to show this, that you're tested negative so you don't contaminate the cans on the shelves or something. So go over to the COVID-19 Credentials Initiative site, deploying privacy-preserving, verifiable credential projects. So you can't verify your identity anymore with the government. It can only be through these private companies that are going to tell you how you have to verify and how you have to pay for this every time you want to verify. And see, when I first started looking into it, it was 60 organizations, and then 100, and now it's more than 200. So a perfect example of this being deployed right now is in China, where you see the girl there. She has her phone turned toward you. It has a green mark on it, which means she's allowed to travel freely, get on a subway, bus, whatever, go into stores. If that's red, they won't let her on anything publicly. So I was wondering, how much is this going to cost? Are all these companies going to eat the cost and just do it for free because they love humanity? Of course not. Now, this is going to be forced by law in the future. The Immunity Pass card or the Immunity Pass digital itself, the physical card's $20 and the digital app that goes along with it is $80. So since most American media is talking about being tested monthly, you can look at this as having a $90 monthly test to prove that you're not infected wherever you go. You get the card with it. It's a nice looking card. But see those barcodes and the QR code on there? This is where it gets a little bizarre. Because anybody can check on you. Anybody who has access to this data system here. All they have to do is either scan either of those, and then boom, your identity is going to come up, and everything about your medical history associated with the COVID inside this thing. Plus, they're going to know your name, your address, your identity, your birth date, the height, and all these things about you biometrically. Now, it's anybody who has access to the system, and that's going to be millions and millions and millions of people. So nothing could go wrong with that, I'm sure. So how it seems to work so far here is you're going to get a color code assigned to your ImmuniPass based on the test results that you have to go to your doctor yourself and get, and then you put it into ImmuniPass, and they enter their credentials for you, whatever the scan code was off of your medical results. White is not tested, and then wherever you go, if it's white or you didn't get tested, you're not allowed in. Red, you're positive. Then you're going to have to take additional tests and additional tests and additional tests until you can prove some sort of antibodies. And then yellow will be some sort of presumed immunity. And then green will finally turn over from the red after you've had umpteen tests to prove something. And ImmuniPass is not the only one out there. And I did a previous video on this talking about just the one aspect of entering a supermarket that's going to be required beyond the mask wearing and all that. You're going to have to show this as green before they're going to let you into a store, where if you're getting stimulus money, at least in the United States, that'll be deposited into your digital wallet with the new Fed coin. You're going to be required to take this in at the point of purchase. No green, no entry. And everybody's going to have this on their phones, or else you're not going to be able to go in anywhere. Buy, sell, trade, or transact. Now, COVIDpass is another one. So there's 3 already rolled out. And these are multi-billion-dollar companies rolling these platforms out to get you tested and into the system. And what gets to the spookiest part of all of this is the ownership of you as a human on this planet by the corporations that are then going to oversee you as a guardian. Just like a parent can oversee a child as a guardian, the corporations are taking delivery of you, your resource, your body, and they're going to oversee it as a guardian agent. Now, they're going to call it digital guardianship, and it's going to be put in the cloud. They're going to call it a cloud agent or a cloud wallet service where, highlighted in blue, an individual or organization willing to take the legal responsibility for managing that cloud agent wallet / Fed coin deposited for your stimulus on behalf of the person under guardianship called the dependent. That's you and me. So when we take a look back here, it should be almost inverted, where the verifier and issuer are the corporations making their own laws to make sure that they can own you, and you're just at the bottom. No say in what goes on, really. You can say no, but I think this is going to be the division of humanity here. You say no, you go grow your own food, you provide your own electricity, medicine, water, and you get out of the system, or you stay in and you continue. But the scariest thing for me that maybe dropped my jaw here for a second was the identity for all council. Now, this sounds something straight out of whatever dystopian movie you can name through history, or even some of these off-world space movies, Star Trek and whatever it would be. The I4A. Sounds so kind and gentle, identity for all. So when you see such innocent language as MasterCard joins the ID 2020 initiative exploring decentralized digital identities, that's exactly what they're doing. To own you. And not just you, individuals. It'll be about organizations, governments, companies, natural things, man-made things. Anything ever made, anything ever grown, natural or synthetic, will be in this system digitized. And when you have to buy your food by the calorie moving forward, which is part of this plan, you will have to understand cloud wallets. You will have to understand blockchain. And you'll quickly come to the realization that without any of that, you will not be able to buy, sell, or trade anything as a human being on this planet. The tentacles are around you. And you might say, well, I will never do that. But the Australian politicians are already a step ahead of you and passing laws down there that says you will be deprived of participation in everyday life if you do not do this.
Two pieces of that. First, on the international, I think it's absolutely essential that we have international collaboration, both at the early stages so that we can understand the characterization of the disease and work collaboratively to try to mitigate the effects of the spread of the disease and hopefully to stem it in the early stages as we're moving forward. As we've said many times, disease knows no boundaries. Disease knows no boundaries. Disease knows no boundaries. So this is absolutely essential. I'm not a lawyer either, but I will move into some uncharted territories here and say that what I worry about with some of this is that much of the explosion in biotechnology today is such that it is happening well before laws and regulations and policies and best practices have been established. It is happening well before laws and regulations and policies and best practices have been established. And so I do worry a little that we wouldn't be able to do any prosecution if there was something nefarious. We wouldn't be able to do any prosecution if there was something nefarious because we have not put in place the superstructure. We have not put in place the superstructure, if you will. We're relying very heavily, and I think appropriately so, on the life sciences community. We talked about biorisk management, the biosafety, biosecurity, and bioethics. And I would emphasize the bioethics is absolutely essential third leg on that stool because we very much want to see legitimate science move forward, but on the other hand, we expect it to be done under the appropriate conditions for the appropriate purposes.
I thought those were both very important questions. I think there is one example that doesn't perfectly fit, obviously, but the case of smallpox, where the international community came together and decided on the rules for further work. It's only done in 2 places in the world with the approval of the World Health Assembly, so there's international support for that. I do think that going forward, there should be international agreement about how to proceed. I think Ilaria said in a public statement earlier, made a very wise comment, which was, we don't want to wake up, I don't mean paraphrase, but wake up sometime in the future and find out there are 200 labs doing, you can correct my quote later, but that was the point that I took from your quote. I think it's not only a legal question, it's an ethical question. That's the reason why I said before that I feel like for this community to proceed, this isn't a panel about risks, it's mostly about benefits. But if this community is to proceed with this research, I think we need to have a common understanding about the high side risks. And if we do, it would give me a little bit more sense, a little bit more confidence that we're proceeding in the same way. But I don't feel like we have convergence on risk. We have a lot of discussion about benefit. Maybe we'll find out later today that we all agree on the risks, but I don't think that's the case.
Okay, go on to the next question here.
Alikoff with Science Magazine. My question is primarily for the non-U.S. panelists. If the United States moves forward unilaterally with adopting the framework which has been proposed, do you think it will be an act of leadership that will bring the rest of the world along to adopt similar policies, similar funding policies, or do you think it would fracture the flu researching community into 2 factions, one which abides by the U.S. funding rules and another which does not?
Dr. Aditama.
Okay. After the last 2 research being done by Ron and Yoshi, I have to say that there is no really public health impact being done in the country. There is no different policy after those 2 issues. But for the third aspect of the politician, it's already mentioned by Malik, there is a big issue whether this is really, because like Malik already mentioned that for so many years we are talking about S5N1, but so-called there's no real pandemic happening. So these 2 research kind of, make people think. So this is really a danger coming ahead. That's the third reaction from those 2 research. The first one is for the lay people. When those 2 research are in the newspaper, people some people are kind of frightening. What is really happening here? But I don't think information is quite well taken by people in general at that time. Regarding the issue of other countries outside U.S. Of funding, I don't think funding issue is one of the important issue for country like Indonesia for this kind of research. I already mentioned that for us, epidemiological research and virus characteristic is more important to be able to control the public health aspect real in the field, not more on the gain of function as it is in the field.
Peter Hale, I'm with the Foundation for Vaccine Research in Washington. We feel that the issue of liability, that insufficient attention has been given to the issue of liability. The organizers of the Asilomar conference 35 years ago made sure that there were lawyers participating in that meeting, which led, as we all know, to guidelines for recombinant technology. There is another community that I think should be involved, that we think should be involved, and that is the insurance companies. The insurers don't like what they're seeing, and they don't like where this is going. The insurers don't like what they're seeing, and they don't like where this is going. And in particular, the largest insurance companies that reinsure other insurance companies, they're experts on risk assessment, and they don't like what they see. And they don't like what they see. They're experts on risk assessment, and they don't like what they see. We understand there have been secret meetings in London at Lloyd's of the reinsurers. How are we going to reassure, even with the most stringent biosafety rules and procedures, how on earth are we going to how are we going to reassure the insurance companies? I mean, we're seeing premiums going up already. How are we going to reassure them that this is safe?
Anyone like to come? Ron?
I would like to point out that this issue that you're raising and has been raised earlier is not related to H5N1 gain-of-function research. This is an issue that is much, much bigger. There are hundreds of labs in the world that possess SARS. There are hundreds of labs in the world that possess SARS. There are hundreds of labs in the world that possess SARS. There are hundreds of labs in the world that possess other dangerous viruses. There are hundreds of labs in the world that possess other dangerous viruses. This issue is not related to H5N1 gain-of-function research at all. And we've seen many discussions come up from the public and from the press asking questions about this research that's really not only related to H5N1. And so these are really, really good questions, and they should be handled at some point, but I do not think this is the right time because it's not specific for H5N1 gain-of-function research.
All right. Nancy?
Yes. I would like to make a comment and then end with a question. The comment I would like to make is that I know it's very difficult for people in the audience and people in the general public to understand the importance of this type of work to public health, but I'd like to explain just a little bit about it. In public health, we're really responsible for helping be as prepared as we can for the next pandemic. H5N1 viruses have worried us tremendously. And we've worked with our partners in the affected countries. I see my colleagues from Indonesia and Vietnam here today. And we've worked with FAO, OIE, WHO, and many other partners outside the US government, as well as with BARDA, NIH, and FDA within the US government family. We are very interested to know, we were very interested to know if these viruses could become transmissible. We were very interested to know if these viruses could become transmissible, and if so, how their characteristics would change, so that we could look at the vaccine viruses that we're always making, so that we could look at the vaccine viruses that we're always making at St. Jude's, at CDC, and other places around the world, to see if those vaccine viruses could be improved, Now, we've had to put our work on hold with respect to viruses with the mutations that have so far been identified. So we haven't been able to develop vaccine candidates against these two particular transmissible forms. Neither have we really been able to do complete antigenic analysis using our panels of ferret sera, et cetera. So there's a tremendous amount of work that's extremely valuable that could take us forward in our preparedness that has simply not been able to take place.
Influenza is unpredictable.
One thing you could predict about influenza is that it's unpredictable.
Influenza virus is highly unpredictable.
Our goal since the beginning of the outbreak is to be one step ahead of the unpredictable virus.
So there's a tremendous amount of work that's extremely valuable that could take us forward in our preparedness that has simply not been able to take place. So I'll leave it there, but I would like to ask the panel members if they think that the escape of SARS in 2003, in 2003, and the escape of H1N1, from a lab, if that's where it occurred in 1977, are really good comparators to escape of H5N1, which is in the United States a select agent pathogen and worked on in very specific conditions. Thank you.
So the shocking admission and open panel question is, are the known escapes of gain-of-function lab-grown man-made SARS-1 and the gain-of-function 1973 H1N1 strain of lab-grown man-made bird flu a comparable example to base the same possible dual-use lab escape of man-made, highly pathogenic H1N1 on in the future? Now, bear in mind that though this is a quite surprising and unexpected confessional for this public forum, these insiders on the panel cannot answer this question without either denying or conceding silently that these are true statements, that the SARS-1 and H1N1 so-called pandemics were actually gain-of-function lab-grown viruses released into the public. Oh, and by the way, if SARS-1 was man-made through this type of gain-of-function research, then by default, SARS-2, its brother or sister, the SARS-2 coronavirus in 2019, or COVID-19 as we call it, as a derivative of that previous strain, is also necessarily, or by default, a man-made lab-grown virus. The same virus as SARS-1 with added gain-of-function properties, and so named as SARS-2. Unfortunately, our lone antagonistic anti-hero speaks again in this overwhelmingly scientistic forum without denial.
Would you like to take that?
Sure. I think your question was whether or not the risk is the same now. I'm trying to understand when you said they're good comparators. I think that the reason they're good comparators to me is to provide anecdotes in a discussion otherwise that has not raised risk at all, almost, of the possibility of accidents even from high biocontainment labs, at least in the case of SARS. I think the work that's gone on to now with gain-of-function work, the work that has been published, has been done in really very high-level labs with good records. I haven't heard any discussion yet about the rules of the road going forward, whether there would be some set of criteria to limit those labs going forward, whether it should be done in all places that now do vaccine candidate development around the world, what those rules would be. So it's hard to make a direct comparison worldwide, but I do think it's important because of the fallibility of human systems, the fallibility of human systems, the fallibility of human systems. We have systems that are designed as best we can design them, but I don't think any of them are foolproof. We have systems that are designed as best we can design them, but I don't think any of them are foolproof. But I don't think any of them are foolproof. But I don't think any of them are foolproof.
Thank you.
We would like to start panel 2. This panel focuses risk and concerns associated with influenza genome function research. First, the panelists will identify and discuss the range of concerns that have been raised over influenza genome function research, including biosafety risks, biosecurity risks, dual use and informational risks, and the implications for national and global security in conducting the research. We have 3 important discussion points. First one is, what are the specific risks and concerns associated with influenza genome function research? And second point is, which components of influenza genome function research experiment raise particular concern? And the last one is, are there experiments that should not be done? And if so, why not?
There is a very diverse situation around the world with reference to potential of carrying out these experiments and potential of these PPPs, potentially pandemic pathogens, to be released. And I personally think that the issue of the concerns, which are mainly related to accidental release, are not a major concern in certain parts of the world, namely in this country. I think that in this country you have all the systems in place to avoid that accidental releases can occur, and also in many European countries. I don't think that a similar situation occurs in other parts of the world. And I also think that this problem needs to be addressed, not with reference to what is going to happen tomorrow morning or within the next year, but with reference of what can happen in the next 20 years or so. Also, with technology which is becoming more and more widespread. Many people in this room have taught colleagues from developing countries to use reverse genetics. And reverse genetics, which maybe 10 or 15 years ago was something only used by a handful of laboratories, is now widespread. And reverse genetics is now widespread. And reverse genetics is now widespread. Many people in this room have taught colleagues from developing countries to use reverse genetics. And reverse genetics is now widespread. And reverse genetics is used virtually everywhere. Now, if we forget about bioterrorism, which is not my field of expertise, what I can tell you is that some countries have social unrest and have other problems which are related to, well, many factors, including civil issues, which can have an effect on the accidental release of these pathogens from the laboratory, which can have an effect on the accidental release of these pathogens from the laboratory. And I would like to just mention what has happened in Egypt with the Arab Spring and with the revolution. The veterinary laboratory was looted. The veterinary laboratory was looted. And these things happen, and so my question is, in a nutshell, if we generate this data and this data is being put in the public domain, and I am a supporter of the public domain, just so that, I mean, we get things right, and Exponentially, more and more labs will be able to use this information to do their own experiments. Exponentially, more and more labs will be able to use this information to do their own experiments, and these viruses will be kept in laboratories which are not, which do not have the biocontainment measures, These viruses will be kept in laboratories which do not have the biocontainment measures, but because they live in because they are placed in a country which is a country in which biocontainment is difficult, I think that the risk of accidental release may occur. I think that the risk of accidental release may occur. Accidental release may occur. And going back to Tony Fauci's question, which I think we really need to pinpoint and concentrate on, is, does NIH want to support the funding of experiments which can have repercussions on public health, such as the accidental release? I think that if we continue to disclose this information, this cannot be ruled out. Thank you.
Dr. Woolley, please.
Thank you very much. I was asked to speak on the biosafety risks and the OC health. The biosafety risks for gain-of-function research with highly pathogenic avian influenza are enormous, considering that loss of containment for a variant with increased virulence, increased transmissibility, or both, has the potential to be the most devastating pandemic ever seen to humankind. Has the potential to be the most devastating pandemic ever seen to humankind. The most devastating pandemic ever seen to humankind. Currently, biosafety level 3 enhanced is the recommended level of containment for high-path avian influenza H5N1 for the reconstructed 1918 strain, for the reconstructed 1918 strain, for the reconstructed 1918 strain, and for the H2N2 pandemic strain circulating between 1957 and 68.
Yes, as I showed in Lethal Injection, the story of vaccination, they dug up the 1918 Spanish flu virus that killed between 50 and 100 million people from the permafrost in Alaska, from the frozen remains of the lungs of an Eskimo or Inuit woman, and then re-engineered it in a lab. They then filed the paperwork to patent it. Then, coincidentally, I'm sure, the patent was granted just a few months before the 2009 H1N1 strain, the re-engineered so-called Spanish flu, was upon us yet again.
Risk group 3 agents, such as these have been determined to be, are those associated with serious or lethal human disease for which preventative or therapeutic interventions may be available, representing a high individual risk but low community risk. The rationale for allowing work with these viruses to be conducted at BL3 enhanced was based in part on pre-existing immunity within the population, either residual from previous pandemics or cross-protective from contemporary strains, sensitivity of the strains to current antiviral medications, and low transmissibility, in particular for the avian H5N1. Risk group 4 agents are those that are likely to cause serious or lethal human disease, for which preventative or therapeutic interventions are not usually available, thus representing a high individual risk and a high community risk, Variants created during gain-of-function experiments have the potential to display novel antigenic properties, thus no pre-existing immunity in the community at large, and they have the potential to be resistant to all current antiviral medications, thus no pre-existing immunity in the community at large, and they have the potential to be resistant to all current antiviral medications. Logically, then, such novel variants fall into the risk group 4 definition. In conducting this type of research, we must plan for the worst-case scenario. a novel antigenic variant that is drug resistant. And even if such a novel variant were sensitive to current medications, there are serious limitations on the use of these medications, and the supply would very likely not keep up with the demand. We must also assume that we cannot quickly produce a protective vaccine against such a novel variant. We cannot quickly produce a protective vaccine against such a novel variant. We need only turn our attention to the recent 2009 H1N1 pandemic to see the consequences of this. By the time the vaccine for this strain became widely available in December 2009, the peak of this pandemic had already passed. Fortunately, the case fatality rate for this strain was relatively low.
Thank you.
Dr. Duval, please.
Thank you. So I think it's important to keep in mind that gain of function is a research tool. In fact, it is a human tool for probing nature. And it is a very, very powerful tool. And there was we knew a lot about influenza before such experiments could be done. And you can probably learn a lot without doing gain of function studies. So I'm very reluctant to take such a powerful tool off the table on principle alone. Should gain of function experiments be done? I think so, yes, provided the two questions are answered. Is there information from gain of function experiments that is critical for pandemic preparedness that cannot be obtained by other means? If the answer is yes, this rationale has not been clearly articulated. In my opinion, basic curiosity that drives most science is not enough. If the work is supposed to inform surveillance, then surveillance needs to be done right. If the work is supposed to inform vaccine development, I would like to see the rationale how this will lead to be used in that case. And to my knowledge, this has not been clearly articulated. The second question is, are there alternatives to gain of function that provide comparable information? And here the answer must be no, for the gain of function experiments to be done. But before you say that the gain of function is critical for providing certain information, I point to you to cosmology. We are in the midst of a revolution in understanding the cosmos. It is all done from description and observation alone. And it is hard for me to believe that there aren't alternatives for getting this information when astronomers cannot knock out a galaxy or give a gain of function to a black hole and see what happens around it. And yet they can explain most of what has happened in the universe and where the universe is going. One solution to this controversy, which has already been mentioned, is to make safer viruses, For example, in Ebola, the Ebola example, a lot of work has been now done at BSL-2, including the identification of the receptor, The Ebola example, a lot of work has been now done at BSL-2, including the identification of the receptor. In summary, I believe that gain of function should be done provided that a compelling rationale is developed and that there is no alternative ways to obtain this information. Which components of gain of function raise particular concern? I think that all of those that fit within the NSABB definition of DIRC, increased transmissibility, increased pathogenicity, altering host range, and certainly introducing acquired resistance. So I've been asked to answer the question, are there experiments that should not be done? Well, the ones that I can think of should not be done are combinations of attributes. In other words, to make a virus, for example, not only mammalian transmissible, mammalian pathogenic, and drug resistance at the same time. I will not combine the attributes. I would do gain of function. I will tell you that I think that the community should be focusing on making safer tools. I think that it should be possible to separate transmissibility from pathogenicity. In fact, we already have that. We already have a virus that is pathogenic and not very transmissible. So it should be possible to have vectors that are transmissible but not pathogenic, and so great care should be taken before combining any of these attributes into a single virus.
What of the undermining of international public health agreement? Actually, this is a quite interesting point I've seen on this list of questions, and this point was already raised, at least in Canada, after the creation of recreation of 1918 collaboration with Yoshi, the 1918 influenza virus, recreation of the 1918 influenza virus, because one angle was that it was breaking the Geneva Convention. Again, I think for all these points, the appropriate framework must be in place to evaluate gain-of-function research. Which component of H5N1 gain-of-function research raised particular concerns? And here there was transmissibility, pathogenicity, altering the dose range combination, and I think they all raise concern to some extent. Again, it is a matter of debate and opinion and speculation as to which one is perceived more dangerously and more risky than others, but you're back to what are the benefits, in my view, attached to doing the work or not doing it. And this is often what is not put on the table very clearly. And I know everybody's alluding to it, but it's a point that's very hard to make. It is easy to see the risk of an accidental release, for example, of highly transmissible H5N1. It is easy to see the risk of an accidental release, for example, of highly transmissible H5N1. But it's not easy to convince and to explain the risk associated with not doing the work. Their experiment, and I'm going to close this quickly, that should not be done. An experiment that are trying to reproduce a known or anticipated or plausible environment that can be encountered in nature should be considered. Experiment that could be seen as unwarranted, again, considering the risk-benefit ratio, are experiment not reproducing conditions found or plausibly found in nature, where artificial conditions are created, for example, to force the system and force the generation of a new pathogen, where artificial conditions are created, for example, to force the system and force the generation of a new pathogen, Hybrid viruses could be part of this, where a variant component or defense component of an infectious agent is introduced to create, with an intent, pathogenicity could be an example. But even here, the question remains, what is the benefit to public health and the ratio with risk?
Dr. Cologlazia, please.
Thank you. Well, Dr. Fouchier and Dr. Mahmoud both mentioned the Think report, which was this 2003 U.S. National Academy report, Biotechnology Research in the Age of Terrorism. In the intervening decade, there have been many technical advances, and the pace of change of research is likely to increase perhaps even faster in the future. And as our capabilities have advanced, many scientists and security professionals have worked, I think, thoughtfully to try to develop the tools to minimize the very real risk of intentional misuse of biotechnology, including of the highly pathogenic avian influenza in particular. But these two communities obviously have a shared interest in a safe, secure, and responsibly conducted life science research enterprise. Actually, there have been many conversations about H5N1 among these communities, which actually reside inside the State Department. And I certainly don't need to tell this group of the increasingly global nature of the research enterprise. Good research can emerge in many countries in both the public and the private sectors, and good research ideas that go unfunded by the U.S. government can be redirected elsewhere. So thoughtful oversight of U.S. government-funded research is obviously important, as this conference is focusing on, but it's also important to consider the risk that, absent synergistic international oversight, that research might still be performed elsewhere, but without due attention to the mechanisms being constructed to balance the risks and the benefits. Research might still be performed elsewhere, but without due attention to the mechanisms being constructed to balance the risks and the benefits. And elsewhere does not just mean in the territory of some shadowy rogue regime or an extremist group. We must also think about the tools that we need to engage the global private sector, and in some cases even the growing community of do-it-yourself biologists that are operating in their backyards. The growing community of do-it-yourself biologists that are operating in their backyards. At the 7th Biological Weapons Convention Review Conference, Secretary Clinton underscored the U.S. government's commitment to building an environment of openness and collaboration in our biodefense enterprise. And she outlined a series of unilateral voluntary planned activities called the Biotransparency and Openness Initiative. The dual-use nature of biological weapons agents, production equipment, and advances in biotechnology can make it hard to distinguish between offensive and defensive work. The dual-use nature of biological weapons agents, production equipment, and advances in biotechnology can make it hard to distinguish between offensive and defensive work. And means we must find tools to increase openness while encouraging compliance among the state's parties.
I hope to bring the perspective not only as someone from the Department of Homeland Security, but also from someone who has been thinking about issues of the governance of dual-use biotechnology for a decade and has been working at the interface of science, technology, and security for almost 3. I'd like to leave you with 2 main points today. First, that extremely consequential research warrants a greater degree of scrutiny, as with the framework we're discussing today. And I'd also like to spend a little time talking about the breadth of misuse scenarios, which I think the term bioterrorism does not capture and indeed may be misleading. The breadth of misuse scenarios, which I think the term bioterrorism does not capture and indeed may be misleading. In terms of the consequential research, it's clear that the stakes are much higher. It's clear that the stakes are much higher with the research we're discussing today than with almost any other form of scientific research, where the great risk is wasting money or possibly not spending money on something which would have higher scientific return. Here, we are evaluating the risk of generating, whether accidentally or deliberately, a global pandemic, potentially putting millions of lives at risk, potentially putting millions of lives at risk, and weighing that against the risk that failure to do research would leave us less prepared for such a pandemic that might break out naturally. So the stakes are clearly high here. I think the very existence of this policy, our discussion today, recognizes that there is a line beyond which we should not go, that there are scientifically meritorious proposals that for other reasons would be judged not to be worth their cost. If that were not the case, we'd all be wasting our time. If there were no possibility that an evaluation of the form we're discussing could lead to a decision to either forego research or do it under more restrictive conditions, then there would be no point in having this exercise at all. I think this is an important exercise to have. Let me turn a bit to misuse scenarios. I'll leave biosafety risks to others, and again, other panels have talked about the benefits. But in terms of misuse, I think we have to be extremely humble in assuming that we can know or understand the motives and the capabilities of those who might want to create harm on a massive scale. The concerns include those that go beyond, and in fact I think have little to do with, what might be called bioterrorism. If by terrorism we mean acts to inflict harm against non-combatants or civilians for a political objective. By terrorism we mean acts to inflict harm against non-combatants or civilians for a political objective. In this debate, we've often heard that pandemic flu would be not a useful terrorist weapon, that it would not serve terrorist objectives to wreak that kind of indiscriminate harm. Whether or not that is the case, we have to consider the possibility of misuse by individuals or groups whose goals we would not predict and whose motivations we may not even be able to comprehend. We don't have to look very far in the past to find acts of violence inflicted by people whose motivations seem beyond comprehension. And it's that greater possibility, not so much the idea that a terrorist group may seek to use these capabilities, that one has to keep in mind.
The growing community of do-it-yourself biologists that are operating in their backyards.
We cannot exclude the possibility of psychopathology, individuals with a very warped set of objectives and desired means for pursuing them. I think a point that others have made, Dr. Inglesby and on this panel, Dr. Capua, have pointed out that we also have to be very leery of limiting our consideration of potential misuse to those who have the capability to do so now. Once information is put in the public record, it's there forever. And as has been pointed out well by my predecessor on this panel, technology continually evolves, barriers continually lower, interest and capability in biotechnology penetrate new geographic areas and permeate the economy in ways that will continually make any given level of capability easier to attain over time. Now, can I be specific about the people, the places, and the probabilities of misuse that I'm so concerned about? No, I can't. But it would be dangerous to believe me if I said I could. We may be worried about the actions of people who have not yet formed an intent to act in a certain way, or about groups that have not yet been formed. But we do have to recognize that when this type of research is done and creates the possibility of misapplication, when this type of research is done and creates the possibility of misapplication, that possibility will persist. So we have to be very careful not to inappropriately define a narrow sense of the biosecurity or misuse concerns, and we have to be very careful not to overly constrain the time frame in which we're trying to consider our views. Thank you very much.
Thank you.
There are two key framing perspectives that are important to keep in mind in this regard. First, there is reason to worry about the misuse of biology by bad people. There is reason to worry about the misuse of biology by bad people. Some countries do retain offensive biological weapons programs. Some countries do retain offensive biological weapons programs. There are still some terrorist groups interested in acquiring such capabilities, and occasionally we find individuals who consider using biology to inflict harm. It should be noted, however, that interest in biological weapons is relatively uncommon, and actual use of them is even rarer. Moreover, at this point, most bioterrorist activity is crude, not likely to benefit from advanced biological research. Historically, it's perhaps worth pointing out that the total number of people who've been killed by all known uses of biological weapons is a fraction of the number of people who die every year in the United States from pandemic or from just seasonal influenza. Having said that, the past may not be a good indicator of the future, and I would associate myself with the comments that that Dr. Epstein made about the uncertainties of predicting who might misuse this kind of science. Now, a second framing perspective is that because of these concerns, the international community in general, and the United States in particular, developed a web of treaties, other international agreements, laws, regulations, and policies intended to prevent such misuse of biology. Now, while I'm not a lawyer, it is my understanding that the intent of these efforts is in no way designed to limit research. The focus of those efforts is on preventing the acquisition and use of biological weapons. It's not intended to limit research in developing defenses against biological weapons, and certainly not to limit research in the public health arena. So what does this mean for the issue we're discussing today? The fact of the matter is that if the H5N1 research that we're discussing today that NIH sponsored had been sponsored by the department that I work for, the Department of Defense, for exactly the same scientific purposes, it's likely the United States would have been falsely accused of violating its treaty obligations, and the State Department would be busy defending ourselves against charges of U.S. continuing to maintain an illegal offensive biological weapons program. The State Department would be busy defending ourselves against charges of U.S. continuing to maintain an illegal offensive biological weapons program. Now, I find that most of my colleagues in the national security community understand the importance of disease and infectious diseases and recognize that the benefits of such research, even though there are people intent on misusing science. The benefits of such research, even though there are people intent on misusing science. Even though there are people intent on misusing science. In some cases, this recognition is very specific to our communities. Thus, many of my colleagues in the Defense Department worry about the health of our armed forces, which depends on exactly the same public health medical countermeasures developed for the general population. And those involved in biodefense are interested in enhancing our understanding of scientific processes.
Biosafety issue has occurred in history due to the increase of incidence of laboratory-acquired infections. Afterwards, biosecurity issue rise when dual usage of disease agents, either for good or other risky intentions started. In the last few decades, the world has seen the emerging of several new infectious diseases. Most of them are potential epidemic forms, such as SARS and avian influenza. Many factors may induce the occurrence of these diseases, both man-made as well as natural factors. Many factors may induce the occurrence of these diseases, both man-made as well as natural factors. Both man-made as well as natural factors. Ladies and gentlemen, in conclusion, research in the laboratory which work with dangerous pathogens, no matter what it is, will increase transmissibility or pathogenicity or even alter host range of HPAI, H5N1 virus. Will increase transmissibility or pathogenicity, will increase transmissibility or pathogenicity or even alter host range of HPAI, H5N1 virus.
I ask you, is this an issue for the scientific community in isolation? Or should we be speaking to our communities and nations in the vocabulary pertaining to doing this work? If the public doesn't see the value and want this done, should the work be done? Why did we put salmonella into a microgravity environment? Was the outcome part of the hypothesis? Why do we do genetic knockouts for cancer research? Is there an analogy to that model? The questions posed as written in question 2 all raise concern within multiple communities. However, it's important to understand that we, as human beings, tend to form our initial opinions on the basis of intuition and emotion. This, of course, is directly related to our experience and knowledge. We then, when given the opportunity, allow reasoning to become a greater component of that thought. The initial intuition and emotional response may become tempered. Taking these questions and rephrasing them puts a different light on the subject. Suppose we were to ask, what causes transmittability? What causes pathogenicity to increase or decrease? How does the virus change its host range? The context of the question could be everything as we talk to the community and our civilian communities. Now, we see the original questions differently, and our ability to move away from emotion and intuition is attenuated. We now can engage in reason because we have reframed our thought processes and patterns. In doing so, we have moderated our potentially exhilarated deliberation. So we end up saying yes to the question. All three questions, singularly and combined, raise concern. They should. Intent is everything. I am sure we are going to discuss this along with controlling policies. Age and experience become harsh mentors of our ability to successfully change a potential bad or unpredicted outcome. Viruses, like other living organisms, strive for balance. Many researchers have told me, as they tell you, manipulation in one area has consequences to the organism. Manipulation in one area has consequences to the organism. Increase its transmittability, may decrease its virulence. You know all the permutations, I need not go on. All science has a risk of being used inappropriately. All science has a risk of being used inappropriately. Do we suppress and cloister science, or do we find better ways to detect those who may have psychological, moral, or ethical aberrations that are at variance with all of humanity? Thank you very much.
Thank you.
In case these seemingly wise words sounded reasonable to you, what you just heard was a master propagandist speak to the proper neurolinguistic mind control techniques that should be implemented when this gain-of-function dual-use research is discussed with the actual public, that is, you and I. Your intuition is your natural response, a design found in all species throughout the animal and insect kingdoms. But through word magic, these propagandists of government and corporations are able to dull or deaden your automatic, natural, spiritual response toward these crimes against nature, a semantic game intended to cause you to love and support what you should instinctively hate and reject.
First, are there experiments that should not be done in the context of HPI H5N1, in particular in gain of function? So let me say my default position on this is the same as Thomas Jefferson. When he founded the University of Virginia, he said, "This institution will be based on the illimitable freedom of the human mind. Here we are not afraid to follow the truth wherever it may lead, nor are we afraid to tolerate any error so long as reason is left to combat it." So that's my default position. There are no experiments that shouldn't be done.
No experiment against nature's design, against all life itself, should not be done? Really? Jefferson must surely have turned in his grave when this was stated, for there is no true freedom without absolute moral, spiritually driven self-responsibility. For your consideration, not once was the Hippocratic Oath, the concept of do no harm, mentioned in this panel discussion. If we are to use the unalienable pursuit of truth as an excuse to recreate nature's design, that is, alter the truth to our image, should we not first have an understanding and agreement of what is truth and what is not truth? To a mad, that is, psychopathic or sociopathic scientist, what is truth? When money and profit is involved, what is truth? Can what man recreates or mutates in a lab really be called the truth or source of nature? If truth, that is, nature itself, is only what is self-evident and self-existent, existing despite man's wills, designs, or interventions, then how can a gain-of-function research be justified as the pursuit of truth? Nature, untouched by man, is the only truth. Indeed, even the Bible declares God is truth, and we should be worshiping and protecting that truth. We can only survive, we can only breathe in the truth, in nature, according to its laws. Science is defined as the study of nature and its law as it already exists, not the recreation of it. Gain-of-function research is, in fact, anti-science. Gain-of-function research is anti-truth. Now, finally, we come to the truth, to the story and intent behind the story. What is the reason, the intention, the ulterior motives behind these mad scientists in their unwavering support of such unscientific, unnatural dual-use research? Why is it even a question that such deadly research, which cannot possibly happen in or be reproduced in nature, should be disallowed, that is, banned as a rule? Now the plot thickens.
However, in this context, reason is the key thing, and we've heard a number of reasonable explanations and reasonable objections to doing some experiments. In terms of gain of function, I think as the life sciences converge with biology, but with physics, mathematics, and engineering, gain of function is absolutely essential. The cyber-physical program at the National Science Foundation, actually is premised upon that fact, that if we can actually engineer something in and predict its properties, that is gain of function. If we can actually engineer something in and predict its properties, that is gain of function. If the life science research and this particular field is to go forward, with all the reason behind it, we have to do gain-of-function experiments. We have to put them in the context of mathematics, physics, and engineering, or else we will make no progress. We've been asked to survey the landscape of international agreements, and how does it affect infectious diseases. Well, the capabilities are quite different, but the rules and regulations and the advisory committees are legion. So we have the World Trade Organization, we have the Biological Weapon Convention, we have NSABB as an advisory board, export controls, Australia Group, international health regulations, the trade-related aspects of intellectual property, TRIPS, and now we have the United States International Strategy for Cybersecurity, which clearly deals with how we're going to exchange information over the web. All of these international agreements directly affect the kinds of things we're talking about here today, and clearly affect individual scientists. You don't have to go very far into the audience to realize and recognize individuals whose research have actually been stopped according to certain of these kinds of rules, regulations, and agreements. You don't have to go very far into the audience to realize and recognize individuals whose research have actually been stopped, according to certain of these kinds of rules, regulations, and agreements. And in fact, what I'm concerned about is that we will put in place regulations and rules and advisory committees that will hinder the kinds of research that absolutely have to be done, as Arturo clearly said. So I think that what we really have to do is recognize that science is an international endeavor. There was a National Science Foundation study published just last August showing that in the life sciences, 23.9% of those responding actually have international collaborators. We have to be concerned that there are places in the world that can't meet the set of criteria that we will set up. So this differentiated capabilities has to be something we have to take very, very seriously.
I think it would be most useful for this panel to really reflect on question number 3, if we could, just a bit more, because I think this is kind of at the heart of what it is that we're talking about. And question number 3 is, are there experiments that should not be done? And if so, why not?
I believe that if you cannot speak to why you want to do a specific piece of research, if you have not really looked at the why, the intent, and the possible consequences, if you haven't met those criteria of knowing why you need to do the work, it shouldn't be done. So I guess I sum it up by saying that intent is everything, and if you have positive intent and can't explain the why, that particular individual or that particular organization should not be doing that work.
Thank you. Other comments?
So as a former member of NSABB, I'm not on the committee anymore, so I can speak more freely, I guess. Don't forget, dual use of concern doesn't mean don't do the experiment. When the decision to publish the Spanish flu papers came out, everybody, Kurzweil and Joy in the New York Times wrote an op-ed piece, said this is the end of the world. So nothing bad has happened since then, as far as I can tell, from the Spanish flu set of experiments. And it was all predicted this, all hell would break loose. So nothing bad has happened since then, as far as I can tell, from the Spanish flu set of experiments, So I think the key thing here is reason. And I don't know how many of you have written NIH grants, but if you don't write the significance, and if you don't write very clearly why you want to do these experiments and what would happen if they go wrong and so on, and are the facilities up to date, and are the human subjects protected, are the animal subjects protected, there's a whole bunch of stuff you have to do when you write these grants. So that goes without saying. It's the kind of folks that Jerry has reminded us that I think are the real problems. And we can't predict who's going to be out there. Who's going to be out there, and should we tailor our set of experiments for the worst of all psychopaths in the community? Should we tailor our set of experiments for the worst of all psychopaths in the community? I'm not sure I'm willing to go there just yet.
In terms of what shouldn't be done, I would look at the actual phrasing of the NIH proposal that we're evaluating today. I don't think that's on the screen now, but for example, one type of experiment I think would be hard to defend would be creating strains that would be extremely unlikely to arise if it were not for the fact that we were creating it. If it were not for the fact that we were creating it. Creating strains that would be extremely unlikely to arise if it were not for the fact that we were creating it. And in that case, we would be accepting the risks of such a work, but we would not be attaining any benefit because we would be anticipating a problem that was extremely unlikely to happen. I realize a lot is tied up in extremely unlikely, and that's what the discussion would key around. I think a second category, looking at another of the bullets in the proposed review framework, would be experiments producing results that for whatever reason cannot be usefully applied. Experiments producing results that for whatever reason cannot be usefully applied in mitigating the concern that is motivating that work. So again, all I'm doing is restating objectives that are in the review framework, saying these are the questions one should ask, and if one can't answer them, then that's a clue that maybe the work that's being proposed doesn't meet that bar.
I think that whatever gets done, there ought to be sunset provisions. For example, whatever we decide, it should be at this time, because things move very rapidly. You know, I drew the line in making a virus that is highly pathogenic, transmissible, and drug resistant. I mean, I think at this time, that probably is very hard to defend. On the other hand, I could imagine that things could move. And that I could imagine that you would need to know that information because it turns out that the three attributes together influence yet another aspect. So one of the things that worries me, and I go back to this, is that all these regulations, and as you begin to put in all these red flags, people, the smart people, the people basically which are limited are not going to be doing this work. The smart people, which are limited, the smart people, which are limited, the smart people, the people basically which are limited are not going to be doing this work. Why would you want to be a graduate student or a postdoc doing a lot of this work if in fact society makes it so difficult? On the other hand, we need to not only protect society but also make sure that this community remains vibrant. And one of the problems in microbiology is that, and you can see this across fields, when fields don't have tools or where fields cannot do certain experiments, they tend to fall behind.
I think that's right. If you start limiting the human ability to question and push the boundaries, that's going to kill the field. And in particular, the issue that we were discussing, that Jerry raised about unlikely to arise, there's a whole area now of biological research looking at stochastic events. And it turns out, and I think you've even, it turns out that the unlikely events may in fact be very, very important. In fact, they are very important. So this notion of being able to predict ahead of time that this may or may not be important because it's unlikely probably won't fit in with the current way we think about important evolutionary concepts. That this may or may not be important because it's unlikely probably won't fit in with the current way we think about important evolutionary concepts. Important evolutionary concepts. Important evolutionary concepts. So stochastic events, unlikely events, may in turn out to be very, very important. Unlikely events may in turn out to be very, very important. And whether that's a combination of other unlikely events or just a single unlikely event, we don't know just yet. But we have to be able to look at it. And I think in the context, we can think of very safe ways to ask those questions, and those are the kinds of things we do on the benchtop. And we can think of very unsafe ways to ask those questions.
Here we find the prime universal example of what is called dual-use research. It's not so much a secret as it is a normalized part of the medical, university, private, government, and military systems, which are funded and work together in mutual benefit through open research findings and shared patenting. The correct term is syndicalism. What is found in a medical study, in other words, might be funded and used by the Department of Defense, while also contributing to advances in technology or other artful anti-sciences. This indirectly beneficial dualistic use and sharing is not, of course, accidental, nor in the least unexpected. If we reconsider the reasons behind so much bad science, behind so many failed and retracted studies, and behind so many irreproducible experiments, we just need to consider that these are abuses of the funding being provided, and in my own research, I've found an extraordinary amount of what is referred to as synthetic biology, that is, the creation of synthetic life and the creation of synthetic environments both inside and outside the human body. And this allows us to understand why science has gone astray. Its intentions are upon transhumanism, that is, futurism, which is the ultimate fulfillment of the dogma of scientism. And so while government laboratories claim that none of their gain-of-function research is classified, this certainly does not mean that the results of those studies aren't being continuously used within classified projects and corporations such as DARPA, Google, etc. But most shockingly, after researching this subject for over 15 years, the most insane sociopathic dual use for this biological gain-of-function capability has to do with what is collectively known as transhumanism. Transhumanism, the unnatural combining of man and technology, cybernetics, and artificial intelligence, is the anti-science of artificial evolution, or unfolding of the biology of man so as to induce such unnatural traits as immortality and human-computer interface. For human biology to thrive in synthetic environments and systems, this gain-of-function research must take place, because transhumanism ultimately entails the intentional mutation of the cell biology of man with the goal of infusing the real with the synthetic. In other words, transhumanism is the merging of truth with lies, of what is artificial with what is natural, or, for the religious-minded, the fusion of Satan with God.
Every aspect of our lives has been reshaped by technology, from the way we get around, the way we seek information, and the way we communicate. It's easy to think that if only our technology advances enough, we'll finally be satisfied. The fact is, we remain shackled by our primitive Darwinian brains. Humanity, for whatever progress we have made, is the result of an unguided, natural, 3.8 billion year long experiment of chemistry. Evolution is the process that has made you what you are, but it is not far-seeing. It does not and cannot consider the future, make decisions about where we ought to go, how we ought to be. Passing on genes is the only objective. But as thinking human beings, we care about far more than that. Consciousness means that we have the capacity to experience the world, to reflect upon, and, most importantly, to shape it. And so, what begins as humanism, our most sympathetic understanding and treatment of human nature, becomes transhumanism, the drive to fundamentally revolutionize what it means to be human by way of technological advancements. Changing human nature might be the most dangerous idea in all of human history, or perhaps the most liberating. Generally speaking, transhumanist thought does two things. First, it considers current trends to see how future technologies will develop and how they might affect us. Second, it calls for the use of current and upcoming technology to bring about beneficial societal change. We'll examine three central areas of transhumanist thought: superlongevity, superintelligence, and super well-being, dubbed the three supers because of their extraordinary transformative potential. So let's begin with a thought experiment to get your intuitions flowing. Consider this. An evil organization creates an airborne virus. It infects you and the entire human race. As a result, 100,000 people are dying every day. Within 30 years, 1 in 7, 1 billion people, will have died because of the virus. Now, how much money should world leaders put into research to develop an antidote? How high on a list of global priorities would you rate this? There is no denying the situation would be dire. Most people would demand immediate action. But hey, this is just a thought experiment, right? Not quite. 100,000 people really do die every day from diseases caused by aging. But no one treats aging as a global priority. So what explains this double standard? Are we simply resigned to death by aging? Aubrey de Grey, an expert in research on aging, argues that our priorities are fundamentally skewed and that we must start thinking seriously about preventing the huge number of deaths due to aging, the greatest cause of fatal diseases in the Western world. The goal of this strand of transhumanism is super longevity. Today, we have the minds and the equipment to begin developing technologies to combat aging. Unfortunately, we lack the will and the financial support to do so. Most of us are so accustomed to the idea of growing old that aging seems like just a fact of life. If modern medicine is supposed to keep us alive and healthy for as long as possible, then the anti-aging movement takes medicine to its logical conclusion. It's what happens when, as long as possible, means as long as we want. But what would a world without aging look like? How would we manage the huge population growth? And who would own the technologies that make it possible? These are huge questions, but we only have time to raise them. We'll investigate them in depth in future videos. Let's move on to the next area of transhumanist thinking. Every year, computers are getting more powerful. What used to fill up a room now fits in our pockets. More crucially, the time it takes for computer power to double is also getting shorter. At the outset of computing, the doubling process took 18 months, and this interval appears to be getting smaller. Plot this on a graph, and it's not a straight line, but an exponential upward curve. We need only project into the future to see that there is a point at which this line is practically vertical, a moment in human history referred to as the technological singularity. The futurist thinker Ray Kurzweil postulates that as these technologies develop, we will likely edit our bodies in order to integrate with computers more and more. This concept should be familiar. We're already in a symbiotic relationship with technology. You can send your thoughts at incredible speeds to recipients on the other side of the planet, find your precise location using satellites, and access the world's repository of recorded human knowledge with the device you carry with you at all times. And all of this was unthinkable 20 years ago. Out of this predicted computer capability explosion may eventually come artificial intelligence, a simulated consciousness in silicon. Given the rate at which an AI will be able to improve itself, it will quickly become capable of thought with precision, speed, and intelligence presently inconceivable to the human mind. If Kurzweil is right and we end up integrating ourselves with technology, we could be in private contact with this AI whenever we choose. The result of this is that we effectively merge with this AI, and its abilities become our own. This would propel the human race into a period of super-intelligence. But perhaps, as some argue, no non-biological computer could ever become conscious. Or what if, as every other dystopian science fiction plot goes, this AI's goals differ from our own? And what does our increasing reliance on computers mean for our future? Super longevity and superintelligence are all well and good, but only insofar as they make us happier, more fulfilled, more content. Let's look at the last section, which deals with the issue of well-being. Imagine you're soon to be a parent. Your doctor informs you that, if you wanted, you could choose certain features of your child's biology. You could choose how genetically prone to depression they will be, their levels of anxiety, jealousy, anger, and even their pain threshold. Would you choose a high likelihood of chronic depression, an intolerably low pain threshold? How about panic attacks and anxiety? Probably not. The last major branch of transhumanism, spearheaded by philosopher David Pearce, aims to investigate and phase out suffering. He argues that, ultimately, all our conscious states, our feelings, moods, and emotions are all an expression of our brain chemistry. For Pearce, it is clear that natural selection hasn't designed us to be happy, it's designed us to be good at surviving and passing on genes. A species that is permanently anxious and discontented will have a higher motivation to watch out for predators and take precautions for survival. But in today's world, these emotions are vicious. Our biology has barely changed in 200,000 years, which means that whilst culture and society has arguably made progress, we are still those same aggressive, jealous, anxious, savannah-dwelling hunter-gatherers. This is why Pearce argues that if we ever hope to increase the well-being of our species, we will have to edit our genes. Minimising our suffering, and the suffering of those we care about, is a crucial part of what drives us. Hence, so-called abolitionists argue that we start using modern technologies to do exactly that: minimise and eventually abolish suffering, ushering in an era of so-called super well-being. At present, every child is a roll of the genetic dice. Pearce argues that the least we can do is load the dice in our favour to create happier, healthier, longer-living humans. But might our compassion, curiosity, and pursuit of knowledge become secondary to our hedonism? If we're all content, why visit the stars? And isn't suffering sometimes a good thing? These are the three key areas of transhumanist thought, and we've only begun to scratch the surface. The three supers, super longevity, super intelligence, and super well-being, might radically change human history if, or when, they are realised. One of the main issues facing transhumanist ideals is that they are seen as far-fetched or perceived as just science fiction. But this is a big mistake. We are already transhuman. We're living longer, integrating more with technology, and emphasising quality of life. We're in the process of redesigning what it is to be human, only the effects are still so subtle and so slow that it doesn't look like much. But these changes will come faster and faster, and it's only wise to be an active, informed participant in the next stage of human development.
The world is on the verge of global change. The speed of data transmission has increased by multiples of millions. The rate of globally significant events and that of discoveries and crises is growing exponentially. Our civilization is like an uncaptained ship sailing on rough seas with neither chart nor compass, all the while moving faster and faster. The time we have to make the right decisions is shorter and shorter. We are facing the choice to fall into a new dark age, into affliction and degradation, or to find a new model for human development and create not simply a new civilization, but a new mankind. Historic crises show that to break the deadlock, we need technological revolution. It is clear that today's revolution will also require the deepest social transformation. The world's community and leaders should encourage mankind, instead of wasting resources on solving momentary problems, to focus on the technologies of the future: nanotechnology, biotechnology, information technology, cognitive technology, genetics and robotics. Doing so will allow us to find new sources of energy, create fundamentally new architecture and transportation, allow unprecedented developments of human cognitive abilities, refine artificial intelligences and brain-computer interfaces, simulate complex systems, create humanoid robots and cyborgs, and with the help of nanorobots, we may develop manageable matter. Find ways to transfer one's personality to an artificial carrier. Yet what we need is not just another technological revolution, but a new civilizational paradigm. We need new philosophy and ideology, new ethics, new culture and new psychology, and even new metaphysics. We must reset our limits, go beyond ourselves, beyond the earth, and beyond the solar system. This is an adequate response to the challenges of our time. Thus, new reality and future man will arise. Could it happen spontaneously, by itself? Unlikely. Humanity does not have a master plan of its development. It seeks stability. It lives in the present and does not plan. It preserves the status quo and tries to escape development. It does not tend to map future centuries and take responsibility for evolution. In consumer society's culture, there is no evolutionary vocabulary and rhetoric. To break the deadlock, the Russia 2045 movement was founded. It is a mega project intended to reach new heights and meanings. We intend to create a new vector for civilization, aimed at constant human development and evolution. As happened with the mega projects of the last century, the nuclear and the space programs, we integrate the latest discoveries and developments from the sciences: physics, energetics, aeronautics, bioengineering, nanotechnology, neurology, cybernetics, cognitive science. May everlasting spiritual ideals and values help us avoid going astray. Our project will give new meanings to the millions of people on Earth, will become a result of their joint creativity, and will lead us out of the impasse. Away from the murder of nature and physical death, forward to the realm of freedom and creativity, to the depths of the ocean and to the stars, to the infinite universe of our inner world. Our forecast for the next 40 years. February 2012. Global Future 2045 Congress is held in Moscow. It is a debate platform for discussion of our civilization's prospects for development. 2012 to 2013, the global economic and social crises are exacerbated. The debates on the global paradigm of future development intensifies. New transhumanist movements and parties emerge. Russia 2045 transforms into World 2045. Simultaneously, the 2045.com international social network for open innovation is expanding. Here, anyone interested may propose a project, take part in working on it, or fund it, or both. In the network, there are scientists, scholars, researchers, financiers, and managers. 2013 to 2014. New centers working on cybernetic technologies for the development of radical life extension rise. The race for immortality starts. 2015 to 2020. The avatar is created. A robotic human copy controlled by thought via brain-computer interface. It becomes as popular as a car. In Russia and in the world appear, in testing mode, several breakthrough projects. Android robots to replace people in manufacturing tasks. Android robot servants for every home. Thought-controlled avatars to provide telepresence in any place of the world and abolish the need for business trips. Flying cars. Thought-driven mobile communications built into the body or sprayed onto the skin. 2020 to 2025. An autonomous system providing life support for the brain and allowing it interaction with the environment is created. The brain is transplanted into an Avatar B. With Avatar B, man receives new, expanded life. 2025. The new generation of avatars provides complete transmission of sensations from all 5 sensory robot organs to the operator. 2030 to 2035, Rebrain. The colossal project of brain reverse engineering is implemented. World science comes very close to understanding the principles of consciousness. 2035, the first successful attempt to transfer one's personality to an alternative carrier. The epoch of cybernetic immortality begins. 2040 to 2050. Bodies made of nanorobots that can take any shape arise alongside hologram bodies. 2045 to 2050. Drastic changes in social structure and in scientific and technological development. All the prerequisites for space expansion are established. For the man of the future, war and violence are unacceptable. The main priority of his development is spiritual self-improvement. A new era dawns. The era of neo-humanity.
why we are posting. Human beings should be free. Free from death, free from limitations, from gravity, even from the biology maybe. And now it's the unique moment when I think the technology is advanced enough to be sure that it is not the science fiction, it's the reality. It's here, almost here.
I'm Ray Kurzweil. I'm an inventor, author, and futurist, and I invite you to join me at Global Future 2045, which will be a fabulous congress to explore the brilliant future ahead. Well, we're going to become increasingly non-biological to the point where the non-biological part predominates and the biological part is not that important anymore. In fact, the non-biological part, the machine part, will be so powerful it can completely model and understand the biological part. So even if that biological part went away, it wouldn't make any difference because the non-biological part already understood it completely. We'll also have non-biological bodies. We can create bodies with nanotechnology. We can create virtual bodies in virtual reality. The virtual reality will be as realistic as real reality. The virtual bodies will be as detailed and convincing as real bodies. We'll have different ways we can create bodies. We do need a body. Our intelligence is directed towards a body, but it doesn't have to be this frail biological body that's subject to all kinds of failure modes. Well, I think we'll have a choice of bodies. We'll certainly be routinely changing our parent body in virtual reality. So today you can have a different body in something like Second Life, but it's just a picture on the screen. Although research has shown that people actually begin to subjectively identify with their avatar. But in the future, it's not going to be a little picture in a virtual environment you're looking at. It'll feel like this is your body and you're in that environment, and that your body is the virtual body, and it can be as realistic as real reality, and the environment can be as realistic as real reality. So we'll be routinely able to change our bodies very quickly, as well as our environments, in virtual reality, but it will feel very real. We'll ultimately be able to do that with real reality too, like self-organizing swarms of nanobots that can link themselves up into a virtual body. If we had radical life extension only, we would get profoundly bored. We'd have a profound existential ennui, running out of things to do and new ideas. But that's not what's going to happen. In addition to radical life extension, we're going to have radical life expansion. We're going to have millions of virtual environments to explore. We're going to literally expand our brains. Right now, we only have 300 million pattern recognizers organized in a grand hierarchy that we create ourselves in our neocortex. But we can make that 300 billion, or 300 trillion. We can expand it. The last time we expanded it with the frontal cortex, we created language and art and science. Just think of the qualitative leaps that we can't even imagine today when we expand our neocortex again. We'll be thinking grander, deeper, more hierarchical thoughts than ever before, creating whole new institutions like art and science that we could not articulate. So we're not going to get bored. If that weren't the case, then I think living for hundreds, thousands of years would be a profound philosophical nightmare. Living for hundreds, thousands of years would be a profound philosophical nightmare. But instead, we're headed for radical life expansion.
Transhumanism is the idea that man is merging with technology. And Marvin Minsky asked the question, will robots inherit the earth? And he says, yes, they will, but they will be our children. Transhumanism is a term used for a broad range of ideas that technology is greatly enhancing human capabilities. This means everything from bionic limbs, brain implants, artificial intelligence, to even someday uploading our minds to the internet. And we have already a symbiotic relationship with our technology. And in that sense, we're already transhuman. And I'm sure many of you here today brought your smartphones with you. But transhumanism is the future of connectedness. It is technology as an extension of ourselves. It is technology as an extension of ourselves. So how do we get from where we are today to this science fiction future? Well, rarely are there major technological jumps. Technology usually follows a very predictable evolutionary path. Technology usually follows a very predictable evolutionary path. And it's the groundwork that's laid by these previous technologies that allow these new levels to be achieved. So for example, the cell phone infrastructure that was created allowed for the rise of mobile computing. And the internet allowed for the rise of social networks and cat videos. But it's hard to imagine where these advances take us as a species because we're only able to observe these small incremental steps along the way. Now, I believe that the pace of technology will evolve to the point where biology will become the limiting factor. The pace of technology will evolve to the point where biology will become the limiting factor. So how many thoughts can you hold on in your mind at once? 2, maybe 3? How many of you are still memorizing phone numbers? So we've already begun the process of augmenting the capacity of our minds onto our devices. So one of the principles of this evolution in technology is that technology is getting smaller, faster, cheaper, and more powerful every day. And in terms of physical size, it's 100 times smaller every decade. And this principle is known as Moore's Law. And it's the idea that technology will grow exponentially or double about every 2 years. And this is the type of growth that enables us to carry supercomputers in our pockets that once used to fill an entire building. And in the future, will allow us to carry computers the size of a blood cell. Now, wearables are widely believed to be the next big thing. And the concept of wearables isn't a new idea. For thousands of years, there have been tattoos and piercings and jewelry. I'm sure many of you may have, fitness trackers that you wear already. But in the near future, your family, your friends will have computers embedded into their clothing, in addition to their wrists and their glasses. But further along the wearable continuum will be things like embeddables, like small sensors that are placed underneath your skin. And as technology becomes smaller, cheaper, and more powerful, 50 billion devices will be connected over the next decade. And the internet will be more like a utility, like electricity. It'll be something you plug into. And this invisible force will connect all of these fragmented systems, becoming a digital nervous system for the planet that you, me, everything we interact with will become a part of. And soon thereafter, you won't just wear technology, but you'll ingest it. Smart pills powered by your body, using the same principle that allows a potato to power a light bulb, will monitor your vital signals for up to 72 hours, and it'll provide real-time insights on your health wirelessly to your doctor. And if this sounds a bit science fiction, it's already received FDA approval, and it's expected to be released in late 2015. So if that wasn't weird enough for you, Body 2.0 will include artificial eyes with zoom capabilities, infrared sensors, and night vision will be possible. And the prosthetic limbs of the future will be even more flexible and powerful than our original organic limbs that we have today. Retinal implants in a few years will allow the blind to see and recognize faces. And there are already arms that are controlled by human thought using a small implant in the brain. And if you think Inception was just a movie, MIT scientists have implanted memories into mice. They've isolated an individual memory in a mouse's brain and induced recall of that memory by forcing those neurons to fire. And researchers have also been able to pinpoint memories and erase them, eliminating them without affecting other memories. So in the future, it may be possible to remove or implant memories, and even treating things like dementia, Alzheimer's, post-traumatic stress, or personal traumas, or other types of neurological disorders. Now on the left, what you're looking at is a random YouTube clip. But on the right, it's a clip reconstructed from brain activity using an MRI. Now stop and think about that for a minute. This is the beginning of the age of mind reading. We're at early steps now in the research for seeing through another person's eyes. Perhaps one day it could record your dreams. And by the way, this video is about 3 years old, so they've come quite a bit further since then. Computer chips won't just be in your laptop or in your phone. Doctors will implant them in our brains. Doctors will implant them in our brains. Doctors will implant them in our brains, and they'll restore sight to the blind and hearing to the deaf. Today, there are already 300,000 people with cochlear implants. It's a form of a neural prosthetic that allows certain types of deaf people to hear. And Michio Kaku describes brain implants as your very own augmented mind. And this is the beginning of the brain net, a possible successor to the internet, a form of virtual telepathy that will allow you to create music, drive a car, communicate with other people, and even surf the web at the speed of thought. Movies will no longer be these two-dimensional slate tablets that you look into and blast sound at you. They'll be fully immersive experiences, complete with feeling and emotion the way the director originally intended. Everything is stored, every memory recorded, and available on a cloud service. And mind uploading will allow your friends to share their digital vacation experiences that never actually took place. It all just happened in their mind, similar to Total Recall. About 1.5 million years ago, the stone axe was the most sophisticated item on the planet. But for 50,000 generations, early man used the same stone tools with very little innovation. Until one day, an ancient ancestor decided to put water inside of an ostrich egg, and he buried that ostrich egg into the ground. And by doing that, he tapped into something that was truly remarkable for the human species: imagination. And these creative people, they lived on. Imagination is an insurance policy for problems in the future. And we are the species that transcends and transforms. It is what we are. And it's how we advance and extend our reach. But our biology hasn't changed in 200,000 years. We're still the same anxious and aggressive hunters that we once were. Perhaps we have not yet reached the pinnacle of human potential. Now, I realize that this may seem far-fetched in science fiction, and that some of these ideas maybe are a bit spooky. And that's simply human nature. And there are certainly ethical and moral dilemmas that we'll face as we go through this process. A recent survey reports that 72% of the US population is not interested in brain implants. About 53% think that it's probably a change for the worse, even if it could augment memories or increase our mental capacities. But that all changes with the children of today. But that all changes with the children of today. But that all changes with the children of today. Some call them digital natives. This is my son. And to him and his generation, anything that isn't computerized, digital, or touch-enabled will seem unnatural. Anything that isn't computerized, digital, or touch-enabled will seem unnatural. Anything that isn't computerized, digital, or touch-enabled will seem unnatural. To them, the smartphone is already an extension of their brain. To them, the smartphone is already an extension of their brain. To them, the smartphone and mind uploading powered exoskeletons will be just a part of life. Just a part of life. To them, the smartphone and mind uploading powered exoskeletons will be just a part of life. And transhumanism will seem just as natural as evolution. And transhumanism will seem just as natural as evolution. And anyone who doesn't follow suit will seem thoroughly inhuman. And anyone who doesn't follow suit will seem thoroughly inhuman. And anyone who doesn't follow suit will seem thoroughly inhuman. What new things could we accomplish if we didn't have the limitation of biology? What if our thoughts weren't limited to the 2 or 3 things that we can think about at once? What if we could explain our thoughts and feelings without the limiting barrier of words? Maybe we could explore the stars, save the planet, or enhance lives for people here on Earth. There's a lot to be hopeful for. And technology is not a panacea. There are many facets that make up this complex array of risks and benefits. But why wait until a breakthrough in designer babies? But why wait until a breakthrough in designer babies, before we begin the discussion? Or until nanotechnology is at our doorstep before we consider the risks and dangerous implications? Now, I believe that the future is hopeful. And I'd like to share another quick video called The Future is Ours. And it was created by my friend and director, Michael Morantz. And it's a video that's an inspiring message of hope. So I hope you enjoy it.
Climate change,
deadly earthquake,
a suicide bomber,
terrorism,
nuclear device in the heart of the nation's capital.
It's no wonder that we're pessimistic. It's no wonder that people think that the world is getting worse. But perhaps that's not the case.
Perhaps we forget that we've only just begun.
Doing what's never been done before is intellectually seductive, whether or not we deem it practical.
What we're seeing is unfathomably new possibilities all of a sudden becoming available to us.
The future doesn't belong to the faint-hearted. It belongs to the brave.
You have to trust in something. Your gut, destiny, life, karma, whatever.
We truly are living in an extraordinary time, and many people forget this.
So now technically your device is on. Can you tell? Can you hear me?
The yearning to do what we do in the service of something larger than ourselves.
Where did we come from? Are we alone in the universe? What is the future of the human race?
When you conduct those exercises, innovation follows just as day follows night.
We can do this. I know we can because we've done it before.
Stay hungry, stay foolish.
Terence McKenna said, "You are an explorer, and you represent our species. The greatest good you can do is to bring back a new idea, because our world is endangered by the absence of good ideas. Our world is in crisis because of an absence of consciousness. My goal is that I've awakened something within you. The world of possibilities is at our doorstep. Let's embrace it. Push humanity forward." Thank you.
No one is in control. No one is in control. No one is in control. This is something to know. Like, for instance, on this nanotech thing, if you read the journals, where the breakthroughs are just happening week after week after week. Well, then the question becomes, who is making this happen? Do governments want this? And they say, no, governments don't want it. It's so weird, they can't, they're just trying to come to terms with, the computerized battlefield. And when you start talking about aerosol, I mean, no, governments do not want this. Does the world corporate state want it? You say, well, no, they're not exactly sure how you make money off machines that cost 1/10 of a millicent each and are distributed by aerosol spray. They're having a little trouble with that. So then the question is, who wants it? Why is this moving with such incredible speed? And the answer is, who wants it is the people who are cool enough to make it. In other words, in these research and development skunk works and think tanks, all the heads love it. You say, this is so cool. Look at this. I've got 16 motors running inside a tube 1/10 the diameter of a human hair. Check this out. So everybody's staying off hours and scratching their head and getting drunk together, and it's incredibly exciting just to see if you can make it do it. Meanwhile, the managers are saying, but our whiter, brighter toothpaste project is falling behind, while you people, they say, but don't worry, we're designing plaque-removing nanocytes that will live under your wisdom tooth and come out twice a day. I said, well, all right. And what it seems to mean is that, information uses bright people. Information uses bright people. Information uses bright people. The thing that is I think the unspoken truth behind human history is that you can't trust smart people. You can't trust smart people. Information uses bright people. You can't trust smart people.
So one of the things that worries me, and I go back to this, is that all these regulations, and as you begin to put in all these red flags, people, the smart people, the people basically which are limited are not going to be doing this work. The smart people, which are limited, the smart people, which are limited, the smart people, the people basically which are limited are not going to be doing this work. We need to not only protect society, but also make sure that this community remains vibrant. And one of the problems in microbiology is that, and you can see this across fields, when fields don't have tools or where fields cannot do certain experiments, they tend to fall behind.
That you can't trust smart people. If you're the king, the prince, the pope, the bishop, the Lord High Chancellor, or whatever, you need them, but you can't trust them. What does it say in Julius Caesar? Yon Cassius has a lean and hungry look. Such men, send me such men as are sleek-headed and fat, such as he think too much. Those thin guys who think too much. And they'll work for you, they'll appear loyal, but at the first chance, they either run off with your bank account, your woman, or your technology, and maybe all three. And it was ever thus. So intelligent people are largely in the category targeted by psychedelics. And so, you can't trust them to be heterosexual, you can't trust them to be unstoned, you can't trust them to vote right. They're just always slipping out of the corner of your eye and doing some horrible and incredibly subversive thing right under your nose, and yet you need them. And this is happening more and more. I mean, I go to these skunk works places like Paul Allen's Interval in Palo Alto and all that. And they're just colonies of the most stoned, arrogant, smart people I've ever met. And what they're trying to do is produce weirder and weirder things for their own amusement. And I think they serve the zeitgeist very well. I mean, people say, well, they're spoiled and they're pampered and why. But on the other hand, this is where the first computers came from. This is where virtual reality and the glove and all that came from. It's where the net came from. Apparently, creative dreaming in the presence of technology is a place where haunted by genies, genies that will wrap you in the blanket of their mysterium and lead you forward. This has always been known. I mean, alchemy, interestingly, is about, of all the traditions of the West, it maintains a kind of shamanic connection. The alchemist, the artificer, is aided by demons, D-E-A-M-O-N-S, the demons of artifice, Blake called them. And they teach you how to make things. But as was discussed yesterday, is it a con? Whose agenda does it serve? They only teach you to make certain things which amuse their demon aesthetic. And so, our progress into the domain of techne has been at the behest of this unseen companion and its curious obsessions. But to make machines smaller, tighter, the Swiss have been at this for a while, but the best Pierre Cardin watch is gross beyond all imagining compared to nanotechnology.
That was the phase of his evolution that I sometimes think of as the techno phase. He was at that time preoccupied with nanotechnology and prosthesis and the use of various technologies to extend the capabilities of the human species to the next stage of evolution beyond humanity, the so-called post-human stage of evolution.
Perhaps with some amount of prosthesis, electronic processing, signal processing, I'm not sure what it would be. If we had $500,000 even, which isn't a lot of money, we could probably find out. But we need to build an environment where we can produce acoustical signals that we can see and play with in real time. And I've seen, and you probably have too, things somewhat like this. I've seen spaces that you could dance into and create a visual impression on a screen. In other words, a system of lasers is being interfered with by your body, and it's being uploaded into a complex hallucination-like piece of performance art. But I think we need to push on visual language. I think this is what these elf things want us to do. And that, you see, from an engineer's point of view, what we're talking about here is what's called bandwidth. And bandwidth is more and more the problem. Bandwidth is how fast you can move information between 2 points. And the more bandwidth you have, the more complicated and detailed the information you can move is. When I speak to you, this is one of the most inefficient forms of communication ever invented. We're going here at about 30 baud, 30 to 40 baud. The baud rate means bits per second. A standard modem operates somewhere between 14,400 baud and 28,800 baud. An ISDN line is 5 times faster than the upper end of that. T1 is 10 times faster than that. Bandwidth allows depth of field, and communicating at 30 baud with an acoustical signal of small mouth noises is just, it's amazing that we can crawl onto the theater of discourse and claim to be communicating at all. And yet, notice, we have built a global civilization on small mouth noises and the consultation of local dictionaries. In other words, we don't even have a shared sense of meaning. When I talk and you listen and you think you understand, the unspoken assumption is that your dictionary and mine is the same. And since we're all English speakers, it's maybe 60% true. But since you're of one tribe and I'm of one from the other side of the planet, we don't even have the comfort of local associations. And so as you listen, you strive for a kind of internationalizing of your dictionary. And as I speak, I try to be as non-colloquial and self-referential as possible and speak a kind of standard international English. But it's a feeble thing, hedged about by these kinds of limitations. If we could produce language sculpturally, a lot of the ambiguity would be drained from the system, and every iota of ambiguity that leaves the communication enterprise brings us closer together. I mean, our whole task is to make ourselves understood and to understand others as they wish to be understood, not as we wish to understand them. And so telepathy, for my money, is not hearing people think, it's seeing what people mean. And it may lie ahead of us in the future, springing out of our own technologies and neurological and physiological organization.
Paul, telomerase hit the headlines. However, I think we have to put it into perspective. It is not the fountain of youth. However, it is a significant breakthrough. We have to put it into a much larger perspective. First of all, we know that DNA is sort of like a shoelace. It has plastic tips at the end. Every time a cell reproduces, the tips get shorter and shorter and shorter until finally they fray. And you know that your shoelace without the plastic tips will simply fall apart. That's what happens inside a cell. A cell, for example, your skin cell, will divide about 60 times. That's called the Hayflick limit. Then the cell goes into senescence and eventually dies. So in some sense, every cell has a biological clock. It is doomed to die after about 60 reproductions. However, telomerase can eliminate some of the contraction of the chromosomes, and the chromosomes can maintain their length. So at first you may say, aha, we can now defeat the biological clock, but not so fast. First of all, cancer cells also use telomerase. Cancer cells are immortal. Cancer cells are immortal, and that's precisely why they kill you. Why are cancer cells so dangerous? Because they are immortal. They grow and they grow and they grow until they take over huge chunks of your body, meaning that your bodily functions cannot be performed and you die. So we have to make sure that when you hit ordinary cells with telomerase, that you don't also trigger cancer in the process. Now, also you have to realize that genes are also very essential for the aging process. It turns out that we know what aging is, Aging is the buildup of error. That's all aging is. The buildup of genetic and cellular error. As cells begin to age, they begin to get sluggish because genetic mistakes start to build up. Now, cells, however, have a repair mechanism. They can repair damage to their cells. Otherwise, we would all basically rot very soon after birth. However, even the repair mechanisms eventually get gummed up, and then the cell really starts to get old as a consequence. So then the question is, can you accelerate cell repair? That is another branch of gerontology which is being looked at, using genes and using chemicals to accelerate the repair mechanisms. For example, if I take any organism on the planet Earth, from yeast cells to spiders, insects, rabbits, dogs, and even monkeys now, and I reduce their caloric intake by 30%, they live 30% longer. In fact, the only organism which has not yet been deliberately tested by scientists are Homo sapiens. All the other species obey this basic rule. You starve them to death, they live longer. This is independent of telomerase. This is a function of the wear and tear that we have on the cells. And this is the only known way of actually deliberately extending the lifespan of any organisms almost at will. Now, what we want is a genetic way of mimicking this mechanism without having to starve yourself. Because how many people do you know would be willing to starve themselves in order to live 30% longer? Not too many. So then the question is, are there genes that control this process? And the answer is apparently yes. There's something called the sirtuin genes, SIR2 being the most prominent of them. They in turn stimulate certain enzymes, among them resveratrol, which is found in red wine, for example. So this does not mean that drinking red wine or taking telomerase is the fountain of youth. I don't think that anyone has a fountain of youth yet. What I am saying is, we are now finding pieces of the fountain of youth, tantalizing clues that mean that perhaps in the coming decades, we might be able to actually unravel the aging process. We don't have it yet. Don't go out to the drugstore and stock up on these kinds of chemicals and enzymes thinking you're going to live forever. However, it is conceivable that in the coming decades, we'll come very close to finding it.
One of the great things about transhumanism is it's kind of what we call transhumanitarianism. There's this idea that we're really trying to help people. We're really trying to solve suffering, solve medical issues. And exoskeleton technology is one of the most important. About a third of everybody on planet Earth has some type of mobility issue. So exoskeleton technology is really leading the way in terms of getting people to be able to be mobile again and do things like that. And what's really interesting is also that you think you can't climb Mount Everest, but there's a chance sometime in the future you may be able to climb Mount Everest just because you have exoskeleton technology. There's also the idea that you might be able to run a marathon. As I wrote recently for the New York Times, you get the exoskeleton technology working well enough, you'll be able to run on water. I mean, this stuff is really incredible. So can you imagine running a marathon on water? Well, this is what people want to do with exoskeleton technology. Now, I would say the biggest thing about transhumanism, and where a lot of people go for getting into pocketbooks of investors, is really when you start talking about disabilities. A huge amount of people around the world have disabilities. So we already have things like this, the Argus 2, which allows a blind person to basically see, navigate streets, go into rooms and things like that. So I think, overcoming disability is really a huge part of also what we're doing. And that goes as far as cochlear implants with deafness. We don't really have deafness anymore on planet Earth. People that are born that can't hear end up having a cochlear implant put in, if they're allowed. There are some cultural differences too. But deafness as a disability has been mostly wiped out due to radical technologies. Again, transhumanist technologies. And of course, one of these things is putting implants in your body, implants in your brain, implants in your arms, as I have in my hand, and doing all sorts of things. We call this biohacking, where you actually put all sorts of stuff in yourself to monitor what's going on and also to help you do things that you wouldn't be able to do, like hear or see. So the idea of brain plants is really complex. I was doing an interview earlier, and they were asking about the future of work. And you know, where I'm from, Silicon Valley in Los Angeles, California area, there's a couple different companies now like Neuralink with Elon Musk that are working on basically brain implants in order to allow your brain to interface directly with machine intelligence. Now, why is this important? Well, machine intelligence is important because, you know, already like in Wall Street, for example, there's a bunch of mid-level traders that are being wiped off the floors. And the reason is because they can't compete with algorithms. So what a lot of kind of entrepreneurs want to do is, in order to have humans keep up with machines, they want to merge our brains with machines so that we can start thinking in real time in the internet. So the thing you have in your pocket, cell phone, smartphone, basically can be put in through your head through an implant or a headset. Already in Florida, they're doing things like racing drones around a track and stuff like that, just using brainwave technology. And already you can have conversations in your mind with people through brainwave headsets and brainwave implants and things like that. And we're going to get to a point, you know, Elon Musk says there'll be a commercial device in 5 years. I don't know if that's the case, but definitely within 10 years, there's a good chance that there will be a commercial device on the market that will allow you to talk to your friend or give a speech in real time like I'm doing now, where people would actually hear it and listen and maybe answer back, and you would hear it in your head. So the question, are we going to get brain implants? It's almost 100% yes.
I think that's right. If you start limiting the human ability to question and push the boundaries, that's going to kill the field. And in particular, the issue that we were discussing, that Jerry raised about unlikely to arise, there's a whole area now of biological research looking at stochastic events. There's a whole area now of biological research looking at stochastic events. There's a whole area now of biological research looking at stochastic events. And it turns out, and I think you've even, it turns out that the unlikely events may in fact be very, very important. In fact, they are very important. So this notion of being able to predict ahead of time that this may or may not be important because it's unlikely probably won't fit in with the current way we think about important evolutionary concepts. So stochastic events, unlikely events, may in turn out to be very, very important. And whether that's a combination of other unlikely events or just a single unlikely event, we don't know just yet. But we have to be able to look at it.
I'm a basic researcher, but I'm also a biosafety professional. So I fully understand the benefits of basic research. And I think that if we do gain of function research, there's no doubt we're going to learn a lot. But what I was hearing from the first panel is that what we learn may not directly translate to saving us if this were to be released accidentally or however. And so, you know, and that's often the case with basic research. It's not directly translational. Sometimes it can be. And I'm also not convinced that the natural transmissions that have occurred, that goes back to the question that I asked in that session, I don't think we fully exploited the information that we could have gotten from those natural transmissions and done all of the molecular analysis and biochemistry and everything else that could be done. And if we haven't done all of the work on the natural ones, why create new ones that we then have to study as well? And so I strongly support studying some of the natural isolates. And I think that there perhaps are experiments that we should not do. And I think we really need to seriously review the ones in creating mutations that might not occur naturally. And I think we really need to seriously review the ones in creating mutations that might not occur naturally.
I have a lot of trouble with cloistering information, especially scientific information. Anytime you try to put what you're doing under any sort of a silk cover or an iron shield, you create a perception that something is happening that shouldn't be happening. And so the perception becomes very, very powerful. And the only way to break that perception down that you're not doing something wrongful is full disclosure. Now, help me here, because I might say this wrong, but one of the groups that was very successful in sort of containing their research work was the nuclear physicists around the Manhattan Project and years after. They formed the Janus Group, I think it was called.
I can't think of a moment bigger than this one.
I've likened this as the moment for biology akin to the moment when fissile nuclear material was figured out by physics and offered both a way to solve the problems of the universe and nuclear weapons.
If one is not able to disclose fully, it means that the benefits globally are not justifiable enough to make it happen.
So one issue that has come up repeatedly, including by the questions that I laid out, is the usefulness of the information in combatant preparedness. And I think it is important to realize that even if one cannot at this moment identify an immediate usefulness, that humans are very creative, and that information may become useful in the future.
I think we need to think about the timing of the experiments, and maybe the question is, are we ready for these experiments now? In my scenario, I put forth some arguments thinking of the worst case scenario. We talked about the combination of attributes, whether that's intentional or it comes about during this experimentation. Increased virulence, increased transmissibility, and not amenable to a vaccine. If those things came together, and they could, whether it's intentional or unintentional, and it accidentally gets out, I'm not even saying intentionally gets out, it gets out. We can't handle it. We are not ready to handle it. We can't handle it. We are not ready to handle it. And so should we be doing something right now that we are not ready to handle? We can't handle it. We are not ready to handle it. And so should we be doing something right now that we are not ready to handle? That's a question I'm putting out.
Since we understand that H5N1 is mostly in Asia so far, and also in animals, and then we don't know that in natural what happened with the mutations. I think I'm agree that the experiments that should be done now is how we can prepare or prevent for the disease. It's rather than create a new one virus. The research has to be in the appropriate laboratory, and then in developing countries there is no, maybe about the climate also can also affect the mutation itself. So we don't know so far if we make a new one creation to understand how the transmissibility or pathogenicity or something, and maybe will make worse. Maybe will make worse about altering host strains and everything.
I heard from Dr. Rubin that, of course, it is obvious that we have some institutions around the world who are capable of managing this sort of work, and others that can't, and we have to make clear that there is this distinction. The problem is that you can identify them, but you can't really prohibit anyone to do anything. I mean, how are you going to stop them? You can't really prohibit anyone to do anything. I mean, how are you going to stop them? How are you going to stop them? How are you going to stop them? And this goes back to other things that we've heard this morning, which is that you can't stop technology. I mean, you can find anything on the web, and so my concern is that if you put in place a regulation and you say these are labs which can and these are labs which can't, the ones who can't, if they want to do it, they just say, well, so what? They just say, well, so what? The ones who can't, if they want to do it, they just say, well, so what? You know, if I want to do it, I'll do it. And the key question, which was posed by Tony Fauci at the start, is should NIH fund this work? And therefore, NIH only funds work which can be disclosed. So I think that we all recognize the importance of work. There's a gray area which maybe the benefits of releasing all the information may be not as, they're not, maybe not an efficient cost-benefit analysis. So what I'm asking you is, do you refer to classifying the research, or do you refer to not doing it at all?
Well, based on the other things that we have heard here, for example, the concerns about biocontainment, the concerns about biosafety, the fact that we are learning how to handle this, I think that unless you can come up with a clear scientific rationale for doing the work that is just not curiosity, then I don't see that the work should be done at this time, classified or unclassified or whatever. But I want to emphasize one thing, that I think it is really important that whatever gets done here has sunset provisions that will force the community to rediscuss it again. You know, if you're going to basically end up with a moratorium, 1 year, 2 years, and then rediscuss it again, because things are changing very rapidly. And I may give you a different answer a year from now. I think a year from ago, I would have given you a different answer. At the time, I was not as aware of the issues that we're discussing, and I would have probably said, you know, you should not, science should be unfettered. We should be driven by curiosity. And we should learn because we don't quite know how this information is going to become useful. So my own view has shifted, and I think it may shift again. And I emphasize, my one thought to you is to make sure that whatever gets done has sunset provisions, so that it forces people to have this discussion again.
You know, people who are intent on doing bad things with biology are drawing on the same science that all of the people in this room are drawing on. There isn't a separate set of science that's biological warfare. There isn't a separate set of science that's biological warfare. You know, people who are intent on doing bad things with biology are drawing on the same science that all of the people in this room are drawing on. There isn't a separate set of science that's biological warfare. There isn't a separate set of science that's biological warfare. And so it seems to me there's always a risk that we willingly accept that somebody will try to misuse science that was done for perfectly good reasons that creates lots of benefits in other ways. There's always a danger that somebody will find a way to misuse it, and I don't think you can predict that.
Indeed. On October 8, 2002, CBS News reports that the United States admits bioweapons tests. The United States secretly tested chemical and biological weapons on American soil during the 1960s, newly declassified Pentagon reports show. The tests included releasing deadly nerve agents in Alaska and spraying bacteria over Hawaii, according to the documents obtained Tuesday. The United States also tested nerve agents in Canada and Britain in conjunction with those two countries, and biological and chemical weapons in at least 2 other states, Maryland and Florida. The summaries of more than 24 tests show that biological and chemical tests were much more widespread than the military has acknowledged previously. The Pentagon released records earlier this year showing that chemical and biological agents had been sprayed on ships at sea. The military reimbursed ranchers and agreed to stop open-air nerve agent testing at its main chemical weapons center in the Utah desert after about 6,400 sheep died when nerve gas drifted away from the test range. But the Pentagon never before has provided details of the Alaskan, Hawaiian, Canadian, and British tests. The Defense Department planned to formally release summaries of 28 biological and chemical weapons tests at a House Veterans Affairs Subcommittee hearing Wednesday. The documents did not say whether any civilians had been exposed to the poisons. Military personnel exposed to weapons agents would have worn protective gear, the Pentagon says, although the gas masks and suits used at the time were far less sophisticated than those in use today. The head of the House Veterans Affairs panel called for further investigation of the tests. And then we can add the unethical side of many of these unscientific experiments being done to further the futurist and transhumanist agendas. That is, creating synthetic life and synthetic environments. On January 21, 2019, CNN Health reported, "Unethical experiments, painful contributions to today's medicine." Medicine is very much a word like science. It's not actual medicine, it's chemicals, poisons, radiation, and other so-called treatments that have nothing to do with medicine. Scientist takes on genetically modified humans. Chinese scientist Herein sent shockwaves around the world last year with his claim that he had modified twin babies' DNA before their birth. The modification was made with gene editing tool CRISPR-Cas9, he said, and made the babies resistant to HIV. Scientists from China and around the world spoke out about the experiment, which many say was unethical and not needed to prevent the virus. The scientists had also been warned by peers not to go down this path. But remember, in scientism, science must go forward regardless of morals, ethics, values, etc. His experiments, which are still clouded with the uncertainty of his claims and his whereabouts, open a Pandora's box of questions around ethics in experiments with humans, even though these dilemmas aren't new. Historic examples of human experimentation include wartime atrocities by Nazi doctors that tested the limits of human survival. Another led to the creation of the hepatitis B vaccine prototype. Wendell Johnson, who made several contributions to the field of communication disorders, tried to induce stuttering in normally fluent children. In the 1940s, prisoners in Illinois were infected with malaria to test anti-malaria drugs. Such experiments have been criticized as unethical, but of advanced medicine and its ethical codes, such as the Nuremberg Code. When he made his claim of genetically altering humans, the response from the global medical community was swift and condemning. It is out of the question that the experiment is unethical, said Jingbao Nai, professor of bioethics at the University of Otago in New Zealand. Without medical necessity, it is not ethical to carry out gene editing. Sarah Chan, director of the University of Edinburgh's Mason Institute for Medicine, Life Sciences, and the Law, adds that the balance of risks and benefits made it hard to justify this experiment. Genome editing of embryos is still not fully established, and virtually all scientists will say we don't yet know enough about it to be able to recommend that we just go ahead with it clinically, she said. If it were the case of a life-threatening disease that will cause tremendous pain, and the one way to alleviate this pain would be a risky experimental procedure, then Chan thinks, given the immense benefit, perhaps taking that risk is justified. When it comes to medical ethics, different principles need to be weighed against each other by an institutional review board deciding over experiments involving human participants. I don't know about you, but I'm a little tired of ethical review boards full of experts. What is immoral should stay immoral. What is unethical should stay unethical. The advancement of science, and for that matter, the forced evolution of man and nature, is not an excuse to lose our moral and ethical foundations. As a definition of medical ethics and researchers commonly hold that there are 7 general rules for an ethical experiment involving humans, explained Govind Persad, assistant law professor at the University of Denver. Experiments should be socially valuable and scientifically valid, and people have to be selected fairly and respected. The risks and benefits to participants and the benefits to society need to be weighed against each other, and there needs to be an independent outside review of the ethics of the experiment, Persad said. And it would be nice if the participants in the experiment actually knew it was happening, thank you, U.S. government. The risks and benefits equation sometimes includes third-party consideration, such as tests of a vaccine that includes a virus that can shed and infect others who are not research participants, Persad said. Research on smallpox vaccine is one example. And, as we'll discuss in future documentaries, this is one of the big problems with vaccines, is that you shed the disease and are contagious for at least 2 weeks after receiving the vaccine. And yet you send your kids to get vaccines and send them right off to public school. If He's experiment produced any mutations, these could be passed down to the twins' children and then diffuse into the general population, which didn't consent to that change, Persad explained. And again, we'll cover CRISPR-Cas9 experiments and how they affect the DNA chain in future documentaries. I don't know how large of a risk that is, Prasad said, because again, it depends on the odds of the mutation, whether the mutation was one that would end up staying in the population or whether it would be selected out over time. And don't forget that this is the definition and indeed the application of what is called eugenics. Many of the national and international protocols, like the 2005 UN Declaration on Human Rights and Bioethics, include some of these 7 principles, Prasad said, but as with most international documents, these protocols are not legally binding. The first document, outlining how research should be done in a fair way, was a product of Nazi war atrocities. But some doctors felt this Nuremberg Code did not apply to them. Hunter Henson released an article in 2017, The Top 10 U.S. Government Experiments Done on Its Own Citizens. Throughout history, the U.S. government has managed to keep secrets hidden from the public. When they release these secrets, many are quite surprised and astonished at everything they've managed to get away with. Countless conspiracies involving programs such as Area 51 and MK-Ultra have become mainstream in modern-day culture. The desire for such secretive knowledge is ever abundant, and no matter how much we think we know, we're always proven wrong. Despite the current rise in conspiracy theories involving government testing, there are many verified instances of such cases that have remained hidden from the public for decades. Here are 10 of those. Most people are familiar with the Tuskegee syphilis study. The U.S. Public Health Service conducted this study beginning in 1932. A total of 600 African-American men were chosen to be involved in the study, 399 with syphilis and 201 without. Those with syphilis were denied proper treatment for the disease. The government wanted to ensure that they were able to track its progression without interruption by medication. The men were never given the option to participate in the study because they were never told it was an experiment. The experiment was only supposed to last for 6 months, but it became a long-term study that lasted up to 40 years. When penicillin became the main drug to treat syphilis, the patients were denied access and weren't given the option to opt out of the study. In exchange, though, they were given free medical exams and burial services. A lawsuit was later filed, and the government granted free burial services to all surviving patients. Perhaps they'll do that to the COVID-19 victims. The Stateville Penitentiary malaria study was conducted by the U.S. government in the 1940s at Stateville Penitentiary, which is located in Illinois. It involved over 400 prisoners who were illegally infected with malaria and subject to studies. The goal was to test experimental drugs in an effort to find a cure for the disease. In addition, the tests were administered and documented solely by the prisoners themselves. Not only were they the patients, they were also the proctors. The prisoners also decided which of them would take part in the experiment. The testing process counted toward their sentence and allowed some to serve much less time. The prisoners would also choose who was eligible to receive a reduced sentence. However intriguing this may have been for the prospective patients, the experimental drugs often had irreversible side effects. One of the most famous prisoners involved in the experiment was Nathan Leopold, who many may recognize from the Leopold and Leib murder case in 1924. He stated that the prisoners would often deal with the horrifying side effects without complaint. Despite the immorality of this experiment, it was praised by many for the benefits it would create for society, just as gain-of-function work is being praised for the same reason. Citizens saw it as a sacrifice to find the cure for malaria at the prisoners' expense. And then there were Navy-sponsored beef blood transfusions. Edward Cohn, a biochemist working at Harvard University, conducted an experiment in 1942 with sponsorship by the U.S. Navy. The Navy had contracted Cohn to engage in this secret project to discover a possible biological weapon. His work involved injecting prisoners with cow blood in an effort to detect a protein that could be used in the event of an upcoming war. The 64 subjects who were injected with the cow blood all suffered catastrophic effects, ending in death. And it is important for you to know that cow serum, that is blood, is used in many vaccines that are given to yourself and your children, along with other animals, insects, and fetal tissue from human aborted babies. Although this government experiment ended in failure, it was soon learned from Cohen's methods that the true way to identify the protein was not in cow blood, but in human blood. The methods were replicated using human blood, and the protein was not only isolated, but it was also pure. Instead of being used to harm others, this protein was later used to effectively treat shock patients. There was also plutonium testing. During the mid-1940s, the U.S. was busy with the Manhattan Project, the effort to create the atomic bomb. Because the effects of radiation from the bomb were largely unknown, the government spent years studying them, including with experimentation on its own citizens. Plutonium is one of the many radioactive materials the government used in these types of tests. Patients would receive doses of radioactive plutonium in the form of injections. The majority of these patients were terminally ill, which made the results of the experiment difficult to fully understand. They were never told what was being done to them, partly because the word plutonium remained a government secret until after World War II. Although most of the patients did not die from the effects of the plutonium injections, the government's secrecy and willingness to subject its own citizens to such experiments raised suspicion from many. During World War II, there were mustard gas experiments. The U.S. government conducted many experiments on its own soldiers to test the efficacy of gas masks and protective clothing. It is estimated that nearly 60,000 human subjects were used during the studies, mostly Caucasian men. However, many Japanese and African Americans were also used to identify any possible skin differences within the experiment. There were several different tests used to determine the strength of such protective clothing in both gas chambers and the field. Field tests involved releasing the chemicals in an open area outdoors, using human subjects to test particular clothing items and monitor the effects the chemicals had on nature, such as on animals and water quality. There is also evidence that some of the soldiers were not offered any protective gear or clothing. In chamber tests, troops wore the masks and clothing and stood in the deadly gases from 1 to 4 hours. The tests were repeated daily using the same people as subjects until they exhibited dangerous reactions to their exposure. Now, I want to stress again here that all of this is considered as legal and not against the Biological Weapons Act because of the exception clause that says they can do this as a peaceful or friendly test, a so-called scientific or defensive test. In Operation Paperclip, the U.S. raced against the Soviet Union to attain as many Nazi scientists as possible before an impending war. The many advances German scientists had made, including synthetic rubber and much more, led to them being the most sought-after researchers. If the U.S. could employ these scientists, they could use their intelligence, the intelligence of unfettered, unregulated smart people, to develop many more advancements, ensuring national security if the Cold War were to escalate into another world war against the Soviets. The U.S. paid the Nazi scientists to work for the government, providing them with immunity from prosecution for their offenses following World War II. This is not unlike offering basic research scientists who are knowingly creating weaponized bioweapons by calling their work friendly, defensive, and legitimate. This immunity allowed them to escape jail and possibly execution. The U.S. government also offered to care for the scientists' families if they agreed to work for them. Originally, only rocket scientists were wanted, but the U.S. eventually employed up to 1,600 Nazis until 1990. These actions were highly illegal and remained extreme government secrets for many years. In September 1950, the U.S. Army was involved in a secretive experiment to test the possibility of biological warfare near the West Coast. They did so by releasing biological weapons into the streets of San Francisco to test their effects. They released a type of bacteria to gain information on how it would affect the population. this was done without the citizens' consent. By the end of Operation Sea Spray, 6 different biological warfare tests had been performed on the residents of San Francisco. These releases killed many people and hurt several more. The government then concluded that it was very likely for a coastal city to be affected by such warfare. One of the many deaths caused by this experiment was Edward Nevin. He died after bacteria from the government testing spread from his urinary tract to his heart. Other cases soon followed him, but many were cured after long, torturous hospital stays. Again, this is reminiscent of the current COVID crisis. But as we've seen, the problem is that government doesn't want to admit nor take liability for its actions. It is also important to note that many of these biological weapons tests and experiments were admitted to only after the statute of limitations had run out. It's very easy to admit your crimes when you can no longer be punished, or when the people who actually carried out the experiments are now dead. And then there's Operation Big Buzz. This catastrophic experiment conducted by the U.S. government in 1955 may not seem as harmful as it was. The government released millions of mosquitoes known to carry yellow fever into Georgia parks. The bugs quickly dispersed into the suburbs. The goal was to determine how effective insects could be in biological warfare by tracking biting habits on citizens. Although these mosquitoes weren't infected with yellow fever, the government still tested the potential for biological warfare by experimenting on its own citizens, with the people of Savannah, Georgia being the target. It is also recorded that government officials disguised themselves as healthcare officials in order to effectively record the mosquito bites and track their locations. Many other experiments similar to Operation Big Buzz were also performed, such as Operation Dropkick and Big Itch. Operation Dropkick is very similar in that they both tested mosquitoes in Georgia. Operation Big Itch involved the government releasing fleas into the public to study their biting and travel habits. Like the goal for Operation Big Buzz, Operation Big Itch was meant to determine how effective fleas would be in spreading disease in biological warfare. All of these studies show the government's determination for developing advanced biological warfare methods. And let us never forget the Willowbrook experiments. The extremely shocking experiments were aimed at discovering a cure for hepatitis. The continuous study lasted from 1956 to 1970. The subjects were taken from Willowbrook State School, which is located in Staten Island, New York. They were mentally handicapped children. The series of tests involved injecting the children with experimental drugs that were meant to cure hepatitis. Not only were the children unable to provide consent, but they would often die from the treatments. When questioned about their actions, officials justified themselves by stating that hepatitis was prevalent in the institution and nearly all patients would become infected anyway. The children who did not contract the disease naturally were infected by the administrators to carry on the experiments. Thousands of vaccine experiments included many on the measles. Experiments involving the measles vaccine were conducted from 1990 to 1991 by the Centers for Disease Control, the CDC. The doctors wanted to know if they could use it to replace natural antibodies in babies. To test this, doctors injected thousands of babies in the Third World with the drug. The vaccine eventually led to several immune problems in the babies and caused many deaths, although the exact number is unknown. Knowing the drug had this effect, the government still tested on African American and Hispanic babies in Los Angeles. They injected more than 1,500 babies in the United States with the experimental drug. However, the study came to an end when it was discovered that African children were dying at an alarming rate up to 3 years after receiving the vaccinations. It is important to note that most side effects from vaccinations happen long after and up to dozens of years after the vaccine is administered. Thus, many of the side effects of vaccines are never reported. The CDC later admitted that the parents were unaware that their children were being injected with an experimental drug that had not yet been verified by the Federal Drug Administration. Unfortunately, all vaccines are experimental drugs. None of them are tested for safety and effectiveness, and the Federal Drug Administration admits openly that the majority of the vaccine trial happens after the vaccine is given to the general population, where the public is supposed to report adverse vaccine reactions. Every vaccine you receive is an experimental drug. Remember, the FDA does not conduct any safety trials and relies on drug companies to do it themselves. Another article included 10 terrifying U.S. government projects carried out on domestic soil. There's no doubt that the U.S. government, as well as governments around the world, are into some pretty shady stuff. One of the most notorious U.S. government operations is probably MKUltra, which was the blanket name for the CIA's attempt at mass mind control. And for those that don't believe that's true, it's actually on the CIA's website. That's far from the only questionable and frankly terrifying government project that has been done on American soil. There's Operation Big Itch again. And the Dugway Proving Grounds in Utah was established as a military installation with the reallocation of public lands in 1942. By the 1950s, there was a definite need for a remote testing facility, a role that Dugway Proving Grounds would fill nicely. It became the location of the Biological Warfare Assessment Laboratories in 1954. With that title came the necessity of figuring out whether or not insects were viable for use as delivery systems for various types of nasty diseases. Specifically, they wanted to know if they could deliver the plague via fleas. Operation Big Itch dropped countless fleas on the Utah desert. Cages of guinea pigs were set up on the ground to gauge how successful the drop was, as there were concerns about whether or not the fleas would survive and how much they would spread if they made it to the ground. The fleas were sealed in containers that were designed to rupture with the firing of a CO2 cartridge at an altitude of 300 to 600 meters, or 1,000 to 2,000 feet. The experiment was something of a success. The guinea pigs became infested with fleas, but it was also decided that it was necessary to drop the fleas close to the target to make sure that the bugs made it to their hosts. Not all of the drops went off flawlessly, though. In one attempt, one of the cartridges fired while it was still in the plane, and the crew was swarmed with fleas. Overall, the test was enough of a success that there were plans in the works to build a massive flea breeding facility that would raise 50 million fleas a week. Producing enough plague virus for all of those fleas wasn't as easy, however. When researchers failed in their attempts to figure out how to do so, the plan for this biological weapon fell by the wayside. In Operation Do 1, dozens of zinc-cadmium sulfide containers were released in 5 trials from the U.S. Tercel over a test area over North and South Carolina and Georgia. The ship ran a parallel course along the coast, and although the first trial's contaminants were swept out to sea, the aerosol releases in the other 4 trials went up to 280 kilometers, or 175 miles inland, across all 3 states. Do 2 released spores from Lycopodium, a type of fern. Do 1 wasn't the only time that the Army played with the zinc-cadmium sulfide either. Similar batches were released over Minneapolis, St. Louis, Corpus Christi, Fort Wayne, and as many as 29 other locations across the U.S. Zinc-cadmium sulfide was used for a few different reasons. It was generally accepted as safe for humans, plants, and animals. It would disperse like a biological weapon, and it was cheap. It also shows up under UV light, which made it easy to see where it ended up. Needless to say, people were rather outraged when it was found out that they'd been test subject in a mock biological weapons strike. A subcommittee was put together to assess whether or not that chemical was harmless as the Army claimed. Basic results of experiments like feeding the compound to rats suggested that it was harmless, but the subcommittee recommended that the Army had better go back and run some more tests because they weren't sure what the ultimate effects of the compound really were. especially when it was inhaled in any quantity. The cadmium that was released was of particular concern because of its known carcinogenic or cancer-causing properties and association with lung cancer. Ultimately, it was decided that those who were exposed to the test had only a minimal chance of developing respiratory problems or lung cancer. As if that makes it better. And finally, there's Project 112 and Project SHAD. In 2002, the U.S. military started reaching out to veterans who may have been involved in Project 112 or Project SHAD. Both operations involved exposing personnel to substances chosen to act as stand-ins for biological weapons, allowing researchers to see how they spread. Project 112 was done at the Deseret Test Center in Utah in a series of tests from 1962 to 73, and Project SHAD, the shipboard hazard and defense, was done aboard warships in waters around the world. The details of the project might have remained under wraps for much longer than it did if it wasn't for the Department of Veteran Affairs requesting an investigation into the operations, as well as information on whether or not service members' health may have been compromised. Nearly 6,000 people, both military personnel and Department of Defense civilian staff, were exposed to the tests with and without knowledge. When information about the tests was released, it was thought that the agents released were harmless. However, it was also stressed that veterans with concern should come forward and that it was acceptable to reveal information about dates, places, and possible side effects with healthcare providers. There's a long list of biological agents that the test subjects were exposed to, including Q fever and staphylococcal enterotoxin B, which causes food poisoning. They also used some pretty horrific nerve toxins, including sarin, now classified as a weapon of mass destruction, and soman, a clear, colorless liquid that can cause death in minutes. Both can be fatal if only the tiniest amount gets on the skin. These same psychopathic, sociopathic personalities in the Defense Department and throughout the corporate defense contracting world believe that to protect ourselves from bioweapons, we may have to reinvent science itself. This from the Defense One website. Getting far better at predicting what research will produce may be the only way to save the world, by Patrick Tucker. This statement, of course, is the complete opposite of the scientific method. In June 2012, a team of researchers from the University of Wisconsin published a paper in the journal Nature about airborne transmission of H5N1 influenza or bird flu in ferrets. The article changed the way the United States and nations around the world approached man-made biological threats. This was not the researchers' intent. The team had altered the virus's amino acid profile, allowing it to reproduce in mammal lungs, which are a bit colder than bird lungs. That small change allowed the virus to be transmitted via coughing and sneezing, and it solved the riddle of how H5N1 could become airborne in humans. Why do the research on how to make the world's most dangerous viruses and bugs more lethal? The answer varies tremendously depending on who is asking and for what purpose the research is taking place. While experts differ on their views on how and where such work should be done, there is wide agreement that the barriers to entry for new biological creations, including ones that could kill millions of people, are decreasing. Today, there is little international enforcement of limitations on bioweapons. For chemical materials like sarin gas, the Chemical Weapons Convention provides a treaty-based legal framework for stopping proliferation, and a watchdog group, the Organization for the Prohibition of Chemical Weapons, to investigate potential violations. No similar watchdog exists around biological weapons. All but a few countries have ratified the Biological and Toxic Weapons Convention. For instance, among the no-shows is Syria, where President Bashar al-Assad's regime is suspected of harboring strains of smallpox for research. In light of this, it is indeed Assad's biological weapons complex that poses a far greater threat than his chemical weapons complex, wrote bioweapons expert Jill Bellamy. Some of the biological research that the Assad regime was conducting was based in Homs, putting smallpox samples or possibly rudimentary bioweapons within reach of the Islamic State, or ISIS. Defense One also released this article entitled Dangerous Research, Unknown Unknowns. The genetic engineering of deadly pathogens is not the sort of thing that a terrorist or would-be supervillain could easily attempt in a kitchen. But the quickening pace of genetics research has plenty of scientists worried. Suzanne Fry, director of the Strategic Futures Group at the Office for the Director of National Intelligence, told a group at last month's SXSW Technology Conference in Austin, Texas, that synthetic biology was a big concern among many of the technologists she's been interviewing recently. Some very, very prominent scientists have said that worries them very much, she said. George Church, a Harvard Medical School researcher, widely considered a father of modern genetic research, remembering that genetics is code word for eugenics, offered a somber assessment of the future of genetically engineered bioterror. How would we have calculated the odds of the events on 9/11/2001 on 9/10/2001, he said via email, or the Tokyo subway attacks. Hopefully, before anything happens, the good guys will get better at new pathogen detection and immunity soon, both to prevent this scenario and naturally emerging infectious diseases. There's a difference between building better detection kits and figuring out how to engineer new and more lethal versions of familiar viruses. But these areas of exploration are not always distinct. The best and most reliable Ebola detection devices work off of the virus's unique genetic signature, giving a level of certainty unmatched by fever scanners and other symptom trackers. A new Ebola-like pathogen with a unique genetic signature might be undetectable to the most up-to-date devices and methods. And yet that's what they're creating in gain-of-function research studies. The H5N1 case shows how a once difficult challenge is becoming exponentially easier because published results move so quickly in the age of digital interconnection. According to Graymon Bennett, an Arizona State University religious studies professor and biotechnology expert who has written extensively on synthetic biology. It took specialized facilities and millions of dollars for University of Wisconsin researchers to figure out how to create the amino acid sequence that would allow the virus to reproduce in mammal lungs, he said. But once you publish the sequences, once they've done that work, it would take a competent physician a few $1,000s and a few weeks to reproduce the result. Richard Danzig, a former Navy secretary, says that he understands the impetus towards moratoriums. He points to the years following the Manhattan Project, when research was classified and researchers monitored, as an example of a research control regime that worked. But today, the Internet makes implementing a similar control regime much harder, Danzig said at a 2013 Atlantic Council event. Our capabilities to control information seem inadequate, so that the distortive effects of those kind of controls tend to outweigh, I think, the positive effects, he said. It turns out that the information leaks in 100 ways, or is independently recreated in a global world outside the reach of your jurisdiction. In other words, the sharing of information cannot be regulated by a nation's law. For the military, the reasons to conduct gain-of-function research may outweigh reasons to suppress it. Military leaders are fond of saying that they don't want to ever find themselves in a fair fight. They always want the advantage. In the context of biological threats, that means understanding how to weaponize Ebola even if international laws and treaties like the Geneva Convention prohibit the use of such weapons in the field. Of course, understanding is different from actually developing a weaponized Ebola strain, which is illegal under the Biological Weapons Convention. Last year, the Pentagon's Defense Advanced Projects Research Agency, or DARPA, opened a biological technologies office to explore issues such as synthetic biology and epidemiology. Its work includes everything from creating new high-energy density materials from organic matter to exploring how diseases spread and become more dangerous. The DARPA director recently said she created the office because of the rising potency of biotechnology powered by information technology. When I say I see the seeds of technological surprise in that area, that's exactly what I mean. Hugely powerful technologies are bubbling out of that research. She said the agency constantly considers the political and scientific controversies surrounding biotechnology research, but that it isn't in the interests of DARPA or the nation to hold back because of the controversy. We are responsible to be in those areas, but we're also responsible for raising those issues and convening that dialogue. What we've done that's been very helpful is tap experts in different areas. We have experts in neuroethics, other experts who are very smart in synthetic biology and law, policy and societal issues. But DARPA is not going to make up answers to these societal choices. That's a much broader undertaking. The cheapest and most effective way to do that research is to open it up to more scientists and better publication opportunities. If the U.S. doesn't, someone else might. It's a huge dilemma for our military, said Gary Marchant, professor of emerging technologies, law, and ethics at Arizona State University. He spoke at New America Foundation's Future of War conference in February. Defense One is a media partner of the foundation's. He said, "We know that there's going to be people out there who won't follow those same self-imposed restrictions. To understand what those threats are and be able to counter them, do we need to make the monster ourselves? And when we do this stuff, do we publish it? The problem of how to control bioterror information in the information age is complex and important enough to cause a shift in science itself, according to some members of the research and university communities. At the very least, it's enough to prompt a change in the way in which some scientific research happens. In other words, there can only be one change to legitimate science, and that is to delegitimize it, to stop requiring the scientific method and its tenets. There is no other way to look at that statement. Again, our capabilities to control information seem inadequate, so that the distortive effects of those kinds of controls tend to outweigh, I think, the positive effects. When we go over to DARPA's website, the Defense Advanced Research Projects Agency, we find this article, Bolstering the Front Line of Biological Warfare Response. DARPA advances sensitivity and durability of antibody-based biosensors to better protect warfighters. Technologies to transition to DoD's Critical Reagents Program. This in February of 2013. Also, from the DARPA website, we find the Biological Technologies Office. And Gizmodo reports in 2014, DARPA's new biotech division wants to create a transhuman future. From Transhumanist News in 2019, we read, Thought-Controlled Weapons. DARPA funds 6 organizations in a bid to develop brain-machine interfaces for soldiers. One of the last things anyone should want is a machine-brain interface, even if it is a non-invasive type that doesn't require brain surgery. However, the U.S. military recently announced that it was throwing money at 6 organizations to come up with a system that lets its user control equipment with a thought. The Defense Advanced Research Projects Agency, or DARPA, wants American soldiers to have the ability to use their equipment with their minds. From the Dawn of the Giants website, following transhumanism and the technological singularity, in March 2015, we read DARPA and transhumanism. Biology is technology. Now remember that statement, biology is technology. This absolute fallacious statement is very, very important. DARPA is back in the game with a big vision and its Human Plus. DARPA, the world's most technologically advanced organization, is pursuing transhumanism technologies and supporting the transhumanism singularity movement. Just a thought to keep in mind while reading this: DARPA doesn't do science fiction. Again, biology is technology. Nothing could be farther from the truth. DARPA is back in the game with a big vision, and it is Human Plus. H plus is a transhumanism moniker representing the forced evolution of humans into transhumans, or what is called Futurism. DARPA, the Defense Research Projects Agency, is perhaps best known for its role as progenitors of the computer networking and the Internet. Formed in the wake of the Soviet Union's surprise launch of Sputnik, DARPA's objective was to ensure that the United States would avoid technological surprises in the future. This role was later expanded to causing technological surprises as well. The Times of Israel recently reported in August 5th of 2020 a very disturbing look at the company that Tony Fauci and the NIH is supporting for the new COVID-19 vaccine. The article entitled Gene Editing, Moderna, and Transhumanism. As gene-based mRNA vaccines from Moderna are being designed and tested at warp speeds to fight COVID-19, this is also bringing the debate over transhumanism into the forefront. Transhumanism is a type of futurist philosophy aimed at transforming the human species by means of biotechnologies. Transhumanists see disease, aging, and death as undesirable and unnecessary, and aim to transform human beings into post-human species with greater capacities than those of present human beings. The philosophy is based on secular humanism and sees human nature as an evolutionary work in progress, with room for improvement and enhancement. However, it is more radical in that it promotes not only traditional means of improving human nature, such as education and cultural refinement, but also direct application of medicine and technology to overcome basic biological limits. When technology and medicine are used in their reverse capacity, meaning to allow viruses and other pathogens to actually infect and reprogram the DNA of human beings, you know there's a big problem. Transhumanists give special attention to genetic engineering, robotics, molecular nanotechnology, and artificial intelligence. And the COVID-19 pandemic is providing gene-based vaccines a chance to break through into the global health market. Moderna and gene editing. Currently, there are various companies such as Xenovio, Moderna, and CanSino Biologics that are testing mRNA and DNA vaccines to counter SARS coronavirus 2, which causes COVID-19. But Moderna is the frontrunner that recently nabbed $472 million from U.S. government's Biomedical Advanced Research and Development Authority, or BARDA, to develop the vaccine. This is in addition to the $483 million it had already received back in April, bringing its total funding to $955 million. With U.S. government funding at nearly $1 billion for one company, Moderna may be too big to fail. However, this is perplexing for a company that has never produced a single vaccine. According to a CNN report, Moderna was only established in 2010, has never brought a product to market, nor gotten any of its 9 or so vaccine candidates approved for use by the FDA. However, it has been a long-term Pentagon contractor for biodefense, working closely with Defense Advanced Research Project Agency, or DARPA, on gene editing and mRNA therapeutics. DARPA is focused on developing emerging disruptive technologies to maintain a competitive edge over adversaries, including many transhuman projects such as genetic engineering and soldier enhancement via robotics. In the case of Moderna and mRNA therapeutics, DNA vaccines is considered a new paradigm that would disrupt the pharmaceutical industry. Its vision is to harness a new technology that synthesizes messenger RNA, So synthetic RNA or mRNA, which is an instruction manual in every living cell for creating protein to prompt the human body to make its own medicine. So instead of injecting a piece of virus into a person to stimulate the immune system, the synthesized genes would be shot into the body, whereby the genes are edited, deleted, added to re-engineer human DNA to resist the disease. If successful, scientists hope DNA vaccines could be a transformative treatment for heart disease, metabolic and genetic diseases, kidney failure, and even cancer. Moreover, it could be an effective form of biodefense to protect the population against biological warfare, which is also the mandate for DARPA and BARDA. Transhumanism and hybrids. Indeed, DARPA is also developing other forms of human enhancement in addition to gene editing. Already, scientists are merging robotics with the human body in brain-to-computer interface, or BCI, wherein individuals with physical injuries can regain their functions, and soldiers become smarter and more powerful through the fusing of their brain with machines. It should be stated here that this is very much like pharmaceutical drugs. Often, when you take one drug, 3 more drugs are needed to battle the side effects of the first, and so on and so forth. In comparison, the more synthetic biology and environments you place into your own body, the more dependent you're going to be on the technology and biological transhumanist interfaces being invented. The addiction of tomorrow will be on synthetic biology and machine interface. In a way, the Pentagon is now building a real Iron Man similar to the American superhero based on the Marvel Comics character. Soldiers in exoskeleton suits are physically more powerful than those without, while other soldiers with bionic limbs perform better than adversaries with human limbs. When one adds artificial intelligence with BCI, the sky is the limit for an army of these genetically modified and robotically enhanced humanoids. Take away empathy, morals, values, and ethics, and you've got a super soldier willing to do anything for his handlers. But United States is not the only country engaged in human enhancement and transhumanism, as Russia and China are also in hot pursuit with exoskeletons, vaccines, and brain implants. As the competition gains traction, one wonders what the future of their militaries may look like as human beings are steadily integrated with machines to become armies of iron men. Here, the book of Daniel may lend some insights. In interpreting King Nebuchadnezzar's dream of an image with a head of gold, chest and arms of silver, a belly and thighs of bronze, legs of iron, and feet of iron and clay, Daniel revealed the parts as the sequence of world empires, with the feet of iron and clay being the last. In Daniel 2:43 it is written, "As you saw iron mixed with ceramic clay, they will mingle with the seed of men. But they will not adhere to one another, just as iron does not mix with clay." That seems to describe a hybrid of man, or flesh, the metaphor of clay, mixed with machine, or iron. And as transhumanism and biotech gain momentum, armies of hybrid humans of iron and clay may be a real possibility in a not-too-distant future world. That is, after all, what DARPA and other corporate structures say they're planning.
Just a minute to put a little bit things in perspective, because we're talking about H5N1, highly pathogenic, transmissible between ferrets. In my opinion, there's more dangerous viruses out there, and they are contained and they are worked with every day, and they are contained and they are worked with every day. There's more dangerous viruses out there, and they are contained and they are worked with every day, and a lot of the speculation that is happening right now is not happening for those other viruses. Not to say they will never happen, but what I'm saying is let's keep this a little bit rational. Again, that it's maybe again a matter of debate, but there is a lot of more aggressive and more dangerous viruses. out there than this one.
Okay, the question, please.
Hi, I'm Edward Hammond. I'm a policy advisor to Third World Network, which is an NGO with offices in Malaysia, Ghana, Uruguay, and Geneva, and former U.S. lab biosafety gadfly. I wasn't going to say anything, but I can't let that question 1D go by without a specific consideration vis-a-vis the PIP framework at WHO. Because if we had a meeting like this a few years ago, the question that would have been on the table would not have been, are we going to do gain-of-function experiments on virus isolates? It would have been, are we going to acquire the virus isolates at all? Because the problem at that point was that a number of developing countries, not just one, were not providing virus isolates to the international community because, or to the WHO system in particular, because it was felt that there was a lack of sharing of information and practical benefits from research that was conducted on them in the North or in the developed countries. And if in proceeding with this framework, we begin to undertake a large amount of research that a large number of people feel should be restricted somehow, either by classification or by some other bizarre artifice to try to keep it within a certain closed group of people. What I think a very real risk there is that you're going to create another situation that parallels the one that led to the crisis that we had several years ago, where there was a breakdown in international cooperation because it was not felt that there was adequate information flow and equity in the relationship between developed countries and developing countries that had endemic virus problems. So I think that it merits very serious consideration looking into the future on this issue in particular, thinking about what will the effect be on the countries where this virus is endemic in terms of their willingness to play ball with this group of researchers writ large, if they feel that they're being dealt out of the results that come from this research. And, you don't want to have government lurch from almost fixing that last problem. And we may, within a matter of months, have new money from the private sector going into the PIP framework to support surveillance. Isn't that what we want? But we don't want to undermine the PIP framework then by creating this world of information that will not be seen as being accessible by countries whose cooperation is needed, not only by us, but the whole world needs that cooperation.
Any comments? Yeah, please.
That's a really good point, and that's why this notion of common but differentiated capabilities as a fundamental framework for a global governance structure is really important, because that it leads to a way to incentivize countries to participate with a recognition of differentiated capabilities, and in some sense be able to support those capabilities, or maybe put a governance structure in that says you can't work on this for various reasons, but have it in an incentivized governance structure that's agreed upon by all parties.
Okay, we have 2 questions, please.
Yes, Peter Hale, Foundation for Vaccine Research. This session obviously is about risk, and it's very difficult to assess the risks if people don't have all of the facts. For example, we have had extreme difficulty accessing statistics that would tell us how many accidents or incidents there have been in labs in the last 5 years where a lab worker has become infected. So my question, I think this is for Dr. Woolley and maybe Dr. Kapua, is what reporting mechanisms are there in place that would answer that question and identify what kind of incidents, whether they were major incidents or minor incidents? And why isn't that, if I'm assuming there's a reporting mechanism in place in this country, but why isn't the data more readily accessible?
on lab-acquired infections. The ones that we know about are just, the tip of the iceberg. The ones that we know about are just, the tip of the iceberg. Some of the lab-acquired infections are just not detected. Some of the lab-acquired infections are just not detected, just not detected, just not detected. They happen and it didn't cause a lethal disease and, literally not detectable. Sometimes some of the lab-acquired infections just mimic other common infections and they're not pursued. Some of the lab-acquired infections just mimic other common infections and they're not pursued. They're not pursued. There's a lot of psychological reasons why people may not want to report something. People may not want to report something. We could probably use a psychologist on the panel to delve into that. I mean, there's the denial factor. There's the denial factor. There are people who have even recently died of lab-acquired infections who were experts on that agent and failed to recognize the symptoms. There are people who have even recently died of lab-acquired infections who were experts on that agent, died of lab-acquired infections who were experts on that agent and failed to recognize the symptoms and act quickly, and they could have been saved if they had. We're working right now on the new biological safety textbook, the American Society of Microbiology, and we do have someone trying to accumulate data. But a lot of it is anecdotal. There's no real formal reporting mechanism. There's no real formal reporting mechanism, especially at universities. If you get a needle stick, sure, you're supposed to report that. But there are a lot of accidents and things that happen that just simply go unreported and undetected. There are a lot of accidents and things that happen that just simply go unreported and undetected. Unreported and undetected. Unreported and undetected.
It be to put a reporting mechanism in place and make it mandatory, at least for BSL-4 labs if you're working with tier 1 agents?
Yeah, even in my 5-minute summary, I said absolutely, if this work is done, it has to be the most rigorous surveillance possible. But with this particular virus, again, the symptoms being very vague, I think it's going to be a challenge, for the workers. But I think the training has to be of the utmost in terms of, you would think that if they're working on the virus, they're experts on it, they should know the symptoms. But again, this is a very vague list of symptoms. We all know the list is huge for influenza, and it mimics so many other things. And so then you've got the challenge of, people just coming down with a normal cold and overreacting, and how many things get reported and how many people go in isolation. Just coming down with a normal cold and overreacting, and how many things get reported and how many people go in isolation. But in terms of the biosafety level 4, it is the highest level that we have, and I think the reporting is the most rigorous in that level of containment, and that's why I would suggest if it's done, that's what we would use. An accident, certainly in an environment like that, would have a much higher likelihood of being reported to the appropriate authorities. Because there are cases where someone has just been in a laboratory, there was no known accident, and they acquired an infection. Now, I'm not talking about just biosafety level 4, I'm talking about, in general, biosafety labs.
So we're eager to get to the bottom of, to answer the question, what is the incidence of accidents in laboratories? How often does it happen? We've heard numbers like in the last 20-year period from 1978 to 1999 that there were 1,200 incidents in BSL-4 labs in which a lab worker was infected. There were 1,200 incidents in BSL-4 labs in which a lab worker was infected. We've not been able to get any data since that, and we still don't know who did that study. It's awfully important to know. If we use those particular numbers, it suggests that there are between 50 and 60 incidents a year in BSL-4 labs where a lab worker is infected.
I think Tony had a question.
In the discussions of the NSABB, when you talk about funding, obviously there are biosafety right up there, biosecurity, things that you take into considerations. And when we fund a group, as I emphasized in my presentation, like Ron's and like Yoshi's and others who are well-trained, well-regulated, experienced, et cetera, et cetera. True, there is the possibility, as we mentioned, that there may be an accident, but that's a group that's very, very highly capable of conducting the research. The situation that was brought up in one of the discussions is that you're not worried that much about them, and it isn't who we fund that people are concerned about, but it's the people who will then use the research from the people we funded. So the real fundamental question is, when we make a funding decision, do we have to make a decision based on funding even the most careless, most unregulated, because they're going to use the information? That sets an extraordinarily high bar for what you're going to fund or what you're not going to fund, because you're going to say if people are going to use this information, they may use it with no biosafety or biosecurity concerns. So I'd like to hear sometime during the next couple of days where that consideration fits in our funding decision. I mean, it's very, I wouldn't say easy, but it's clear when you're funding people who have a very good track record, who are very well trained. But do we need to take into consideration beyond that? Not who we are funding, but who's going to use the research information that we're funding. That makes a decision very complicated.
Yeah, I just wanted to add that, to bring us back to where we started from, the problem is H5N1. I mean, we're all doing these experiments because we are afraid that a virus which is endemic in many countries may spark off a pandemic. So it doesn't really make sense to start working on a virus which is, a one-off in a wild bird isolate, and that doesn't have those very peculiar characteristics that H5N1 has, because we don't have to forget that H5N1 is a truly unique set of viruses, because no other influenza virus of high or of low pathogenicity has contained all the peculiarities, particularly relating to animal health and to human health, that H5N1 has. So I really wouldn't see a point. I mean, it would take us, I don't know, like 20 years to get the model right, and we may all be dead by then. Then we may all be dead by then. Then we may all be dead by then.
I do agree that we really need to more clearly delineate the benefits. Just based on what I've heard and read, I'm not completely convinced that the benefits outweigh the risks. I think that basic science information is generally useful in the long term, but we have to get to the long term. We have to live to get there. And in the short term, the worst case scenario we have to plan for, we are not ready to deal with it at the current time. If the worst thing happens, we're not prepared.
While this panel discussion did continue for a few more sessions, enough of the bulk of the risks and benefits have been spoken. And so, in the end, we focus on what happened after these debates, after these so-called public panel discussions on whether or not to continue with gain-of-function research. It's important to note here the many viruses that have been altered through gain-of-function research, if for no other reason so that you might know what to expect in the future when accidents or biological terror events happen. For instance, the journal Nature reported in January of 2006, the risks of resurrecting the 1918 flu virus outweigh the benefits. I would certainly agree. It would be hard to disagree with that assessment. Shortly thereafter, the American Society for Microbiology reported on the reconstruction of the 1918 influenza virus, unexpected rewards from the past. While this whole event was covered in my first documentary, the video part of it, let's read a little bit about what happened. The abstract reads that the influenza pandemic of 1918 to 1919 killed approximately 50 million people. The unusually severe morbidity and mortality associated with the pandemic spurred physicians and scientists to isolate the etiologic agent. But the virus was not isolated in 1918. Only in 1996 did it become possible to recover and sequence highly degraded fragments of influenza viral RNA retained in preserved tissues from several 1918 victims. Again, video evidence shows that they dug up these Inuit Eskimo women to extract their lungs and the virus within. These viral RNA sequences eventually permitted reconstruction of the complete 1918 virus, which has yielded, almost a century after the deaths of its victims, novel insights into influenza virus biology and pathogenesis, and has provided important information about how to prevent and control future pandemics. The Spanish influenza pandemic of 1918 to 1919 stands as the deadliest single event in recorded human history, killing approximately 50 million people worldwide. The unprecedented burden of morbidity and mortality frustrated physicians and scientists who were unable to identify the etiological agent, but also spurred advances in microbiology, clinical infectious diseases, and public health practice. Influenza viruses were finally isolated about 15 years after the pandemic, but scientists of the early 20th century were not then capable of studying the biological basis of pandemic viral emergence, pathogenicity, or disappearance, let alone of gaining insights that might help to prevent or mitigate future pandemics. The cause of the 1918 pandemic and the determinants of its severity remained one of the most discussed medical mysteries throughout most of the 20th century. The American Society for Microbiology also reported on the re-emergent 1977 H1N1 strain and the gain-of-function debate. The abstract reads that the 1977-1978 influenza epidemic was probably not a natural event. Again, probably not a natural event, as the genetic sequence of the virus was nearly identical to the sequences of decades-old strains. And this is an interesting point, that if a virus from 1918 comes along and expresses the same exact genetic traits, added to the fact that influenza is known to mutate to the point of being completely unpredictable within less than 1 year, this is yet another sign of its man-made origins. While there are several hypotheses that could explain its origin, the possibility that the 1977 epidemic resulted from a laboratory accident has recently gained popularity in discussions about the biosafety risks of gain-of-function or GOF influenza virus research as an argument for why this research should not be performed. There is now, or was, a moratorium in the United States on funding GOF gain-of-function research, while the benefits and risks, including the potential for accident, are analyzed. Given the importance of this historical epidemic to ongoing policy debates, we revisit the evidence that the 1977 epidemic was not natural and examine 3 potential origins: a laboratory accident, a live vaccine trial escape, or deliberate release as a biological weapon. Based on available evidence, the 1977 strain was indeed too closely matched to decades-old strains to likely be a natural occurrence. While the origin of the outbreak cannot be conclusively determined without additional evidence, there are very plausible alternatives to the laboratory accident hypothesis, diminishing the relevance of the 1977 experience to the modern gain-of-function debate. And finally, we see in The Lancet, the Reflection and Reaction section, a comment on the recent study reporting the recreation of the 1918 influenza paleovirus. Beyond this scientific tour de force, these experiments raise huge ethical concerns. The medical justifications for such an exercise are open to debate. First, it is not certain that a better understanding of the virus's pathogenesis in a mouse model would be helpful for treating influenza infections in human beings. Second, the predicted efficacy of neuraminidase inhibitors and immunogenicity of H1N1-derived vaccines have already been demonstrated in recombinant influenza A viruses bearing the appropriate H1 and N1 genes. Third, to have a pandemic flu vaccine available on the market in the shortest possible time, the European Agency for the Evaluation of Medicinal Products encourages manufacturers to work on a core dossier using a mock-up strain. However, because of the danger that the virulent 1918 flu strain presents, the strain will never be distributed worldwide for such purposes anyway. By contrast, the risks of such an exercise are real. First, it is astonishing that this virus has been manipulated in a biosafety level or BSL-3 laboratory and not a higher level of containment. Severe acute respiratory syndrome virus escapes from BSL-3 labs were reported. Again, SARS, severe acute respiratory syndrome virus escapes from BSL-3 labs were reported. In addition, the H2N2 1957 pandemic strain was accidentally sent to 3,700 reference laboratories in 2005. The 1918 flu virus poses a high risk of life-threatening disease with high-spreading aerosol transmission and without enough therapeutic stocks of antivirals available for mass treatment, and thus should have been classified as a BSL-4 agent and even contained in a special high-security facility, as in the case of variola, that is, smallpox virus, until its expected destruction. Again, to be clear, there are not enough therapeutic stocks of antivirals available for mass treatment of the very gain-of-function dual-use research projects, that is, viruses and other pathogens, that may be coming to a town near you. Now, shortly after this panel discussion in 2012, the CIDRAP, the Centers for Infectious Disease Research and Policy at the University of Minnesota, reported in January 2013 that the research moratorium on modified H1N1 viruses ends. A year-long voluntary moratorium involving transmissible H5N1 avian flu viruses ended today with a letter from a group of so-called scientists that supports resuming the work in countries that have addressed the biosafety issues involved. Today's letter, endorsed by 40 scientists who signed on to the voluntary moratorium last year, appeared in the journals Science and Nature. The research pause was designed to allow countries and the so-called scientific community to discuss biosecurity and biosafety issues that were raised by the publication of two controversial H5N1 papers, one from a group in the Netherlands led by Ron Fauchier, Ph.D., and the other from a team at the University of Wisconsin led by Yoshihiro Kawaoka, DVM, Ph.D. The scientists wrote that the moratorium allowed the community to explain the public health benefits of the transmission studies, to describe the systems that are in place and to protect the researchers and the public, and to allow organizations and governments to review their policies regarding the experiments. Quote, thus acknowledging that the aims of the voluntary moratorium have been met in some countries and are close to being met in others, we declare an end to the voluntary moratorium on avian flu transmission studies, they wrote. The moratorium was originally planned for 60 days. Three of the researchers who signed the letter, Fauchier, Kawaoka, and Richard Webby, Ph.D., from St. Jude Children's Research Hospital in Memphis, spoke about the end of the research moratorium today at a media telebriefing sponsored by the American Association for the Advancement of Science, the publisher of the journal Science. Kawaoka said the research work needs to resume because it has important public health benefits, such as providing more clues about what it would take for the H5N1 virus to become more transmissible in mammals. We understand the risk and we take every precaution, he said. The benefit outweighs the risks. That is why we need to resume. Many countries have reviewed their oversight of H5N1 research issues, so the need for the moratorium has passed, said Webby. The researchers said the number of countries in which H5N1 transmission studies take place is limited, and it's not entirely clear which ones are ready to resume work. They said the list includes the Netherlands, European Union countries in principle, and China. Canada has already said H5N1 transmission work can take place under biosafety level 4 of BSL-4 conditions. And that lands us firmly in July of 2014, where The Independent reports the exclusive that controversial U.S. scientist creates deadly new flu strain for pandemic research. Scientists express horror over the creation of a virus that could render the human immune system defenseless. A controversial scientist who carried out provocative research on making influenza viruses more infectious has completed his most dangerous experiment to date by deliberately creating a pandemic strain of flu that can evade the human immune system. Yoshihiro Kawaoka of the University of Wisconsin-Madison has genetically manipulated the 2009 strain of pandemic flu in order for it to escape the control of the immune system's neutralizing antibodies, effectively making the human population defenseless against its reemergence. Most of the world today has developed some level of immunity to the 2009 pandemic flu virus, which means that it can now be treated as less dangerous seasonal flu. However, The Independent understands that Professor Kawaoka intentionally set out to see if it was possible to convert it to a pre-pandemic state in order to analyze the genetic changes involved. The professor has so far kept details of the research out of the public domain, but admitted today that the work is complete and ready for submission to a scientific journal, so that any bad actor in the world can get it, including all governments. The experiment was designed to monitor the changes to the 2009 H1N1 strain of virus that would enable it to escape immune protection in order to improve the design of course, vaccines, he said. Through selection of immune escape viruses in the laboratory under appropriate containment conditions, we were able to identify the key regions that would enable 2009 H1N1 viruses to escape immunity, he said in an email. Viruses in clinical isolates have been identified that have these same changes in the viral protein. This shows that escape viruses emerge in nature, and laboratory studies like ours have relevance to what occurs in nature. So, to be clear, the excuse of vaccine research is being used on a virus that was specifically created to be immune from any possible antibodies the human body can make. This vaccine research excuse is wearing a little thin. The report goes on to ask what has been done previously in this laboratory. Professor Kawaoka of the University of Wisconsin-Madison attempted to increase the transmissibility of the H5N1 bird flu strain by genetic manipulation and repeated infection in laboratory ferrets, an animal model of human influenza. H5N1 is highly lethal when it infects people, but in the wild it is very difficult to transmit from one person to another and is usually caught by direct contact with infected poultry. Professor Kawaoka's most recent published research was on reconstructing the 1918 flu virus, the genetic structure which was known from samples retrieved from the frozen corpses of its victims buried in the Arctic from wild strains of bird flu isolated from ducks. He managed to do this, sure, but the study was widely criticized as stupid and irresponsible. Again, the interviews and video footage of this are available in Lethal Injection, The Story of Vaccination, my first vaccine documentary. Have there been any accidental releases from labs in the past? Some experts cite the unexpected emergence of a new H1N1 strain of flu in 1977, which spread globally over 3 decades, as an early example of a flu virus being accidentally released from a lab. Genetic evidence points to it having escaped from a lab in China or the Soviet Union. There are many examples of other infectious agents escaping from labs. Smallpox virus escaped from Birmingham Medical School in 1978 and killed a medical photographer, Janet Parker, the last person to die of smallpox. Foot and mouth virus escaped in 2007 from a veterinary lab in Surrey, and in 2004 the SARS virus escaped from a high-containment lab in Beijing, infecting 9 people before it was stopped. Again, it is acknowledged that in 2004 SARS escaped from a high-containment lab in China. Also in June 2014, American scientists controversially recreate the deadly Spanish flu virus. There are countless reports out there that we could go over, but the point has been made. And this scary folly of gain-of-function research projects brings us into October of 2014, where The Scientist reports that a moratorium on gain-of-function research is now put in place. In the wake of a handful of biosafety lapses at federal research facilities, the U.S. government is temporarily halting funding for new studies aiming to give novel functions to, you guessed it, influenza, SARS, and MERS viruses. The White House Office of Science and Technology Policy and Department of Health and Human Services last week, that is October 17, 2014, announced it was launching a detailed review into so-called gain-of-function research, in which pathogens are manipulated to alter their capabilities. Such research made headlines in 2012 after two groups instilled the avian influenza virus, H5N1, with the ability to transmit between ferrets through the air, a feat that prompted a year-long moratorium on H5N1 research. You know, the voluntary moratorium in which the gain-of-function research scientists decided no longer was needed? Now, in the face of threats like influenza, SARS, and MERS, which have killed scores in the Middle East and Asia, the government is instituting a pause to gain-of-function funding for experiments involving these deadly viruses. Now, it is what happened between this 2014 forced pause, or moratorium, on gain-of-function research in October of 2014, and this, in December of 2017, the last few remaining weeks of the Obama administration. CIDRAP again reports that feds lift the gain-of-function research pause and offer guidance. The National Institutes of Health today, the NIH, lifted a 3-year moratorium on funding gain-of-function, or GOF, research on potential pandemic viruses such as avian flu, SARS, and MERS, opening the door for certain types of research to resume. The action coincides with today's release of a U.S. Department of Health and Human Services framework for guiding funding decisions about proposed research involving pathogens that have enhanced potential for creating pandemics. Experts involved in the discussions welcomed the development, but some said the new framework still leaves key unanswered questions, such as how to responsibly report findings from the funded lab work in medical journals. So, in other words, the very issue that prompted the initial moratorium, which was the fact that these studies are being published so that anybody in the world with the right equipment, including the do-it-yourself scientist in a garage, can reproduce these viruses from the published literature. This was never solved. Meanwhile, in research labs, some scientists plan to resume experiments and are relieved the pause has ended. Both groups are eager to see how the new review process works in real life. In a statement today, NIH Director Francis Collins, MD, PhD, said, "We have a responsibility to ensure that research with infectious agents is conducted responsibly and that we consider the potential biosafety and biosecurity risks associated with such research." He added that he is confident that the review process spelled out in the new framework will help to facilitate the safe, secure, and responsible conduct of this type of research in a manner that maximizes the benefits to public health, blah, blah, blah. Scientists gear up to resume studies. Research projects that were paused under the moratorium will now be reviewed based on the new framework, and the ones that clear the process will be able to proceed with appropriate risk mitigation measures in place. Yoshihiro Kawaoka, who heads a virology research group at the University of Wisconsin-Madison, said his team will propose experiments they couldn't do during the funding pause. He was lead author of one of the two controversial H5N1 avian flu papers published in 2012 that brought the gain-of-function controversy to a head. Specifically, we have been identifying amino acid changes that enhance the polymerase function of avian influenza viruses in mammalian cells to understand the molecular mechanism of avian to human transmission, in a system not involving live virus. Kawaoka said, "We will now propose testing the effect of those amino acid changes on virus replication." Gain-of-function research involves studies that enhance the pathogenicity, transmissibility, or host range of a pathogen to better understand the threat. However, gain-of-function studies have sparked dual-use concerns that center around the threat of accidental lab release or the pathogens becoming bioterror threats if they fall into the wrong hands. Or perhaps the right hands. The framework follows advisors' daunting task. In light of controversial research on H5N1 viruses in 2012, the Obama administration in 2014 announced a pause of federally funded gain-of-function research and asked a government advisory group to reevaluate federal gain-of-function funding policies and put together recommendations to help officials make their decisions. The expert group, called the National Science Advisory Board for Biosecurity, or NSABB, commissioned a 1,000-page risk-benefit assessment to help them make their final guidance, which they finalized in June 2016 along with an ethics white paper. As part of the process, the NSABB held 2 National Academies symposia on gain-of-function issues. Collins, in his statement today, thanked and praised the NSABB and the National Academies of Sciences, Engineering, and Medicine for their work on the issues. Of course, the depths of scientism have certainly worked their way into all of these so-called academies and institutions of arts and sciences. The work of these committees was instrumental in guiding the United States government in its job of creating rigorous policy that allows vital research to move forward. Tight framework centers on 8 criteria. The framework, condensed into a 6-page document, spells out a multidisciplinary review process that involved the funding agency and a department-level review group that considers the merits and possible research benefits and the potential to create, transfer, or use an enhanced potential pandemic pathogen, or PPP. In January, in the final days of the Obama administration, the White House Office of Science and Technology Policy released guidance the departments can use to follow through with the reviews. There are 8 criteria in the framework for guiding HHS funding decisions, which stipulate, for example, that the research has been evaluated by an independent expert review as scientifically sound, that the potential risks and benefits are justified, that there are no other equally effective but less risky options for answering the research question, and that the research team and facility have the capacity to do the work safely and securely, and to respond rapidly if there are any incidents, protocol lapses, or security breaches. The report seems to forget the absolute fallibility of the human condition. Regarding issues surrounding publication, the criterion says that the research results are expected to be responsibly communicated based on applicable laws, regulations, and policies, along with the terms and conditions of funding. Also, the framework stipulates that the work will be done with the support of funding mechanisms that allow appropriate risk management, an ongoing institution and federal oversight of the research. And finally, the criteria state that the research must be ethically justifiable, a status that is literally impossible to truly justify considering the risks. It is also important to note, as stated in the panel discussion, that under no circumstances does any country out there, does any scientist out there, nor any bad actor have to follow the rules of this framework. The bad guys simply do not give a crap about the framework. And quite frankly, neither do some of the countries that we've stepped on in the past. Steps forward, but unanswered questions. Mark Lipsitch, PhD professor of epidemiology and director of the Center for Communicable Disease Dynamics at the Harvard T.H. Chan School of Public Health, has been deeply involved in the gain-of-function discussions and has argued for a much more rigorous approach for evaluating the experiments. He has pushed for experts to consider safer ways to assess potential pandemic virus threats and for an international approach to tackling the issues. My overall take is that this is a small step forward, he said, adding that it includes a department-level review of the most concerning types of research, which have been defined appropriately after extensive time and debate. The question is how such reviews will play out in practice. Now again, the definition of these viruses is potentially pandemic pathogens, or PPP. However, Lipsitch said one problem is that the guidance specifies that research groups that propose work with enhanced pathogens will need to convince reviewers that there is no feasible, equally effective alternative way of addressing the scientific question with a less risky approach. If this means no alternative to answer with certainty the question of whether a specific strain that occurs in nature can very easily evolve to acquire a ferret transmissible phenotype, then this criterion will always be satisfied. This is a scientific question that can uniquely be answered with dangerous experiments, and cannot be answered safely. But it is not a very useful one because every strain in nature is, of course, different. In other words, the irony here is that in no way can unnatural occurrences be proven in any other way than to cause them to happen unscientifically in a laboratory setting. That is to say that the scientific method itself cannot show something that cannot happen in nature. It's quite a simple concept. And so this word trickery will always ensure that gain-of-function research can go forward. Why? Because without gain-of-function research, things that have not happened and likely will not happen in nature can only be proven through gain-of-function research. Michael Osterholm, PhD, MPH, who was a member of the NSABB during the controversy over the H5N1 papers, said he believes the gain-of-function work can be done safely, but he doesn't agree that scientists doing a federally funded work should be unfettered. In other words, a bunch of sociopaths should not be left alone to their own devices. Osterholm said that his main concern regards the public health implications of the publicly available details about how the work is communicated, which the new framework doesn't spell out. Again, the main reason the moratorium was called in the first place. How we detail that information needs to be considered, such as more finely specifying when findings are open to the general public, when they're disseminated on a need-to-know basis, and when that information is classified. If we consider that these horrible experiments are actually public, we should also shudder to think what's going on behind closed doors in classified laboratories. Until we have that part solved, I'm concerned about the work being done, he said. Osterholm, director of the University of Minnesota's Centers for Infectious Disease Research and Policy, which publishes CIDRAP News, added that some research is needed to answer key questions, such as what it would take for Ebola to become a respiratory virus, findings that would have implications for preparedness. If it were the case, I don't want the public to have a blueprint on how to do it, he said. Again here, the question is asked, what would it take for Ebola to become a respiratory virus? The answer is, it's not going to happen in nature. And so the response by the so-called scientific community is, well, let's gain-of-function the hell out of it and make it a respiratory virus and hope that we don't have an accident or that our published papers don't get picked up by a bad actor. Tom Inglesby, MD, Director and Chief Executive Officer at the Center for Health Security at Johns Hopkins Bloomberg School of Public Health, has also been deeply involved in discussions about issues related to the research pause. We saw him as one of the lone rational voices in that debate panel. He said the requirements for the multidisciplinary department-level pre-funding review and the involvement of the White House OSTP are excellent. He added that the emphasis on enhanced potential pandemic pathogens, or PPPs, is correct and focuses the framework on where it should be, such as on harmful consequences, immunity disruption, conferred resistance, and reconstructed extinct pathogens. Kind of reminds you of Jurassic Park and the lessons that should have been learned from that predictive programming movie. Though for the policy to be successful, it needs to, at a minimum, be able to oversee the creation of novel strains that may be highly transmissible and highly virulent, and should probably focus on that most intently to start. A potentially serious weakness of the new framework is that surveillance activities involving potential pandemic pathogens, or PPPs, including sampling and sequencing, aren't considered to be enhanced PPPs and would be exempt from reviews, Inglesby said, adding that surveillance as a rationale doesn't change potential risks of novel PPP strains. There are serious debates about whether specific enhanced PPP projects are materially useful to on-the-ground surveillance programs, he said. Inglesby's other concerns revolve around the lack of hard details of how the experiment reviews will work, such as the process for weighing the risks and benefits and the criteria for judging if researchers and institutions have the capacity to do the work safely. I would have liked to see if this framework was working as intended before the moratorium was ended, he said, suggesting that agencies funding the work publish their experiences using the new framework to show how it functions. The official announcement from the NIH director, posted on December 19, 2017, reads that the NIH lifts funding pause on gain-of-function research. Today, the National Institutes of Health announced that it is lifting a funding pause dating back to October 2014 on gain-of-function or GOF experiments involving influenza, SARS, and MERS vaccines. Gain-of-function research is important in helping us identify, understand, and develop strategies and effective countermeasures against rapidly evolving pathogens that pose a threat to public health. The funding pause was lifted in response to today's release of the Department of Health and Human Services framework for guiding funding decisions about proposed research involving enhanced potential pandemic pathogens. And you can download this report and read the worthless information inside for yourself. And if you're a bad guy, a bad actor, just toss it in the trash where it belongs. A flash forward 3 years to Tuesday, October 6th, 2020, and we find the most recent article in this documentary from Newsweek, where it states that Dr. Fauci backed controversial Wuhan lab with US dollars for risky coronavirus research. It is noted here that the headline of the story has been corrected to reflect that the Wuhan lab received only a part of the millions of US dollars allocated for virus research. So China's not the only one we're funding. Biomedical research ultimately protects public health, said Anthony Fauci in explaining his support for controversial research. Dr. Anthony Fauci is an advisor to President Donald Trump and something of an American folk hero for his steady, calm leadership during the pandemic crisis. At least one poll shows that Americans trust Fauci more than Trump on the coronavirus pandemic, and few scientists are portrayed on TV by none other than Brad Pitt, the Hollywood propaganda machine in action. But just last year, the National Institute for Allergy and Infectious Diseases, the organization led by Dr. Fauci, funded scientists at the Wuhan Institute of Virology and other institutions for work on gain-of-function research on bat coronaviruses. In 2019, with the backing of NIAID, the National Institutes of Health committed $3.7 million over 6 years for research that included some gain-of-function work. The program followed another $3.7 million 5-year project for collecting and studying bat coronaviruses, which ended in 2019, bringing the total to $7.4 million. In other words, it began in 2014, just before the official ban on gain-of-function studies was implemented in the United States. Many scientists have criticized gain-of-function research, which involves manipulating viruses in the lab to explore their potential for infecting humans, because it creates a risk of starting a pandemic from accidental release. SARS-CoV-2, the virus now causing a global pandemic, is believed to have originated in bats. U.S. intelligence, after originally asserting that the coronavirus had occurred naturally, conceded last month that the pandemic may have originated in a leak from the Wuhan lab. At this point, most scientists say it's possible, but not likely, that the pandemic virus was engineered or manipulated. Dr. Fauci did not respond to Newsweek's request for comment, and the NIH responded with a statement that said, in part, "Most emerging human viruses come from wildlife, and these represent a significant threat to public health and biosafety in the U.S. and globally, as demonstrated by the SARS epidemic of 2002 and 3, and the current COVID-19 pandemic. Scientific research indicates that there is no evidence that suggests the virus was created in a laboratory." That, the official statement from the NIH, which now you know is absolutely ridiculous. The NIH research consisted of two parts. The first part began in 2014 and involved surveillance of bat coronaviruses and had a budget of $3.7 million. The program funded Shizheng Li, a virologist at the Wuhan lab, and other researchers to investigate and catalog bat coronaviruses in the wild. This part of the project was completed in 2019. A second phase of the project beginning that year included additional surveillance work but also gain-of-function research for the purpose of understanding how bat coronaviruses could mutate to attack humans. Again, something that does not happen in nature. The project was run by EcoHealth Alliance, a nonprofit research group under the direction of President Peter Daszak, an expert on disease ecology. NIH canceled the project just this past Friday, April 24th, Politico reported. Daszak did not immediately respond to Newsweek requests for comment. The project proposal states, "We will use S-protein sequence data, infectious clone technology, in vitro and in vivo infection experiments, and analysis of receptor binding to test the hypothesis that percent divergence thresholds in S-protein sequences predict spillover potential." Now, when we examine this statement, we find these exact orthologs belong to SARS-2. Or COVID-19. It does use the S-protein sequence data, and through gain-of-function research, HIV spikes were added for receptor binding to the human ACE2 receptor, which happens to be located in the lungs, the heart, and all the places where COVID-19 is attacking. And this will be covered in the next volume of this documentary series, on the story behind the story of COVID-19. In layman's terms, spillover potential refers to the ability of a virus to jump from animals to humans, which requires that the virus be able to attach to receptors in the cells of humans. SARS-CoV-2, for instance, is adept at binding to the ACE2 receptor in human lungs and other organs. According to Richard E. Bright, an infectious disease expert at Rutgers University, the project description refers to experiments that would enhance the ability of bat coronavirus to infect human cells and laboratory animals using techniques of genetic engineering. In the wake of the pandemic, that is a noteworthy detail. Ebright, along with many other scientists, has been a vocal opponent of gain-of-function research because of the risk it presents of creating a pandemic through accidental release from a lab. Dr. Fauci is renowned for his work on the HIV/AIDS crisis in the 90s. Born in Brooklyn, he graduated first in his class from Cornell University Medical College in 1966, and that certainly makes him a smart person. As head of NIAID since 1984, he has served as an advisor to every U.S. president since Ronald Reagan. In essence, he's the head of the infectious agent mafia. A decade ago, during a controversy over gain-of-function research on bird flu viruses, Dr. Fauci played an important role in promoting the work. He argued that the research was worth the risk it entailed because it enables scientists to make preparations, such as investigating possible antiviral medications that could be useful if and when a pandemic occurred. Now, let's be very clear here that when Fauci and other so-called scientists suggest that the benefits outweigh the risk in gain-of-function research, that means that millions, if not billions, of lives being lost to an escaped or bioterrorist release of these weaponized viruses and other pathogens is worth the risk of doing the research. Millions, billions of lives, or complete extinction of the human race is an acceptable risk. Please understand that is what is being said. The work in question was a type of gain-of-function research that involved taking wild viruses and passing them through live animals until they mutate into a form that could pose a pandemic threat. So-called scientists used it to take a virus that was poorly transmitted among humans and make it into one that was highly transmissible, a hallmark of a pandemic virus. This work was done by infecting a series of ferrets, allowing the virus to mutate until a ferret that hadn't been deliberately infected contracted the disease. The work entailed risks that worried even seasoned researchers. More than 200 scientists called for the work to be halted. The problem, they said, is that it increased the likelihood that a pandemic would occur through a laboratory accident. Dr. Fauci again defended the work, saying, quote, determining the molecular Achilles heel of these viruses can allow scientists to identify novel antiviral drug targets that could be used to prevent infection in those at risk or to better treat those who become infected. Wrote Fauci and two co-authors in the Washington Post, The Same Old Nonsense, on December 30th, 2011. Decades of experience tells us that disseminating information gained through biomedical research to legitimate scientists and health officials provides a critical foundation for generating appropriate countermeasures and, ultimately, protecting the public health. To protect the public health, we must create viruses that could completely wipe out the public, if mishandled. Nevertheless, in 2014, under pressure from the Obama administration, the National Institutes of Health instituted a moratorium on the work, suspending 21 different studies. 3 years later, though, in December 2017, the NIH ended the moratorium and the second phase of the NIAID project, which included the gain-of-function research, began. The NIH established a framework for determining how the research would go forward, which we just read about, and so scientists have to get approval from a panel of so-called experts who would decide whether the risks were justified. And I'm sure the conflict of interests with those experts are equally as troubling. The reviews were indeed conducted, but in secret, for which the NIH has drawn criticism. In early 2019, after a reporter for Science magazine discovered that the NIH had approved 2 influenza research projects that used gain-of-function methods, scientists who oppose this kind of research excoriated the NIH in an editorial in the Washington Post. Quote, we have serious doubts about whether these experiments should be conducted at all, wrote Tom Inglesby of Johns Hopkins University and Mark Lipsitch of Harvard. Quote, with deliberations kept behind closed doors, none of us will have the opportunity to understand how the government arrived at these decisions or to judge the rigor and integrity of the process. So in essence, the process has become more secretive, less open to scrutiny by real scientists, and essentially out of the public purview. Switching over to the Gateway Pundit, who's been tracking this for some time, we have an updated article from May 5th, 2020, which states Dr. Fauci likely broke U.S. regulations and U.S. law when he funded the Wuhan lab to continue coronavirus projects that were banned in the United States in 2014. A month ago, we reported that Chinese doctor Shi Zhengli was part of a team working on a coronavirus project jointly with U.S. doctors in 2014 before it was shut down by the DHS for being too risky. After the U.S. research project was shut down, Dr. Shi continued her coronavirus research in Wuhan, China. Dr. Shi Zhengli from China was part of a team including Dr. Ralph S. Barrick from North Carolina that published an article in a 2015 edition of Nature Medicine. In the article, they discussed bat coronaviruses that showed potential for human emergence. The article was published in 2015. Now, we covered this earlier in the documentary, but it is well worth the read. After the work stopped in the U.S., the Chinese moved forward with the project and ran research and development in Wuhan at the Wuhan Virology Center. From Shi Zhengli's papers and resume, it is clear that they successfully isolated the coronavirus in the lab and were actively experimenting with species-to-species transmission. It's also important to note that back in 2017, we had solid intelligence about a viral leak in a high-security Chinese virology R&D center that resulted in the SARS virus getting out and killing people. This was, of course, since confirmed. This information provides a basis that contradicts the theory that COVID-19 is a variant that just magically mutated in a bat in the wild, and then jumped to a human when they ate a delicious bowl of bat soup. We then reported that the Wuhan Institute of Virology in Wuhan, which was very close to the seafood market blamed in the first place, an effort to hide the truth, Hubei province in China was hiring individuals for an ecological study of bat migration and virus transmission in December of 2019. We now know from Dr. Shi's resume and papers that she was still working on the coronavirus at the time. This report was published shortly after their project was defunded by the U.S. Department of Health and Human Services. The HHS in 2014 sent a letter to the University of North Carolina at Chapel Hill where they announced they were going to defund the program. Dr. Ralph S. Barrick was identified in the letter, which states, NIAID has determined that the above-referenced grant may include gain-of-function or GOF research that is subject to the recently announced U.S. government funding pause, issued October 17, 2014. The following specific aims appear to involve research covered under the pause. Listing Ralph S. Barrick, Ph.D., as project leader, Project 1 was the role of uncharacterized genes in high pathogenic human coronavirus infection. The letter was also sent to none other than Yoshihiro Kawaoka, project leader, in determining the functions of novel genes for influenza A and Ebola viruses, both in viral replication and in experiments increasing pathogenicity. As your grant is currently funded, this pause is voluntary. Organizations conducting gain-of-function research supported by the NH have an opportunity to transition the applicable research to research that is not covered by the funding pause, halt the applicable GOF research until the outcome of the deliberative process is known, or continue to conduct the applicable GOF research until the end of the currently active budget period. But they didn't stop. Now we can report that the leader of the U.S. task force overseeing the country's response to the China coronavirus, Dr. Tony Fauci, was the one who funded the Wuhan bat virus work and then kept it going. If these research projects were banned in the United States, was it legal and appropriate for Dr. Fauci and the NIAID to use taxpayer dollars to continue to fund the research over the next years in Wuhan, China? Was Dr. Fauci ever going to explain his conflict of interest? The article also quotes the Times of Israel, which reported, back in October 2014, the U.S. government had placed a federal moratorium on gain-of-function research, altering natural pathogens to make them more deadly and infectious as a result of rising fears about a possible pandemic caused by accidental or deliberate release of these genetically engineered monster germs. This was in part due to lab accidents at the U.S. Centers for Disease Control and Prevention, the CDC, in July 2014 that raised questions about biosafety biosafety at U.S. high containment labs. A CDC internal report described how scientists failed to follow proper procedures to ensure samples were inactivated before they left the lab, and also found, quote, multiple other problems, unquote, with operating procedures in the anthrax lab. As such, in October 2014, due to public health concerns, the U.S. government banned all federal funding on efforts to weaponize 3 viruses: influenza, Middle East Respiratory Syndrome, or MERS, and severe acute respiratory syndrome, or SARS. Now notice that the Times of Israel had no qualms about saying it like it was. These are efforts to weaponize these viruses. Remember, there is no difference between basic research, which they call legitimate, peaceful, friendly, etc., and biological weapons research. They are, again, one and the same, and therefore the Times calls it like it is. They are weaponizing these viruses. In the face of a moratorium in the U.S., Dr. Anthony Fauci, director of the National Institute of Allergy and Infectious Disease, and currently the leading doctor in the U.S. Corona Task Force, outsourced in 2015 the gain-of-function research to China's Wuhan lab and licensed the lab to continue receiving federal funding. Now remember, 2015 was the new fiscal year. The new budget was not, therefore, approved for continued research in the United States. He outsourced it to China. The Wuhan lab is now at the center of scrutiny for possibly releasing the SARS-CoV-2 coronavirus and causing the global COVID-19 pandemic. What more does Dr. Fauci have in his past that he has not revealed to the American public? Well, there's quite a bit, and that's been covered by other doctors and researchers out there, some of which who've been harmed, fired, put in jail due to Fauci's corruption. From the blog of Christina Lin at the Times of Israel, she reports why the U.S. outsourced bat virus research to Wuhan. A well-sourced article detailing the illegal outsourcing of $3.7 million of U.S. taxpayers' funds by Fauci's NIAID to the Wuhan Institute of Virology to conduct gain-of-function research on weaponizing coronaviruses, which likely led to the COVID-19 pandemic. But don't worry, it's for the public good. We can also read that article in the Asia Times from April 22, 2020, a U.S.-funded $3.7 million project approved by Trump's COVID-19 guru, Dr. Anthony Fauci, in 2015 after U.S. ban imposed on monster germ. The U.S. funded research into bat coronaviruses in the lab in Wuhan, China, that is now under scrutiny for possibly being behind the COVID-19 pandemic. Back in October 2014, the U.S. government had placed a federal moratorium on gain-of-function research, as we read. This was in part due to lab accidents at the U.S. Centers for Disease Control and Prevention, CDC, in July 2014 that raised questions about biosafety at U.S. high-containment labs. In the face of a moratorium in the U.S., Dr. Anthony Fauci, the director of the National Institute of Allergy and Infectious Diseases, and currently the leading doctor in the U.S. Coronavirus Task Force, outsourced in 2015 the gain-of-function research to China's Wuhan lab and licensed the lab to continue receiving U.S. government funding. It is understandable that the Chinese lab likely struggled with safety issues given the fact that U.S. labs share similar problems, and indeed, in January 2018, the U.S. Embassy in Beijing sent cables warning about the safety of the Wuhan lab and asked for help. Additionally, the embassy warned that researchers showed that various SARS-like coronaviruses can interact with ACE2, the human receptor identified for SARS coronavirus, meaning bat coronaviruses can be transmitted to humans to cause SARS-like diseases. Just not in nature. And now, in 2020, the U.S. is up in arms to hold China accountable for the global coronavirus pandemic, filing class action lawsuits domestically as well as building a coalition with allies internationally. Lawsuits have been filed within the U.S. and the International Criminal Court, alleging that China used the virus as a bioweapon, not that it is actually anything else, and other suits are underway at the International Court of Justice. Republican lawmakers such as Senator Tom Cotton and Representative Don Crenshaw have also introduced legislation that would allow Americans to sue China in federal court over the deaths and economic damage wrought by the virus. U.S. spy agencies are also investigating whether the virus originated in the Wuhan lab and seeking evidence that is needed to support the bioweapons of mass destruction theory promoted by Republican lawmakers. If evidence is found that COVID-19 is a biological weapon, which it is, and as we know can be nothing else, some pundits, such as Fox News host Lou Dobbs, have gone so far as to call for the U.S. to declare war on China. As for the U.S.'s creation and involvement in this scandal, it's total plausible deniability. As for Anthony Fauci and the rest of these so-called scientists, bureaucrats, politicians, and corporate monsters, all that's needed is to enforce the Biological Weapons Act, which states, whoever knowingly develops, produces, stockpiles, transfers, acquires, retains, or possesses any biological agent, toxin, or delivery system for use as a weapon, or knowingly assists a foreign state, like China, or any organization to do so, shall be fined under this title or imprisoned for life or any term of years or both. In short, the selective enforcement of this Biological Weapons Act and the complete purposeful ignorance of the Biological Weapons Conventions must end. These provisions must be enforced, lest we continue to suffer from these so-called accidental releases of biological weapons that most of us can't even imagine. The report continues that also, if there was a government ban in 2014 on federal funding being used for gain-of-function research, what are the federal compliance and ethical issues surrounding the fact that the NIH still gave federal funding instead of private funding to the Wuhan lab to continue the experiments? I can almost guarantee that one reason is to keep patent rights for later profits. Moreover, could some strains of the coronavirus have originated in U.S. labs, given the fact that the U.S. government lifted the ban in December 2017 on gain-of-function research without resolving lab safety issues? For now, President Donald Trump's administration is investigating the $3.7 million in taxpayer money that went to the Wuhan lab, while Republican Representative Matt Gaetz called for an immediate end to NIH funding of Chinese research. Since the federal ban on gain-of-function research has been lifted, U.S. labs can continue creating these monster germs domestically and would no longer need to outsource to China. Nonetheless, there still needs to be better oversight on the dangerous experiments and regulations over biosecurity of labs. Currently, the National Science Advisory Board for Biosecurity, a U.S. government interagency panel that advises the NIH's parent, the U.S. Department of Health and Human Services, conducts risk assessment of gain-of-function experiments that pose a significant threat to the public health. And it is that significant threat to the public health which we are being told is what's going to save us. The NSABB has given the HHS a framework to assess proposed research that would create pathogens with pandemic potential, such as research on genetically altering a virus to infect more species, or recreating a pathogen that has been eradicated in the wild, such as smallpox. It is important to note that the only way diseases such as smallpox can infect people in this day and age is because it escapes from a lab. There are many, many extinct or eradicated diseases that fall into this category. However, vaccine development and epidemiological surveillance do not automatically trigger an HHS review. In the post-mortem of the COVID-19 pandemic, this is likely a dangerous loophole that could be exploited with no oversight, and should probably be brought under HHS review in order to protect public health better in the future. Though not the end of this documentary series, the conclusion to this particular story, this current and possibly life-threatening history being made as we speak, should be considered only for what it truly is: a war against man and nature, against not only life as we know it, but against the very beautiful, wondrous, and indeed mysterious design of existence itself. Like it or not, we are all in this battle that is even now being waged against us, a struggle for our very own existence, in a war most of us don't even comprehend as being fought against us. Whether you personally identify with and understand your own moral obligation and duty to nature and its law through the Bible, or from such pop culture tomes as Lord of the Rings, Star Wars, The Matrix, or any similar tale of dark versus light, good versus evil, nature versus technology, or God versus the devil. The stories told are always at their root core the same. But here and now the story is real, and each and every man, woman, and child is part of the cast. And come to think of it, you've got to wonder if the movies that inspire these so-called scientists are movies like Revenge of the Nerds and Weird Science. Perhaps the greatest illusion we have all fallen under is indeed the institutional lie that technology is the same as science, that biology is technology. In fact, nothing could be farther from the truth. For technology is not a result of utilizing the laws of nature through the scientific method, but rather is the death of both. Much of our collective ignorance on these subjects delves from a systematic alteration of the language arts we all practice. Technology comes from the word art, and word or discourse, a description of arts or a treatise on the arts, an explanation of the terms of the arts. In other words, the so-called expert or professional languages that have to do with the arts that are being called as sciences. Art, that is, technology, is certainly not reality. It is certainly not biology, and it is certainly not scientific. To act technologically means pertaining to technology, that is, to the arts, as technological institutes, such as universities. To act technically is to act in a technical manner according to the signification of the terms of art or the professions. We have many men calling themselves scientists and professors, and yet what they profess are merely terms of art, not science. The word technical derives from the word art, or artifice, meaning to fabricate, make, or prepare. And this is the difference between science and technology. What is being called as science, but is really technology. In other words, it is man inventing things that do not exist in nature. In other words, artifice and fabrications. Technical means pertaining to art or the arts. A technical word is a word that belongs properly or exclusively to an art. Every artificer has his technical terms. It also means belonging to a particular profession, as the words of an indictment must be technical. In other words, the so-called scientists, that is, those with the flattering title of science that have nothing to do with natural science at all, those artificers are acting technical. Another word for that is technologist, like scientist, one who discourses or treats of arts, or of the terms of art. And that brings us to the word science, meaning to know. In a general sense, knowledge, or certain knowledge, the comprehension or understanding of truth or facts by the mind. The science of God must be perfect. In other words, natural science must be perfect. It must be proven by the scientific method, and it must be shown to be clear and reproducible. Unfortunately, the word science also relates to the arts. In the note here at the end by Noah Webster, he states that authors have not always been careful to use the terms art and science with due discrimination and precision. Music is an art as well as a science in general. An art is that which depends on practice or performance, and science that which depends on abstract or speculative principles. The theory of music is a science, while the practice of it is an art. In other words, these so-called scientists are practicing an art, not utilizing the theory of the scientific method. They are not practicing science, they are practicing an art. A principle in science is a rule in art. In other words, a truth in science is not a truth in art. Rules are certainly, as we've seen, made to be broken when it comes to the cult of scientism. He also quotes that no science doth make known the first principles on which it buildeth. In other words, when men study nature, it is not to understand creation, it is not to understand the source of that existence, whereas these so-called scientists practicing an art are actually trying to play God, if you will, in order to be the creators, to understand the first principles, which makes their work an art, an artifice, not a science. Their goal is not understanding, their goal is invention. In understanding, then, the meaning of science, we can then understand its polar opposite, which is, again, art. Art is the disposition or modification of things by human skill to answer the purpose intended. In this sense, art stands opposed to nature. These inventions, the synthetic biology and synthetic environments being invented by these so-called scientists, are not science, they are art. They are opposed to nature, its self-existence, and its law. Art is a system of rules, serving to facilitate the performance of certain actions, again, opposed to science, or to speculative principles, truths, as the art of building or engraving. Arts are divided into useful or mechanic and liberal or polite. The mechanic arts are those in which the hands and body are more concerned than the mind, which describes these so-called scientists acting like children with building blocks, rearranging and genetically altering things without having any idea what the consequences will be, and for that matter, not really caring. These arts are called trades. Being a so-called scientist, an employee is, of course, just a trade, having no requirement to utilize the scientific method. The liberal or polite arts are those in which the mind or imagination is chiefly concerned, as poetry, music, and painting. Art is also the skill, dexterity, or the power of performing certain actions acquired by experience, study, or observation, as a man has the art of managing his business to advantage. Now, one of the reasons these so-called scientists are called experts is because they have experience, because they've done the same thing in rote actions over and over and over again, and therefore, because of that so-called skill, they are experts. Again, that's like calling a child an expert at randomized block building. To be artful is to be performed with art or skill, artificial, as opposed to natural. What these so-called scientists are doing is opposed to natural science. They are cunning, remember, they are the smart people. They're practicing art or stratagem, and they are certainly crafty as an artful boy. And what they do proceeds from art or craft as an artful scheme. They are, for all intents and purposes, artificers. And we give them credit where credit is due, for they are artists. They are mechanics and manufacturers. They are people whose occupation requires skill or knowledge of a particular kind, as a silversmith or saddler. They make, they contrive, they invent. And the example here is artificers of fraud or lies. Can what is unnatural be called a fraud, a lie, or an artifice? Absolutely. That's the point. And finally, they are certainly cunning. Artifice, coming from art and fascio, to make, is a stratagem. It's an artful or ingenious device in a good or bad sense. In a bad sense, though, it corresponds with a trick or a fraud. Artifice is an art, a trade, a skill acquired by science or practice. Notice the difference here between those two words, science and practice. Anyone can be a scientist if they have no conscious morals, ethics, or values and simply practice the same action over and over again, observing the completely unknown results of their tricks. Again, what these so-called scientists are inventing is artificial, not of nature, or in other words, not a creation of God. They are made or contrived by art or by human skill and labor in opposition to what is natural, as artificial heat or light, an artificial magnet. Feigned, fictitious, not genuine or natural. Contrived with skill or art, no doubt. Cultivated, not indigenous, not real. Not being of spontaneous growth, as artificial grasses. Artificial arguments in rhetoric are arguments invented by the speaker, in distinction from laws, or natural truths, authorities, and the like, which are called inartificial arguments of proofs. And we certainly heard a lot of those from Ron Fauchier and other artificers plugging their work in gain-of-function dual-use research. And let's not forget the ultimate artificer, the great thunder, Anthony Fauci. An artificiality is the quality of being artificial, or the appearance of art, while to act artificially is to act by art or human skill and contrivance, hence with good contrivance, with art or ingenuity. From art, not from natural science, comes synthetic, or what is synthetical, pertaining to synthesis, consisting in synthesis or composition as the synthetic method of reasoning, as opposed to the analytical. To be analytical means pertaining to analysis that resolves into first principles, that separates into parts or original principles, that resolves a compound, body, or subject as an analytical experiment in chemistry or an analytical investigation, again, opposed to synthetic. To reach a synthesis, the action of putting or setting, is a composition, or the putting of two or more things together, as in compound medicines. This describes, well, all drugs that come from the pharmaceutical industry, simply because you cannot patent what comes from nature. You must compound things into new combinations. In logic, composition, or that process of reasoning in which we advance by a regular chain from principles before established or assumed, and propositions already proved, till we arrive at the conclusion. Synthesis is the opposite of analysis or resolution. In other words, using the child building random blocks analogy, these scientists are arriving at a synthesis, not a resolution or analysis. Their experiments have no principles. They are not based in truth. They are designed to alter truth, to alter nature. In chemistry, the uniting of elements into a compound, the opposite of analysis, which is the separation of a compound into its constituent parts. The water is composed of oxygen and hydrogen, for instance, and that is proved by analysis and by synthesis. So what's the difference? Natural science would be looking at things by taking them apart and examining their individual parts, while unnatural synthetic art is to put things together randomly without reason or principle in order to eventually find some desired effect, or, as we heard, to cause effects that were never even imagined in the first place. And finally, we come to the word natural, meaning to be born or produced. To be natural is pertaining to nature, produced or affected by nature, or by the laws of growth, formation, or motion impressed on bodies or beings by divine power. In other words, the law and laws of nature, that which is completely out of the control of man, or, as the insurance industry says, an act of God. Thus, we speak of the natural growth of animals or plants, the natural motion of a gravitating body, natural strength or disposition, the natural heat of the body, natural color, and natural beauty. In this sense, natural is opposed to artificial or acquired. All experiments being done as gain-of-function or dual-use research are artificial, meaning they are acquired. The results are because of man, not nature. Natural means according to the stated course of things. Poverty and shame are the natural consequences of certain vices. For instance, the love of money. What is natural is not forced, not far-fetched, such as is dictated by nature, and according to the life as a natural representation of the face, consonant to nature as opposed to being inharmonious with nature. Fire and warmth go together, and so seem to carry with them as natural an evidence as self-evident truths themselves. To be natural is something derived from nature as opposed to habitual. The love of pleasure is natural. The love of study is usually habitual or acquired. What is natural is very importantly discoverable by reason, not revealed as natural religion. In other words, the scientific method discovers nature. The scientific method is used because of reason, because there is a reason to use the method, a theory, though not revealed as self-evident, can be arrived at through true science. To be natural is that which is produced or coming in the ordinary course of things, or the progress of animals and vegetables as natural death, opposed to violent or premature. Now remember that one of the tenets of transhumanism is that death is somehow unnatural, that life itself should be expanded and possibly infinite. And of course, this must be forced. It is not derived from nature nor consonant to it. One's natural state, when not being influenced by all these artifices, is to be tender and affectionate by nature, unaffected, unassumed, according to truth and reality. None of these descriptions can be said to apply to what these so-called scientists and technologists and futurists and transhumanists are doing. When we get into the artifice of society, of the legal law, we find the opposite is true for the word natural, where it means illegitimate or born out of legal wedlock as a natural son. It is actually, to the elite of this world, an insult, and implies the word bastard. It means to be native, vernacular, as one's natural language, and this again refers to the aboriginals, the natives of lands that have not been tricked into the legal system, that do not worship flags and countries and men calling themselves kings and presidents. To be natural is something that is derived from the study of the works of nature as natural knowledge. Again, this is the polar opposite of art and the artificers undertaking gain-of-function research. Again, calling someone a natural as a noun or name is to say one is an idiot, one born without the usual powers of reason or understanding, or a natural fool, again, meaning a native or the original inhabitants of a land. Again, this is an insult from people who profess to be a higher or legal status. The Native American Indians, for instance, would be called natives or idiots because they do not subscribe nor wish to be a part of the legal law that controls the United States and its subjects. And this insulting word, natural, is exactly what these so-called scientists believe about the general population. They seek to be gods, and they seek to invent their own nature, one completely unnatural and unscientific. Now we can also look at the antonyms or the opposites for these words. For instance, the word synthetic, where the opposite of that word would be real, bona fide, pure, legitimate, and unadulterated, premium, valuable, and genuine, natural, and true. And yet the stated goal of these futurist so-called scientists is to create synthetic environments and synthetic biology in order to foment transhumanism. In order to do that, they must make us partly synthetic. And here the antonyms for biological. Again, illegitimate, supernatural, meaning something that is above or outside of nature. A surrogate, something adopted or unnatural, unreal. And, of course, artificial. And so we must question the very so-called science of synthetic biology. The two words cannot go together, and therefore, biology is certainly not technology. Life itself is not art. And finally, the antonyms for science. What is considered as non-science? Well, of course, ignorance, rudeness, representations, designs, disorganization, misconceptions, craftsmanships, disorder, illiteracy, misunderstanding, inexperience, shapings, and most importantly, though, is that word art, which is literally the representation of science as something completely unscientific. The truth is that we've all been placed under the spell of language, of legal opinion and tender, even to the point where we believe that corporations are people too. But they are purely fictions, artifices, invented by men to protect their unnatural evil intent through man-made unnatural laws designed to break with the law and laws of nature, to break with the law of God. And it is these artificial protections that keep each of you from fulfilling what is the true meaning of life. For the meaning of life is the protection, caring for, husbandry, and ensuring that life continues in its pure state of being, including your own. To attempt to alter nature and reinvent its laws is man's greatest folly. The truth is, you can't actually harm a corporation. They simply aren't real. You cannot harm a scientist, for the name scientist is merely a flattering title. Now, there is one more very important factor to consider here. The rationale, the fallacious logic behind doing these gain-of-function or dual-use experiments is because no other type of experiment can show these results. In other words, they are correct. The only way to know whether man can do such atrocious things to nature is to do so in a lab. The question you must ask yourself is how far are they willing to go? Because from what you've just heard, there appears to be no limits to the atrocities committed by these so-called scientists and futurists, transhumanists. And so, after seeing the various biological weapons and other testing that government has done legally and illegally in the past, there is one even higher truth that these scientists must also be thinking in the back of their minds, and that is, the only way to really know whether our gain-of-function experiments are truly as horrible and contagious as we think they are, as our animal and computer models show they are. The only way to really know for sure is to test it on humans. If you are fool enough to think that surely our government wouldn't do such a thing after seeing what you've seen in this documentary presentation, well, then maybe there is no hope to stop this. It is time for good men to step up and say no. It is time for good men to act upon their morals, their values, and I dare say it is time to uphold not only the biological warfare conventions coming from Geneva, but also the United States Code and the code of every other nation that forbids biological weapons and the so-called scientists that invent them. The corporations and institutions, calling themselves government organizations, cannot be allowed to continue in this vein. There comes a time in every man's life where he must make the ultimate choice. He must return to nature and its law, or allow it to be destroyed by inaction. We are at a crossroads, a time where history is either going to be made by the followers of God, of nature and its law, or by these artificers who seek to destroy all life in order to reinvent it in their own image. The choice, ultimately, is up to you. If the legal law of governments that are supporting these types of unscientific research projects, these dual-use weapons of mass destruction, if the same people supporting this research are the ones telling you they are the untouchables, well, then you no longer have a legitimate government. And so the only question left, the one that your children, if they live through this, will be asking you, is what did you do about it? In the next documentary of this series, we'll focus instead on what is the logical fallacy, the greatest excuse for doing this type of research, which is the creation of new, patented vaccines. In order to understand this greatest of all lies in the medical and scientific profession, we must uncover the truth about vaccines and why, perhaps, they are the greatest destroyer of human and animal health in the entire history of man. Look for Lethal Injection 2, The Corruption of Blood, which I'll be putting out very soon. Thank you for watching. And please do not let this knowledge fall on deaf ears, especially your own.